Uh, Today, we are actually having a international speaker from Baylor College of Medicine, Dr. Abhinav Sobe. So he is my actually long-term collaborator, and we had worked on many anti-aging compounds. So we had actually, while he was in during his PhD period, we had a lot of collaborative works with IIT Mandi and PGI Chandigarh. So thus it became actually long journey. So welcome, Dr. Abhinav. I now request our chairpersons for the session, Dr. Isabella Topno, Madam, Professor and Head of Pharmacology at Pondicherry Institute of Medical College, Pondicherry. With over 20 years of teaching experience, Dr. Topno is a prominent figure in the medical education. She has held key roles such as chairperson of Paraclinical Curriculum Committee and the Institution Ethics Committee. Additionally, she is a member of several committees, including the Board of Studies for MBBS and MD Pharmacology, and has authored numerous publications in national and international journals. Let's welcome her warmly to the stage. Please, ma'am. Okay. Welcome, Dr. Isabella Topto, madam. Our second chairperson for this session is Dr. Istisri Pustri, madam, Assistant Professor at SCB MCH Kotak, Odisha. Dr. Pustri is a life member of various renowned associations such as IPS-OSB, IPS, NEPTICON, and MPSCON. She has also contributed to the field with published research articles and is an active speaker in the scientific workshops on rational medicine use. Please give her a warm round of applause as she joins on the stage. Let's welcome our chairpersons with enthusiasm as they guide us through the session. Thank you. Welcome, ma'am. Rajkumari. Please take over the session, ma'am. Thank you. Hello. Hello.
A wonderful morning to all, and we'll start now the scientific session. As I'm introduce the speaker, Dr. Abhinav Chaube. Dr. Abhinav Chaube, MS Pharmacy, PhD, IIT Mandi, currently working in postdoc associate Baylor College of Medicine, Texas, Houston, USA. He has more than 216 citations and 7H index and more than 11 original research papers. And he won Best Abstract Award in Person in Nutritional Obesity Symposium and many awards to his credit. So please welcome Dr. Abhinav Chaube, and you can start your presentation. And he's presenting.
Yeah. Thank you, ma'am, for your kind introduction. Hello. Shall I proceed? Hello.
Yes, you can proceed.
Okay. Good morning, everyone. I'm here to discuss about one of our work about exploring therapeutic potential of muscle adaptive response during physical exercise. So, if you look at the pathophysiological chain of event for most of the metabolic disease, you will find these four features which are very relevant to almost every metabolic disease. And if you look the current therapy that are available mostly or mostly being targeted is at the management stage, where already the beta cells and all the state of the disease is very high. So much better approach than this is the beta cell protection stage, because at this stage you can see the decline of insulin production start happening. But if something has to be preventative, even earlier stage, like it's called diabetes or metabolic disease prevention, and this at this stage all these features can be targeted. So now question arises, like how will we target this stage at this particular stage, which either delay the progression towards the predisposition individual towards metabolic diseases, or prevent them from being towards that. So looking into the easiest thing is, like we thought to target exercise, because we know that exercise is the primary defense from aging associated metabolic disease. So if we actually ask question for long term as well as acute exercise, so I'm going to start with the long term exercise. So when we say that we should exercise, the next thing comes to our mind is what is the ideal timing of exercise. And current date, there are no any good research which can indicate like what is the optimal timing. So if you look into the ancient literature, some Indian literatures also, like so the every physical activity of 24-hour have been prescribed. Just for example, the last meal should happen before dusk and all, but there is no any mechanism like how why this has been prescribed. So in order to address this, we started with a model by exercising the mice on treadmill at two different times, one on the morning and another on the another at the evening. So we exercise them at two different time, and we are calling here at ZT. ZT is the, I think everybody must be knowing, it's a zetiver time which decides the phase of the animal housing. So this is dark phase and light phase, dark phase. So the in aim the animal was subjected to exercise at this time, which is ZT 23 to 1, and then at PM the animal was exercises as ZT 11. So when we exercise animal, we targeted muscles, because muscles we know that is one of the major organ which is involved in adaptive response to exercise. So we targeted
Sorry to
we
interrupt,
analyze.
sir. Sir,
Sorry.
slides are not moving according to your presentation. Sir, please see. Sir.
Okay, I will slow. Thank you for letting me
Thank you.
know. Yeah, so we conducted strength, endurance of the muscles. Why we targeted muscles? Because it is one of the important organ for adaptive response during exercise. And then we tested at different timing also, like in morning and evening, for all those animal which did exercise at two different time, morning versus evening. So here what we found
Dr.
that
Abhinav, sorry to interrupt you. Your slides are not moving, and
I am not
very
moving.
faint. And can we
Okay,
have a slideshow mode, please?
it is slideshow mode. Are you seeing my pointer? Could you see the pointer?
Yes, yes,
Okay, so if I am dragging the pointer, is that visible?
yes.
Yeah, so I am not moving slide now. I am just telling that. Uh,
Okay.
okay, I
Can
will
we
be
have it in
slow
slide mode, please? Slideshow
like
mode.
this. Yeah, it was in the slide. So now it is okay.
It's yet to show. Okay, okay, fine. Carry on, sir.
Okay, so after doing the grip strength and endurance and glucose intolerance, we come to the conclusion that exercise AM is much benefit than exercise PM, because we could see it is higher strength and higher endurance and improved glucose intolerance. And then thus this happens at the both time, and in the morning as well as in the evening. But the exercise who were subjected to the morning exercise were having better these muscles phenotype properties. So this result suggests that AM exercise is much better. So now we asked the question like, what is the role? So because this is time dependent experiment, so we decided to go to find some molecular clock gene in the muscle. So we generated a mouse line in which we did muscle specific reverb knockout. Reverb is a muscle clock gene, and then this time we used a higher instrument, higher technology to assess the muscle strength and endurance using aurora. Aurora in this instrument we can directly target the different types of muscles, and we put input as a current and it gives output as a torque. After doing this, we found that the aim exercise has better strength at both ZT and better fatigue resistance, you could see here, and improved glucose tolerance as compared to exercise PM. And this AM versus PM effect was gone in the knockout condition, muscle specific knockout condition. In every features, like the strength was not in, there was no difference between knockout AM, knockout PM. The endurance also there was no difference, and in GTT also there was no difference. So the main takeoff message of this result was that the time dependent adaptation to exercise in the muscle was dependent on the muscle specific reverb gene, because in absence of this the morning versus evening effect was gone. So before moving to any molecular mechanism, we were interested to know that is there any basal role of this reverb muscle gene, and we didn't find any basic fundamental disruption in wild type or knockout. Here knockout is again muscle specific reverb knockout. There is no any change in mitochondrial DNA or the oxphos, the um, and also any fiber structural differences in the muscles as well as energy consumption rate. So these all experiments suggest that they, at basal level, the river don't have any fundamental disruption. So moving again to the next step, like how river is controlling muscle, river is controlling the strength, endurance, and then glucose homeostasis. So we did a clamps study, and then we found that the mice who subjected to exercise in the morning has higher beam break. Beam break means more activity, and they have a higher oxygen consumption rate as compared to PM. And this morning versus PM effect was gone in the knockout condition. So again, the same thing happened, that in the wild type, AM is better than PM, and then when we did knockout in the muscle specific knockout, this morning evening was gone. So we deep down into the molecular mechanism, like how why this muscle specific reverb is controlling whole body dependent after exercise, adaptation after exercise. So we did transcriptomic analysis from the muscle in wild type and knockout mice at two different ZT22 and ZT10, and then we found that knockout has higher level of amino acid catabolism and fatty acid oxidation. These two pathways were upregulated in knockout, and glycolysis was downregulated. So we move to check whether we is there any binding affinity for this muscle reverb after exercise. So we found that after exercise, the reverb whole genome binding was very high. And we also did extension of this transcriptomic analysis, which is called metabolomics. In metabolomics also we found the same thing, that knockout has higher amino acid catabolism and fatty acid oxidation and reduced glycolysis. So when we integrated our transcriptomics and metabolomics data, we found the same thing, that the glycolytic genes were downregulated and the fatty acid oxidation pathway was upregulated. So the question was very simple, that what happened to the glucose uptake and fatty acid oxidation in vivo. So we did tracer studies using radioactive compound, and then we found that in knockout, the glucose uptake was reduced. As we could see that it was already downregulated, so it is proving again. And then the fatty acid oxidation was upregulated. So this is the mechanism how reverb is doing. When there is absence of reverb, the glucose will go down and the fatty acid utilization will go high. So the summary of this project end actually at the point where reverb is promoting muscle fatty acid oxidation, glucose sparing. In wild type, there is rhythmicity of the lipid glucose sparing, but in knockout, always the body is preferring towards the lipid. So if there is preferential towards lipid and someone is doing acute long-term chronic exercise, the endurance will be high. So but the scientific question, we close this story at this stage, and this is accepted. But the major outcome of this project can be extended through controlled reverb in phase and antiphase expression, and weather checking. If we modulate the reverb at different time, how we can maintain the adaptation to the exercise. So now going to the acute exercise, like how we can mirror the acute exercise pharmacologically. So this acute exercise story start with the transcriptomic analysis of the muscle from the acute bolus exercise of mice received the from the mice which received the acute bolus exercise, and we found that upon exercise various genes were upregulated. These are the top DGs which were mainly in the stress responsive lipid metabolism, carbohydrate metabolism, and some muscle growth. So we conducted RNA sequencing and we found that these genes are upregulated after acute exercise. When we also did grow-seq analysis. Why we did grow-seq analysis? Just to identify exercise induced enhancer in the skeletal muscles. We perform grow-seq, and this direction you could see the pattern of the increase of this. These all genes are same, and then the pattern in RNA-seq and grow-seq is almost similar. So it indicates that the closely nascent RNA transcription, or we can say transcriptional activation, underlies the gene expression. It means the genes which are nascently happen transcriptional activity happening at the we identified through the grow-seq is also reaching up to the gene expression changes. So we conducted motif analysis because our interest was to find some target, right? So that we can modulate and then we can try to mimic exercise. So we mapped the eRNA sequence from our grow-seq data, and identify these motifs were the top hit motifs, and these motifs are called as AP1 motifs. And the presence of these motifs were specifically on the genes which were upregulated after exercise. So I put few of the example here. HSP1, HSPB1, MT1, and BACH1. These genes were upregulated after exercise. So you could see here that the motif of AP1 is highly present on the enhancer of these samples. So this result suggests that AP1 could be critical initiator for exercise-induced transcriptomic response in the skeletal muscles. So to address this, like because how AP1 is playing role, we started with the in vitro model of exercise in which we expose differentiated C2C12 muscle cell line, and then we expose to electrical pulse stimulation which mimic the contraction to the differentiated muscles, and basically it is in vitro exercise. And then we validated whether all those exercise responsive genes are upregulated after EPS or not. So we could see here that after EPS the genes were mRNA expression of the genes were upregulated. Now coming to the questions like how AP1 is modulating this, because AP1 was the top hit motifs. So AP1 is a big family. It has so many subfamilies. So we individually knock down families and then checked in absence and presence of the electropulse stimulation EPS. So you could see here. So these are the different all 6, 7, 8 families of AP1. So after EPS, you could see again that the mRNA expression of this exercise responsive genes were upregulated. But interestingly, in absence of CFOS and JUND, these two subfamilies of AP1, the EPS fails to increase the expression of these genes. However, other family were not playing any role. This 4-5 family called as ATF3, FOSB, JUN, FRAP, and JDP, FOSB, L2, something, etc. So this happens in all of these gene. You could see that the third and fourth, it has JUND and CFOS guiding RNA. So, in absence of CFOS and JUND, EPS was not able to induce the transcriptional response. So, it means that CFOS and
Dr.
JUND are.
Abhinav, sorry again to interrupt you.
Yeah.
Slides are not moving.
Yeah, I'm not moving now. I'm discussing here. Okay.
Are we on the slide number 4 still? Because we can see slide number 4 as of now.
So this is like what you guys are
Thank you.
getting there. Now, is that yeah, the slide number 4 or figure number 4? This figure number 4, right? The hello.
13.
Yeah. So
Are we on the
I'm
right
discussing.
slide, sir?
Yeah, I'm the title of this slide is CFOS JUND contributes to contraction induced transcriptional response. Is that slide visible there?
Yes, yes.
Yeah. So using this knockdown of different individual family, we found that CFOS and JUND, these two were playing predominant role in exercise induced transcriptional response. So we try to inhibit CFOS in another manner. It is called AFOS. It is dominant negative truncation of CFOS, and we overexpress. And then we found that presence of AFOS, which is the dominant negative of CFOS, the EPS fails to increase the AP1 activity as well as the transcriptional response. So overall take-off message of this, the conclusion was that CFOS and JUND majorly play role, like they are playing role majorly in contraction induced transcriptional response. So we try to prove this at animal phenotype level. So we again induces neonatal AP1 loss of function through introducing AAV containing AFOS, which is dominant negative of CFOS, to muscle specific ML cremi. So once it is introduced, it will express only on the muscle, and it will leads to the AP1 loss of function. And then we test the muscle phenotype, and then we were planning to do the exercise, but unfortunately what happened, there were the at a basal level, because of the overexpression of AFOS, the animal shows severe disruption in the strength. As you could see, the wire hanging and grip strength of the mice was in the AFOS was very low, as well as endurance and the glucose intolerance also. So, but there was no any change in the structural level in the muscle fiber. So this, since it is a confounding factor, it was not a good model to go to take them to exercise and look whether in the absence of AP1 how exercise can be regulated. So to overcome this, we use another model of induction of AFOS. It is called inducible AFOS, and then we control through the doxycycline tetra, TET mediated approach. And then now we overexpress the AFOS, which is dominant negative of AP1. So it is basically adult AP1 loss of function. And now when we check at the basal level, there was no difference between blue and red. So this is basal. So there is no any difference. So now we use this model to exercise them and test whether how it is doing in the presence of AFOS. So here we could see that this grip strength and endurance was increased in exercise as compared to wild type, but in the presence of AFOS, which is the loss of AP1 function, it was abolished. So basically the presence of AP1 is very much required for grip strength, endurance. So, and also we targeted muscle mitochondria because we know that mitochondria play important role in energy production and maintenance of the muscle during the adaptation. So the same thing happened at the mitochondrial level, that uh, after exercise it was increased, but in AFOS it was not increased up to a good mark. So uh, this result suggests that AFOS abolish exercise mediated effect. It means AP1 presence is required. So same thing happened here when we did RNA-seq in this control exercise versus IFOS exercise. The same pathways that we see in the wild type was involved. So again, this hint us that okay, there is some crosstalk between the AP1 and exercise. So now what we try to do opposite, like what if we overexpress CFOS and JUNDI, whether we can mimic the exercise, like, because when we are removing it is not uh, causing the exercise mediated benefit. So let's overexpress and then see whether what is the consequence on the muscle in terms of the strength and endurance, and also glucose up homeostasis. So we what using the TET induced method, we activated CFOS and JUNDI. Here the construct is CFOS and JUNDI. So when we give doxycycline, it will be activated, but we give doxycycline 4 day on and 3 day off for the inducible, and another for the continuous we give doxycycline daily. So here there are two different type of overexpression of AP1. One is dox on dox off, and another is continuous dox on. So it will activate intermittent as well as continuous. And then we found that the strength and endurance was only increase in intermittent, not in the continuous. Same thing happened with the glucose tolerance, that when we overexpress the AP1 intermittently, the glucose tolerance was improved, and it fails in the continuous. Same thing happened to the muscle mitochondria. Uh, the we were interested here to check whether if we overexpress the AP1, can it mimic exercise in terms of maintaining the mitochondrial volume density. So here we could see, like, in wild type after exercise it was increasing, and when we overexpress intermittently it was also mimicking. And this is mitochondrial content, so it is also saying the same thing. So this result suggests that when we overexpress AP1 in intermittent manner, it can mimic exercise like benefit. However, activating continuously, it don't bring any such like any benefit. So we were surprised by this phenotype. So we try to prove the same thing in another model through transgenic activation of the AP1, because previously we used adenoviral mediated, AAV mediated. So it has toxicity and no uniform expression in the muscles maybe. So to just have one more proof
Dr.
of,
Abhinav,
yeah,
just 2 minutes.
yeah.
Thank you.
So we when we did when we overexpress, when we overexpress transgenically AP1 into the muscle, we found the same thing. We overexpress in two different manner, intermittent and continuous manner, using docs. So we found the same thing, like in the intermittent the endurance was high, fatigue was high, but in continuous everything was gone. Same thing happened with the mitochondria. So what we try to do, that we look for the transcriptomic response, like how intermittent versus continuous playing differently. So when we over in the IAP1 is red one, is the intermittent overexpression of AP1. The transcriptomic response was high, but it fails in the continuous. So we are planning to stop this story at this stage, because it is itself another question. Like, at this time we don't know that through the continuous activation, why there is no transcriptomic response. Because there is no transcriptomic response, there is no any phenotypic response happening, whether it is through desensitization or autoregulation. So it is a different question that we are going to ask later. But the main take off message of this work is, if you overexpress AP1 in intermittent manner, it will partially mimic exercise to the muscles in terms of strength, fatigue, and metabolism, glucose tolerance. If you activate continuously, this will not happen. So main extension therapeutic as a pharmacologist, because the conference, the most of the pharmacologists. So the main challenge will be to, because there are so many people who has limitations, who cannot exercise, they want to get benefit or because of several. So the main thing will be to target it at a clinical level, like how will we activate ap1 in intermittent manner in human. So this will be the question that a scientific community will be looking into, and that's it. Thank you.
Thank you, Dr. Abhinav, for your insightful presentation. Any question?
Okay, thank you, Dr. Abhinav, for your elaborative work that you have presented. I think there are no questions coming up. So we'll move on to the next speaker.
So to introduce next speaker, Dr. Ugachandhan Thangrajo. So he is currently Associate Professor AIIMS Raipur. He is a member of World Association of Medical Editors, and he is panel expert in Technology Development Board, Ministry of Science and Technology by Government of India. He has published more than 140 papers in reputed index journals, 5 edited books, and copyright granted 25 is. He has awarded DST, CRB, and CIS travel grant under Young Scientist category, and top second in the Scopus database of AIIMS Raipur in research and publication. He was elected as Fellow of Royal College of Physicians London, FRCP, Glasgow, FRCP, and elected as Central Council member of Indian Association of Leprologists. He was selected as member of Leprosy Focus Group of Clinical and Therapeutic Leprosy, member of Institute Research Committee, and also member of Secretary Animal House Facility Development, member Secretary of Drug and Therapeutic Committee. And he is presenting Bridging the Gap, translating lab research into clinical practice. So now is your time, Dr. Ugachandhan Thangrajo. Sir, you can start now.
A very good morning to each and everybody. Am I audible? Thank you, sir. So my topic of like a discussion today is bridging the gap, translating lab research into clinical practice. And because most of the persons are from for a postgraduate, there is a terminology translational pharmacology in their textbook. So this is a topic of interest as for a 10 mark, and we need to know whatever we are doing as a part of academic study during our UG, PG, or super specialty is a part of translational pharmacology only. Okay, so it is something we are differently looking into. So with this, I will just go. This will be my overview. Introduction, what do you mean by translational pharmacology? What are the components? What are the key steps, 5 key steps? And what a few things on in silico, in vitro, and in vivo models, PK/PD modeling, personalized medicine, and genetics, because these are all a component of this translational pharmacology, and a little impact, the limitations, and what is the way forward and where we are, and with a conclusion. So the scientific progress which we are seeing is not directly translated into the clinical practice. many things are coming up, and whether all are getting translated into clinical practice, that is a questionable and debatable. So there is always a delay in the drug development and implementation, except during the time of COVID, where we have seen a very fast activities. The rest of the times, it is there is a delay in drug development and implementation. So there is a need. This gap has to be filled. So there is a need for integration between the research, clinical application, and what the community needs. Okay. So for this, exactly the term is coming is translational pharmacology, and there are many terminology like it is like translational research, translational pharmacology, is translational medicine. Okay, it fits according to the area where we are going to look into. So exactly coming, what do you mean by translational pharmacology? It's a bridge between the molecular research and clinical application, and it aims to ensure rational drug use, safety, efficacy, and personalization. And more or less, it is an extension of the clinical pharmacology part. So it is a major gap bridge between the basic research with the which is entering into the clinical care or clinical research. So just we are looking into the traditional drug discovery pathway, like there are two phases like developmental pathway as well as the exploratory or the clinical studies. And for developmental pathway, it is a basic research, drug discovery and design and preclinical development. And exploratory clinical studies comes into the early clinical phases, clinical trial studies as well as the late clinical trials. So if you are looking into it, the basic as well as the drug discovery, it is done as the lab side. So that is a bench. Okay. And the early phase clinical studies as well as the late phase clinical studies, it is the bench to bedside. So they are actually the bedside. So there is always a gap which is happening in the preclinical development. So that gap we have to try to fill it. That the role of translational pharmacology comes. The components of translational pharmacology, if you're looking, as I already mentioned, that it is the first is bench, that is a translational laboratory research. Then comes the clinical practice, where the laboratory research is translated into the clinical care or clinical practice. And one of the major entities, whatever which is seen in the hospital setup, later it has to be translated into the community. Okay, so it is from bedside, and finally the community or the population needs. Okay, so this is the component, and everything is bidirectional. Like whenever something is happening at the level of bedside and it is going into the clinical side, there is always a mechanism of feedback. Okay, whether it is properly translated or not. And we have to look for the feedback mechanism. If there was some deficiency or gap, still we have to go back to the lab and we have to rectify that. So this bidirectional things we have to be very much, because every level we have to screen and monitor, and so that whether the product is properly achieved or not. So again, this is the repetition of basic things like basic research into the clinical research. There is a translation take place, and if we feel that the clinical research or clinical efficacy is not achieved, we can go back to the basic research and we can rectify it. And once the clinical research, which is coming with the endpoint, later can be translated to the population. So thereby increasing the population needs of the healthcare. So already I had told that there are 5 key steps. One is the drug discovery and molecular design, preclinical pharmacokinetic dynamics as well as toxicity studies, early clinical trials with the volunteers on the phase 1 or case-to-case basis on the patients also, evaluation of the dose, safety and efficacy, and clinical use and minimizing adverse effects. So these are the 5 key steps we have to look into as a part of this translational research. And there are phases of translational research. Okay, there are 4 phases, T1 to T4. What is this T1? It is the translation to humans. Okay, something we are making in the lab, and initially we are going into the proof of concept, everything. So the first entry is the translation to the humans. Then when we found that there was some efficacy as well as the safety is good, then we are going for the proper phase 2 or phase 3 and phase 4 clinical studies. With that data, we are introducing into the clinical settings of the population, clinical practice, and then we are looking into the cater the needs of the population. So this is the phases of translational research. So again, if you are looking into this picture, it is again the bidirectional. So whenever we are initiating some work, the basic research, that is a T0, that is a basic science research. Otherwise, it's a preclinical as well as the animal studies, where we are going to define the mechanism, targets, and the lead molecule. Then it is getting translated into the human. That is what the T1, that is I had told, proof of concept or phase 1 clinical studies, where we claim that safety as well as the efficacy and as well as the other diagnostic or other newer modality for the patient has been tested. Then it is entering into the T2, that is translation to patients, that is phase 2 and phase 3 clinical trials, control studies leading to the effective care. T3 is translation to practice, where that is a phase 4 clinical trial. And T4 is, as already we have told, that it is the population needs, and we are catering the population needs, and we are getting the feedback from the population. So if you are looking at all the phases are bidirectional, that is, we cannot take a decision at every level. Like we have to keep on monitoring and we have to screen whether it is workable or not. A few things which as a part of our PG or UG, PG as well as postgraduate super specialty, we can look into it, because we don't want to keep on saying that it is a part of a pharmaceutical company where they are doing. So whichever we are doing at the level of our academic clinical studies only, we can incorporate these things. So one of the things is in silico, in vitro, and in vivo research. Actually, based upon the in silico, it is a computational tools for early discovery. Yesterday, one of the speaker also was presenting a very good oral presentation of this, actually novel anti-tubercular drug. So now at the level of PG, people are getting exposed for this in silico model. Since the era of information technology and all the AI, it's very easy. We can do in silico, all the softwares are free, and we can do it within a week. We can, whichever we need to look into, we can able to make. Then in vitro and in vivo, that is validation and modeling. And the purpose of these things is we can cut short the time, and we can the cost which is being used for the traditional drug discovery, it will be like totally reduced. So this is the advantage of these methods. A few example I can quote. These are my publications only. Exploring the potential of Vintheria somnifera in leprosy and lepra reactions and a molecular docking approach. After my MD, I had the opportunity to work in Central Leprosy Teaching and Research Institute at Tamil Nadu. So I felt that there is a very big gap. Okay, because since it is a stigmatized disease, and WHO declared that we have eliminated, so there is no major research. But now even the Ministry of Health as well as the Ministry of Ayush, if you are looking, they are coming with lots of integrated approach. So this Vintheria somnifera is nothing but ashwagandha. Okay, so the idea came from a few of my patients. They started taking this ashwagandha. They are saying that, sir, prednisolone ke alawa, that is, I am feeling better if I'm taking this more than prednisolone for lepra reactions. So that has stimulated, and we have then worked out at the molecular docking, and we found that it is having a very good anti-TNF inhibiting property. So this is how we didn't go further for the evaluation of the safety profile, because we are not a proper trained Ayush doctor. Still, with the hearsay information, we have able to see scientifically. Next is like molecular docking analysis of Adathoda vesica with thromboxane A2 receptors and as well as antiviral molecule of possible dengue complications. So again, this Adathoda vesica is of a traditional herb like Benadryl. What we are drinking anti-tussio. Benadryl, actually the major component is vasicid derivative only, and this is from Adathoda vesica. Again, this is also hearsay only, and we have tried to look into the dengue as well as dengue complication. And it was also showing a very good result, but it has not been translated perfectly into the next level. Just here we have initiated the signal. And I'm not able to change. Yeah, ठीक है. So next, after Ayush, then we have entered into our core area only. This is actually trying a tenofovir M-triacetamin lamivudine. They are all anti-HIV as well as anti-hepatitis B drug. Okay. This we have explored for the anti-leprotic activity. Okay. Because an antiviral looking for a leprotic activity. Because see, this is how why I have planned this. During the time of COVID, there was a huge accumulation of all these drugs. No patient coming to the hospital. And I being the faculty judge of central pharmacy for whole AIIMS Raipur, I felt that these drugs will be wasted. So during that time, I got an idea and I just worked out whether there is any influence. During that time, leprosy drugs also in deficiency. So this study I have taken up and it was showing that among these three drugs, tell for leprosy, that is tenofovir, M-triacetamin and lamivudine. Tenofovir was having a very good anti-leprotic activity. Next is during the time of COVID-associated mucormycosis, you might be witnessing in whole India the availability of a liposomal amphotericin B or as well as the conventional amphotericin B. That too in Chhattisgarh, like we were like daily we will be calling the ministry only for giving 100 vials, 50 vials like that. Again, this drug was in huge quantity. So I was just, this is all done within a week actually. Okay, so then we have found that again dactylosvir has a very good docking effect for this mucormycosis. Next is, this is actually, thalidomide is my chocolate drug because whenever we are managing the case of type 2 ENL reaction in leprosy. So thalidomide, even though it is in the essential drug list, but the availability is again questionable. And I made this proposal for ICMR also, like pulithalidine, like a thalidomide as well as DEC for severe COVID syndrome. But it was not selected. But we had done a molecular docking study and it was showing that the congener of thalidomide, lenalidomide, it was showing a very good property. Like because people are beginning remdesivir, people are beginning tocilizumab, interleukin 6 during the time of COVID. And I was going back to a older drug for the severe COVID syndrome. So this is also very good because that is what, whatever there is a deficiency, whatever there is a gap, we can able to put our mind and we can take it up further. And this is during my MD time in PJ Chandigarh. Like our team, actually, Polan Sharma is also here. Like we have planned this green tea and its component for ulcerative colitis. And this is also a translational research only because we went for this in vivo as well as in silico modeling. So coming to the next aspect, this is PK and PKPD modeling. So how this translational pharmacology is going to be useful is, like I will just, it helps it links the drug concentration and effect over time. It helps in the, enables the safe and effective trial designs and ensures appropriate dosage as well as response. There is a terminology known as, like, I will go with the example only, model informed drug development for antimicrobials. Okay. This apramycin is the drug which is being used in the veterinary medicine, and they found that this drug is having some potential to be effective with the resistant bacteria. So, by using this model informed drug development, like they are getting all the pharmacokinetic data from the, because it is used in veterinary medicine. So all the veterinary data was there. So they have used this modeling and converted into the mathematical as well as the statistical, and they have seen that this could be a very good potential candidate for drug-resistant gram-negative bacteria. So this is also a part of a translational because at every level, from in silico, next at the level of pharmacokinetic and dynamic, we are looking into. Next is personalized medicine. What is the purpose of translational research? Is mainly targeting the personalized medicine only. Okay. Personalized medicine as well as genetics. How it is going to be useful? It is a therapy based on genetic as well as phenotypic profiling. It helps in reducing the adverse effects and it enhances the treatment outcome. So that is the concept of this personalized medicine. And if you are looking into the European Council conclusion on personalized medicine for patients, what is this? It is a medical model using characterization of individual phenotypes and genotypes, example molecular profiling, medical imaging, lifestyle data for tailoring the right
Dr. Fuga
therapeutic-
Chintan, just 5 minutes more.
Yeah, therapeutic strategy for the right person at the right time and for determining the predisposition to disease and to deliver timely and targeted prevention. So this is the definition of the personalized medicine. And somebody is suffering from an illness and we are looking into their genes. Accordingly, we are developing a model in the silico in animal or a computational, then we are making a drug and find who is a responder and non-responder. If they are responder, then the clinical trial has been initiated. So these are all advantage nowadays because using the models. This is my study only. That is pharmacogenomics, which is we are translating into the translational efficacy of sulfonylurea in patient with the polymorphism of KC and J level or ABCCE8 genetic locus, which is a prospective cohort study in the Chhattisgarh type 2 diabetes population. So, like pharmacogenomics are the personalized medicine, because we in the department of medicine, like there were cases who are not responding for sulfonylurea. Then we started working on this one actually, and luckily in 100 patients we can able to get only 5 patients with the SNP, single nucleotide polymorphism, and only one person was not having a proper control on diabetes. So we felt that our population is responsive with sulfonylurea. So which is the need of that population also, we can use this translational pharmacology and we can look into it. And my next speaker will be speaking on this use of real world evidence in translational pharmacology. And this real world evidence, not clinical study, clinical trials. Rest of the things after phase 4, whatever the data we are collecting, that can be used for the repurposing of the drugs, and we can come up with a good idea with the real world data. So sir will be speaking on this. And there are few how it is going to impact the stakeholders. Okay, academic institution, industry, as well as they like it is going to be a good choice in the academic institution, because in vivo silico modeling software, everything can be very well purchased, or even if it is free also, it will be very easily available in the most of the academic institution. So what could be the limitation and challenges? Limitation, definitely ethical concern, because if something we are doing, because when I was planning this Ashwagandha, there was lots of objection. You are a clinical person and you are working on this, but my team member is from our Ayush department. So there are ethical, regulatory and financial hurdles are there. Academic industry initiative disparity is seen. Infrastructure workforce limitation, because in few of the AIIMS, for example, AIIMS Bilaspur or AIIMS Bhopal, they have a translational medicine department. I mean. So infrastructure is also one of the important criteria to develop. This is an example, whatever I have discussed, how we are coming with a SLG2 inhibitors in type 2 diabetes. So initially there is a basic scientific research foundation of therapeutic innovation, renal glucose transfer, that is the understanding glucose mechanism. Then medication lowering blood glucose, SGLT2 inhibitors, then clinical trial, then for the health needs. Just whatever I have told, the same thing. In various phases. Preclinical, we have targeted a receptor. Then comes the translational step. That receptor, there is a development of few drugs. Then comes the clinical trial. What we have seen the HbA1c level reduction. Then later reverse translation. What we have tried for diabetes, later it was used for non-diabetic condition for cardiac failure. So this is how translational research will translation pharmacology research will end up. Cycle is bench, bedside, community, get feedback at the every level, and it is a continuous as well as iterative loop. Way forward, we have to encourage interdisciplinary collaboration, improve training and infrastructure, and promote policies for shift integration. It is happening, but it takes another 4 or 5 years. Like maybe at least at the institute level, it will be initiated maximum. So with this, I'm concluding. Translational research, it bridges the research as well as the care, drives modern patient-centric therapeutic effect, vital for efficient evidence-based healthcare delivery. So thank you. Thank you.
Thank you so much for the elaborate and wonderful presentation. It is well known that pharmacology is not a limited area that we can work on. We can expand to any limit.
Yes, ma'am.
And this is what I can see from your work, that anything which is not responding, you are going deeper into it, and you are looking ways and means how that particular can be useful. And yes, any questions to be asked? Okay. Yeah, one question here.
So, you repurpose the antiviral drugs for lepra reaction.
Yeah.
So, I just wanted to understand what is the thought process which went behind
Yeah.
this.
See, actually, essential is the what is this? Essentiality is the mother of invention. Okay. At that time, when we are looking at, for example, I was working leprosy areas for several years. There are period where we are not getting thalidomide on time, and there are many cases with dependent on steroid dependent or the steroid totally steroid dependent. Because of that, they have a few cases they didn't die of leprosy. They died of drug-induced complication of leprosy. Steroids, the patient enter into a pyelonephritis, then he went for a nephrectomy, then he went for sepsis and died. Okay, then I felt that there is a need for extension of the some other modalities we have to look into it. This is for reaction phase. And when we are looking into currently also the WHO as well as National Leprosy Eradication Program, they are coming with lots of, even though we have a established MDT, still there are a percentage of cases being noticed, and government is looking for a single level of drugs, going with a single dose of rifampicin, or they are going for PE, PP plus, post-exposure prophylaxis, double plus, like that. So these are the orientation like I was looking into. At that time, being a faculty in the Central Pharmacy, I am seeing a huge quantity of other drugs which is being accumulated. I was just trying whether this drug can be repurposed for the deficiency area. That is all.
No, why only anti-HIV drugs came into your
No, no,
mind? Why
actually,
not others?
selection
So
of
that's
drugs.
what.
Selection of drugs is very much important. For example, randomly, I thought that I have to go with one antiviral. Then we looked into the profile of the antiviral, which is having a very good safety.
Okay.
Which is having a very good safety. I selected like that. Then I had talked with our leprotic antigen.
So in that case, going by the safety wise, we have anti-herpes drugs also. So, why only you selected
That is
anti?
what, ma'am. What first is availability of the drug, which it was in my
Okay,
setting.
okay,
All the anti-HIV as well as hepatitis B drugs were in huge quantity.
okay, okay.
Okay, and we are not majorly handling herpes. So I saw the drug. Okay, why can't we go with this? Like that happened.
Okay, thank you.
Any further questions? If no other questions, we can move on to the next speaker. Thank you, Dr. Tangaraj.
Thank you. Thank you. I can take it.
Thank you, sir, for the wonderful presentation. Now I request our chairperson and MSP members to felicit the most promising MD Pharmacologist Award to Dr. Pugazanthan Thangaraju, sir.
The next speaker we have in this session is Dr. Melvin George. To introduce Dr. Melvin George is a professor and head of Center for Clinical Pharmacology and Center for Clinical Trials and Research, SRM Medical College, Chennai. He did his medical studies, all of them, MBS, MD, and DM Clinical Pharmacology from JIPMER, Pondicherry. He has conducted several clinical trials as an investigator in acute coronary syndrome, primary prevention of coronary artery disease, adenomyosis, COVID-19. He has published a total number of 125 publications in various indexed national and international journals and has a H index of 23 with 1,500 citations. He has won several research grants from governmental and private funding agencies. And last but not the least, he is the founder, treasurer of Medical Pharmacologists Society. So here we present to you Dr. Melvin George. Over to you.
Thank you, ma'am, for the kind words of introduction. At the outset, I wish to thank Medical Pharmacologists Society and Dr. Shivamurthy in particular for giving me this opportunity to speak on this topic of real-world evidence in drug approvals and post-marketing surveillance. So before I start, I would like to acknowledge my mentors, my teachers. I think for those who are youngsters who are probably doing PG, you might not know the people who are on the screen. But of course, the senior people here will know who are these people. So there is Dr. Adityan sir, my guide, and who was a national icon as far as pharmacogenomics was concerned. I have learned a lot through working with sir over the years during my education in JIPMER, and I'm very thankful to him that the seeds of what research is I learned by working with Dr. Adityan. And I'm also thankful to 3 other faculty in the Department of Pharmacology, JIPMER, that is Dr. Ravindran, Dr. Geetanjali, and Dr. Shivade, who are very, who stood really like pillars in making me, inspiring me to take research as a career seriously. And that is what gave me the confidence later to carve a path that is different from what is usually done after education in pharmacology. So I'd also especially thank 2 clinicians in JIPMER who trusted me to design and participate in clinical trials as in different capacities during my DM days. One is Dr. Santosh, a cardiologist above, and the other is Dr. Sunil Narayanan from neurology. They trusted me enough that certain aspects of the trial that they were involved in, these were trials which they were already involved, where the sponsor was a university outside the country. They trusted me enough to take care of some part of their study. And that gave me a lot of confidence that when I even finish my education, I should look into this pathway of the journey into clinical trials. And I'm happy that over the last 13 years, I've been involved in several regulatory clinical trials, long-term cardiovascular clinical trials in particular. And some of those trials have been instrumental in FDA and other regulatory agencies bringing such drugs into the market, changing certain guidelines. So that has been a great learning experience. So today what I'm going to talk about is, it's a little ironical because today what I'm going to talk about is just the other spectrum, and that is not clinical trials. It's about real world evidence. So when we talk about evidence, everybody requires evidence when it comes to deciding whether a drug should be given to a patient, whether a drug should be in the market, whether a drug is worth reimbursing for an insurance company. So this evidence for a long time, for a very long time, the time-tested methodology of gathering this evidence has been clinical trials. It has been the default option, in fact, to gather evidence. And ever since this thalidomide tragedy happened, there was a major shift in the major amendment that came to the FDA regulations, where they said every drug should go through efficacy and safety testing through clinical trials before it is marketed. That was the Kefauver-Harris amendment. And ever since there, for the last 4 to 5 decades, we see drugs in the market only after it passes through at least well-designed 2 or 1 randomized clinical trial in a large population. That is the phase 3 pivotal clinical trials. So clinical trials are ideal because they are still considered the gold standard of evidence-based medicine, because they are ideal in minimizing the bias. Every research is going to have bias. And among all the different study designs that are used, the one that has least chance of introducing bias is the randomized clinical trial. Obviously, the data that comes out of a clinical trial has much better quality, because it is going to be, it is constantly under the radar. There are people who are employed to monitor the data, the quality of the data, and there are pre-specified analysis that are put in place. There is a clear, well-defined protocol, and people are expected to follow the protocol very rigidly, where there is no room for deviating from the protocol. So all that essentially means that the data that generated out of a randomized clinical trial is usually of very high quality. And as a result of that, it has been used by most regulators in judging whether a drug should be in the market. They take these decisions on the basis of the benefit risk, based on the data that is coming from the clinical trials. But at the same time, sometimes these clinical trials, the questions that are asked in clinical trials, if you have to do it, some of the research questions that we have, if you have to actually embark on a clinical trial, it would become a little unethical. And that is a time when you would have to rethink on whether you proceed with a clinical trial design. There are sometimes clear-cut logistical challenges. Because of those logistical challenges, it might simply not be possible to do a clinical trial. And most clinical trials, when you look at the primary endpoint of the clinical trial, it is always the efficacy endpoint that is chosen. Trials are powered based on the efficacy endpoint. As a result of that, what happens is the trial is not actually powered to capture enough adequate safety data. And so one cannot have that much confidence to talk about the safety of a drug in a clinical trial when compared to its efficacy in most of the situations. Of course, there are a few instances where trials use primary endpoint as safety, but that is the exception, not the rule. And we all know that the results that come out of clinical trial also do not necessarily match clinical practice. There are a lot of instances of drugs which have found superlative evidence in clinical trials, but then when the rubber hits the road in real practice, it is not anywhere near the claim that it had in the phase 3 clinical trial. And why is that so? There are many reasons. One of the obvious reasons is because the population that is chosen in a phase 3 clinical trial is usually a very neatly defined cohort, a group of individuals whom the sponsors believe are going to have the maximal chance of responding to the drug. And that makes it easy for them to show efficacy of the drug for the trial. But then we know that in real world, people do not come just with a single disorder. They have multiple comorbidities, multiple other medication being received, and all that ultimately makes it a ground where the drug does not really function or does not have that much of a efficacy as seen in the trials. So it might not be a representative population. So, and the other aspect is patients usually when they are part of clinical trials, they are followed up very, very religiously. There are people who are called clinical research coordinators who keep on calling them, probably pestering them to take the drugs, to see whether they do not miss any schedules, to see that they actually consume the drugs, to help them when there are any challenges that they face. So we have this extra support system that is there in a clinical trial for every patient. And all that makes a patient to consume the medication regularly and to follow even the other instructions that need to be followed while they have the problem, the disease. So this is a very artificial environment that works in favor of the patient and the drug. Okay, so it's good for the trial, it's good for the sponsor, but it does not really match the real world. So that is an artificial environment of clinical trials. So that is why we need to look at something beyond this, and that is the real world data. And real world data is obviously something that has been used for several decades. Again, if you look at it in the context of safety, because we have always been having post-marketing surveillance studies. So in that sense, real world data has been used. So there's nothing novel about this, but it's just that now the thinking has changed, that regulators across have understood that there is much more to it than just looking at safety data. And so what exactly is this real world data? Real world data is basically data obtained outside the context of randomized controlled trials generated during routine clinical practice. So there are several definitions. I found this definition is the simplest. So any data that you take which is outside RCTs, it is automatically a real world data. So, and real world evidence. What is real world evidence? It's insights gained from data collected outside traditional clinical trials. So what are the different sources of real world data? So we have data that can come from electronic health records, data coming from patient registries, patient-generated health data, administrative data, observational cohorts with primary data collection, biobanks, health surveys, interviews and focus groups. So there are multiple pathways by which you could get real world data. And obviously the real world data is not like what you see in a RCT where it's a tightly controlled environment. These are people having different, it's a very mixed population, a diverse, heterogeneous population, taking treatments, multiple treatments. And so it is not the homogeneous clinical trial population that we see in RCTs. So what exactly is the advantage of this real world data and taking this data in the context of making decisions when it comes to real world evidence? Okay, so number one is there is a possibility of getting an in-depth understanding of the disease and treatment. Sponsors always have a challenge with clinical trials. In fact, if you look at India as a country with respect to clinical trials, most of the companies in India, that is Indian pharma companies, are not venturing much into drug discovery and drug development. And one of the reasons that happens is because of the prohibitive cost. Doing a clinical trials costs several $1 million, so it is not feasible. And that disadvantage can be actually, to some extent, that can be removed when you are having the possibility of using real world evidence where you have cost effective studies that can be planned when you're using real world data. And you could also evaluate patient outcomes, could evaluate cost effectiveness and the quality of care. You also get insight through diverse populations, as I already mentioned. And obviously, because real world data we are talking about big data, so the chance of you picking up an adverse event, especially the rare adverse events, is much higher when you employ the real world data. And you can actually look at the real patient behavior when you look at RWD. So as I mentioned earlier, clinical trials are not the real deal because of the fact that it is an artificial environment. Okay, so how does a patient really behave in the world when he's prescribed a drug? That is something that you can see when you do a RWD study. Okay, and many times in clinical trials, they do something called a sub-analysis or subgroup analysis. And this subgroup analysis many times will not have the enough power to look at that particular outcome because that will be a smaller population. Okay, so all those kind of subgroup analysis can be done more effectively when you're dealing with real world studies because of the large, phenomenally higher sample sizes. But however, these real world evidence studies, they also have their own limitations. The obvious limitation is the fact that the data quality is not obviously optimal. In no way can a real world data set match what you get from RCTs. Okay, for example, comorbidities. You obviously will have a lot of comorbidities that can affect an outcome, but it is possible that all those comorbidities are not documented in the RWD, but obviously that would be available in a clinical trial because documentation is done far better in a clinical trial context or even dosing information. Selection bias of patients is obviously possible in a real world data. With safety event reporting, because it is not mandatory in a real world context, there is a high chance of underreporting happening when you're dealing with real world data. That is not the case in a clinical trial because of the stringent guidelines on safety reporting, especially SAE reporting and all that. So you're unlikely to miss a safety outcome when you're dealing with RCTs, but that's not the case with real world evidence. And when you're talking about newer drugs, since there's a lot of focus on the new drug, there's a higher chance of the AE reporting happening. But once a drug is in the market, because it's already in the market, the focus is not that high. As a result of that, there is a slightly lesser chance of a patient reporting or even the healthcare worker reporting the adverse event. And reporting can be influenced by whether the event has been already described in the literature. And inconsistent definition of diagnosis is an important thing, I should say. If you look at most clinical trials, they very clearly specify in the inclusion criteria how is the patient, what kind of patient is going to be taken, and on the basis of what definition of that diagnosis. For example, acute MI as defined by the American Heart Association guidelines 2022. So it's very clear what they are referring to as a patient with MI. But in real world, we know very well that when a clinician makes a diagnosis, not necessarily every clinician in the hospital is following a rigid protocol when it comes to making a diagnosis. So these kind of things can make inconsistencies happen in the context of real world data. So regulators have obviously understood that there is a lot of value to real world evidence. And that is why a watershed moment happened in 2016 when the US FDA said, when the US 21st Century Cures Act came into existence, one of the things that they said was we should give importance to real world evidence henceforth. It should be used as a supportive tool for regulatory decision making. Okay, and subsequent to that, there are several documents were released by the US FDA as guidance documents to make the to give more clarity to sponsors as to how they can best utilize electronic health records, how they could best use registry studies when it comes to showing evidence for drugs. And subsequently, even EMA has issued guidelines on real-world evidence, on using registries, and that is for many other countries, including Therapeutic Goods Administration from Australia, Japanese regulatory authorities, and Chinese regulatory authorities. All have now joined the bandwagon of including RWD as a part of evidence generation in the context of decision-making for drugs. And this is just one example to show how, where they have evaluated 3 drugs, okay, cardiovascular risk of urate lowering drugs. 3 drugs were taken, okay, that is febuxostat, topiroxostat, and allopurinol, to see what was the cardiovascular risk. Was it higher with any of these drugs? And for that, they have used the insurance claims data, okay, that was taken up. And at the end of that study, what they found was there was no much difference in the cardiovascular risk with the 3 drugs. Okay. So how can real world evidence be used in the context of drug discovery and drug development? So I'll just talk about this and close. So real world evidence can be used to identify how many patients currently suffer from disease alongside other comorbidities. For a sponsor to make decisions at the start when he has to venture into the drug discovery program, this is a very crucial data for obvious reasons. To identify the current standard of care for the disease and assess how much of adherence is there to these guidelines, for that real world evidence becomes very useful. And to identify how many patients with a given disease are insufficiently controlled or have inappropriate treatment for their characteristics. All this can be easily obtained through registry studies, through electronic health records, and that can help people sitting in the boardroom to make choices as to how to position their drug which they are planning to launch, which they're planning to take up for the drug discovery program, okay, moving forward. Real world evidence helps in identifying how current pathways might be affected by a new drug, treatment pathways. It can also help in identifying the cost associated with the current unmet need that a new compound will address. And also in the context of planning a clinical trial, how to refine the inclusion criteria, how best to take patients who are most likely to benefit the treatment, that is to identify the target population, we could use real world evidence. To gather data about genetic and biomarker information for, again, selectively identifying patients who are more likely to respond to treatment.
Slept.
Yes, ma'am, 2 minutes. And to use historical or synthetic controls to replace contemporaneous controls. So this is, nowadays, there is this concept where a trial is identified and they use a single arm in the trial where there's just one group receiving the new drug and there are no controls. Instead, controls are used as part of the external controls. That is, historical controls are used, which is from the real world data. So real world data is used as a control and the new drug, that is a test drug, is given to all patients. So this is a very new thing that has been increasingly used and even accepted by regulatory authorities. So to summarize, I would say that RCTs increasingly are being accepted by regulatory authorities. I mean, always been accepted by regulatory authorities, whereas in real world evidence, it is not so far, but increasingly being used. In terms of internal validity, obviously a RCT has a high internal validity, whereas a real world study does not have that much of internal validity. And external validity is much higher for a real world study because it reflects the real population. Potential for confounding is obviously higher in a real world study. And the chance of detecting rare events, evaluating long-term outcomes, evaluating healthcare professionals' behavior and healthcare delivery aspects, all that is much higher in a real world study. So to conclude, I would say, to summarize, I would say, RCTs are the gold standard of evidence-based medicine. They sometimes overestimate the treatment effects, and that is when real-world evidence could complement RCTs, including data from individuals who will probably not fit the inclusion criteria of an RCT. And there are a wide range of sources for developing or identifying real-world data, and they offer high external validity, although the internal validity is relatively lower. And regulators across the world have started using it as a judicious approach to combine evidence from RCTs along with RWT. Thank you for your patient listening.
Thank you, Dr. Melvin. That was elaborate and a wonderful presentation. And I think this is another area where we can explore and work on it. Just wanted to know about this real world evidence. Can it be taken as a post-marketing surveillance?
And post-marketing surveillance is one form of real-world evidence, but it is post-marketing surveillance is not the only real-world evidence. Apart from post-marketing studies, nowadays, because post-marketing studies are traditionally being used to identify the safety of a drug, that is a mandatory requirement that a regulator gives to all sponsors. But nowadays, we are also looking at registry studies, electronic health records being used to identify signals whether a drug also has certain efficacy. Can this drug be repurposed? For example, pembrolizumab was originally used for certain cancers when it was approved by US FDA. But then using real world data, they found it could also be used in other cancers. And that was how the drug repurposing was done. And on that basis, it got additional approvals for other cancers also. So it is not just post-marketing surveillance, but many other data that is taken up.
Yes, Dr. Melvin, thank you for your excellent presentation. And it's what you said, that it plays a vital role in both drug approval and safety of the drugs or medical products. And is any question from House?
Yes, one question is there.
Yes, sir.
Can we have a mic there, please?
Can press the.
Excellent coverage, very interesting topic. I just have one comment that how to address the issues of bias as well as the other thing is about adherence, patient adherence, which can also impact on the conclusions that you derive. Real world evidence studies by nature being observational in nature and not interventional. So you have mentioned about the issues of confounding variables, which can also contaminate the conclusion. But then also the issues of biases as well as the uh, adherence, patient adherence. So if you can have a couple of words on these two areas. Thank you.
Thank you for your question, sir. So in the context of bias, when data is gathered from real world data, we obviously are having certain statistical tools, like for instance, propensity matching and many other tools, which can to some extent try to pick up signals that can overcome these biases that are there because of the confounding factors and all that. So that is one thing that is done. So there are statistical tools that are used. And when it comes to adherence also, that is documented, but then this is what is the real world. Like if a patient is given 100 medication, in the real world, they do not take treatment for with all the 100 tablets. They probably, there's going to be 70% adherence or 65% adherence. So what is real world is reflecting in that data. So that is one thing. And USFDA, and not only USFDA, all regulators, they also judge the quality of the data. And many times, data are also rejected because the quality is not good. So there is a mechanism that regulators have to check the quality of the data that is coming from a real world study. So if the quality is not adequate, a lot of instances, USFDA has rejected such data. So it's not that every RWD data that is submitted to the regulator is happily taken. There is a rigid process of evaluating how good is the quality. If the quality is good, it is used as a supportive tool in taking the decision.
Thank you. What about the sample size?
Yes, ma'am. So when it comes to sample size, there is a advantage in real world data because of the huge volumes that you get, because you are dealing with electronic health records. insurance databases. So larger sample sizes definitely help us in getting a stronger signals of efficacy and safety. That might not be possible because of the prohibitive cost in an RCT. Many RCTs will have to take into consideration the budget aspect, and we cannot just take a very large sample size in a RCT. But that possibility is there in a real world evidence study because you're just looking at records. So that is one thing where RWD can be stronger.
Any more questions?
Madam, I would like to add some. Uh, Targeted or a subgroup where the bias is creeping in, it is not regularly thoroughly pervasive. It is located to some subgroup. It can be detected by bootstrapping techniques, and it is widely used in this real-world evidence data. And many more techniques. Sorry, talking about that. I am doing my work in bootstrapping techniques. They rectify by themselves that after this 5,000 or 10,000 data, niche data, what is the probability it should be? If it is widely outlier from the real situation, then there is a suspicion that signal the sir is talking about, the signal is generated. And many a times, if it is repeated by a subgroup, the whole data set is rejected. It happens so. Thank you, sir.
Thank you
Interest
so
of
much, sir.
time, I request to go ahead with the next presentation. One more thing before that, I would like to appreciate Dr. Melvin. He is the one of our founding members of our society, and he is consistently contributing to our society, and he is also excelled. He is like currently he is the director, I think, head of clinical research center at SRM. One is congratulations to his recent promotion. And at the same time, we would like to tell that he is also a 2% scientist throughout the world. I think he's the top 2% scientist. It's not it. It's not it. Okay, I think you will reach. We wish you. Okay. And with this, I request everyone to give him a standing ovation for his contribution towards clinical research, and hope that he will achieve more in his future also. Thank you. Please, sir. Now we would like to proceed with the award ceremony. So he has been confirmed with MD, a most promising MD. Pharmacologist, senior, MPS, most promising senior MD pharmacologist award.
Now we need to explain. Chair person. Sam Matthew. Dr. Sam Matthew. You can felicitate the chairs.
Okay, Sam. Okay. Now I request Dr. Sab Matthew to felicitate our chairpersons. Sir, please. So moving to our next session, I'm excited to introduce Dr. Farhana Rahman, Professor at Sri Balaji Medical College and Hospital, Chairperson, Chennai. Dr. Rahman completed her PhD in 2023 on the topic of pharmacogenetics and hypertension and is currently leading a mangrove research project funded by the Tamil Nadu Forest Department. Her contribution to research and education are highly commendable. Let's give her a warm welcome. Our next chairperson for this session is Dr. Smita Das, Professor of Pharmacology at AIIMS and SUM Hospital, SOA University, Bhubaneswar, Odisha. Dr. Das has over 10 years of experience in teaching pharmacology and has published extensively in both national and international journals. She is also actively involved in pharmacovigilance activities. Please join me in welcoming Dr. Das to the stage.
So, good morning, everyone. So, I would like to invite our first speaker for this session, Dr. Priya Sodan. To introduce Madam, she is now additional professor at HDM College of Medical Science and Hospital. She is a passionate teacher having 13 publications and has reviewed many articles with the area of interest, mostly anti-inflammatory and neuropharmacology preclinical studies. She has authored a chapter on pharmacotherapy on Parkinsonism also, which is a very good book for PG pharmacology. Then she has secured first prize in poster presentation for a research paper on the title, The Impact of Anti-Tubercular Drugs on Inflammation in Rats. And she has secured cash prize for the best research at National Conference by KSMP SIMS Hassan in the year of 2024. And she has organized many CME and also attended various conferences as resource person. So I invite Madam. Madam, please.
Good morning, everyone. Respected dignitaries on the dais and off the dais. Thank you for having me here. Thank you for the introduction, ma'am. So I'm going to share some few insights on this interesting topic, that is emerging therapies in psychiatric diseases. So sprouts of hope for the treatment. See, whenever we start with any kind of a treatment, any kind of a disease, it is the hope, that is a positive hope from the consultant point of view and also from the patient point of view. Unless we have a positive hope, it is not going to lead into a success of any kind of a treatment. So this is very essential. In psychiatric diseases, it's a very huge arena actually. I'll be covering mostly upon schizophrenia and depression, because schizophrenia is a severe chronic mental disorder which is affecting almost 23 million people worldwide. And if not treated, it goes on progressing. It results into suicidal tendencies, as we all know. And worldwide, schizophrenia is more prevalent in Southeast Asia, as well as in China and India. So the therapy started like this, the treatment for schizophrenia and diseases. Actually, these were like demonic positions initial in the 19th century, in the early 20th century. So that time, how it was treated was like they were put up into asylums, and insulin shock therapy was discovered by this scientist, that is Austrian-American psychiatrist Manfred Senkel, where he gave an overdose of insulin to these psychiatric patients, and this led into hypoglycemic coma, and then the patients went into insulin shock, because of which there was a belief that there was a regeneration of the entire neuropsychiatric system. But this wasn't so. It led to many deaths in that time. The next thing, which became very famous, was this, that is lobotomy. Lobotomy was discovered by Dr. Antonio Moniz, and he has received a Nobel Prize for this. So here, what they did was they took probes and they inserted into the eye sockets, and they were trying to disrupt the neural connections by doing this. By then, they thought that, yes, the science was settled, and now the patients, after doing this kind of a procedure, the patient was docile, the patient was robotic, and he was free of all those psychotic symptoms. They were thinking, yes, the science was settled, but as you all know that the science can never get settled. So by around the early 20th century, this was all going on in 1940s, 1950s. But in 1950s, it led to deaths. That is, Dr. Freeman was trying these procedures, but that resulted into many deaths. So meanwhile, discovery of antipsychotic drugs was going on. So the first generation antipsychotic drug came up into the market, as we all know, that is chlorpromazine and haloperidol in 1950s. So by then, dopamine hypothesis of schizophrenia had become very clear. So dopamine hypothesis was very clear by this time, that is 1940s, 1950s. But this had its own problems. It led to many EPS problems. So this led to the discovery of other set of antipsychotics, that is second generation antipsychotics. Schizophrenia and depression, they are always interlinked. So they're interlinked because some of the schizophrenic patients have depressive episodes, whereas some of the depressive patients also have schizophrenic episodes. So they are interlinked, and it is also called as a bipolar disorder. As we go into the etiopathogenesis, schizophrenia and depression can start from the prenatal exposure. Like, there could be prenatal exposure to mumps, measles, or rubella. resulting into psychological stress. Even stress also could be a most important factor for this. Also, as aging goes on, so Parkinson's disease, rheumatoid arthritis, IBD, there are so many various factors that are resulting into depression as well as schizophrenia eventually. We know that there are three important symptom domains of schizophrenia: positive symptoms, negative symptoms, and cognitive symptoms. The existing therapy, that is the first generation and atypical antipsychotics, are doing an excellent job against these positive symptoms and negative symptoms. But yes, they have their own set of adverse events. So the newer antipsychotics are doing very good work against negative symptoms and cognitive symptoms. Cognitive symptoms are also associated with schizophrenia and vice versa. So why do we need these emerging therapies? The existing antipsychotics, they all are known to cause weight gain, metabolic challenges. There are even challenges in engaging the patients to these treatment, retaining them, anosognosia, where the patients are lacking insight into their illness. So this is very challenging to convince them to adhere to the therapy. So this has, when I was going through so many articles, I saw so many newer targets. So I will be just telling about these targets because there are many drugs in the pipeline. So we will focus only on the hypothesis. We already know about dopamine and 5-HT. The newer hypotheses are, PDE, then endocannabinoid system, TAR1. I'll be just explaining in brief one by one. The first one, that is acetylcholine, M1 and M4 modulating agents. So several imaging studies demonstrated there were some alterations in these muscarinic receptors. Discovery of these reduced level of muscarinic receptors were observed. Cognitive deficits observed in schizophrenic patients may lead to disruptions in the cholinergic signaling. So M1 and M4 agonists, as we all know, that they are all used in the treatment of Alzheimer's disease. Actually, xanomeline was being used for Alzheimer's disease, successfully going on. But then they found out that xanomeline was decreasing the positive symptoms and negative symptoms in the schizophrenic patients. So xanomeline became very successful. It is approved in September 2024. It had cholinergic adverse events. That is the reason trospium is added to reduce these cholinergic adverse events. So this FDC has come into the market, and it was approved in 2024. These are the other drugs which you can see in the pipeline. They are still, some of them are in the phase 1, phase 2 trials. The second hypothesis, that is endocannabinoid system. Cannabidiol targets the endocannabinoid system. Now, this research is going on almost since 30 years. There are both pros and cons. CBD actually enhances the anandamide signaling. It interacts with various intracellular pathways like PI3, AKT, JAK, STAT. So what does it ultimately do? It increases the adult neurogenesis. They have antipsychotic properties. How do they do that? They are working on CB1 and CB2 receptors and TRPV1. Therefore, they decrease the pro-inflammatory cytokines, whereas enhance the anti-inflammatory cytokines. So that is the reason they have a neuroprotective role. That is the reason they increase the adult neurogenesis. And therefore, they are potential targets or future promising targets for schizophrenia. So these are some of the investigational drugs which are still under phase 1 or phase 2 clinical trials. The next hypothesis, that is phosphodiesterase inhibitors. We already know that phosphodiesterase enzymes are needed for the catalysis of cyclic AMP and cyclic GMP. Now, why these are essential targets? There are 11 subfamilies of phosphodiesterase, out of which 10A is particular importance in schizophrenia because these are essentially or densely populated in the pyramidal neurons and the striata. That is the main pathogenesis of schizophrenia. So what these phosphodiesterase 10 is doing, it is increasing inflammation, it is increasing neurodegeneration. They're also seen to cause neuronal cell degeneration. So if we use or if we target these phosphodiesterase, that is phosphodiesterase inhibitors, they can ultimately act upon the neuronal survival. They can increase their synaptic plasticity and also decrease the oxidative stress. So therefore, they can be used in schizophrenia. So some of the drugs in the pipeline. The next one, that is brexpiprazole. Now this is into clinical trials. This is a D2 agonist, a partial D2 agonist, whereas an antagonist at 5-HT2A. These are the completed trials on brexpiprazole, like Vector trial, Beacon trial, Zenith trial. It was seen that there is a significant improvement in positive and negative syndrome scale and clinical global impression severity scale. So brexpiprazole, cariprazine, lumateperone, these are all the congeners. They have the same hypothesis, that is they are partial D2 agonist, whereas there are 5-HT2 antagonists. So they can be used in the maintenance therapy of schizophrenia. Lurasidone, I found it interesting because lurasidone has shown some successful results in depressive episodes with schizophrenia. This is because they are working as an antagonist on 5-HT7. So that is how they show some improvement in the depressive symptoms in schizophrenic patients. So there is a huge comparative studies, a lot of meta-analysis results are there. Lurasidone has been compared with olanzapine, it has been compared with aripiprazole and paliperidone, whereas lurasidone has shown excellent results in comparison. The next new hypothesis was the TAR1. Now TAR means trace amine associated receptor. There are 5 kinds of TAR receptors. TAR1 to TAR5. TAR1 is of need here because this is a target for psychiatric disorders. These are all G protein coupled receptors. Again, why TAR1? Because TAR1, they are present in the striatum and the pyramidal neurons, which is the main pathogenesis for schizophrenic patients. So ulotaront is an agonist. It's a TAR1 agonist. And the advantages by using these TAR agonists is, one is reduction in the symptoms, that is PAN, MADRS. Another advantage is no EPS was seen, no weight gain, and they are effective in the depressive episodes. So the other congener, it is still under the clinical trial. These are the other antipsychotics in the pipeline. The other next newer hypothesis is the glycine transporter inhibitor. Glutaminergic hypothesis of schizophrenia, this you all know it results into excitotoxicity, therefore impaired synaptic plasticity and signaling. High risk of excitotoxicity is accompanied with the schizophrenic episodes. So we can see here that the glycine transporter, it is acting as a co-agonist to NMDA that results into excitotoxicity and therefore schizophrenic episodes. So therefore, targeting these glycine transporter inhibitors could be beneficial in treatment of schizophrenia. So eclipirtine is under study. So the field of schizophrenia goes on growing. There are many things. If we go on surveying, there are many things. These are some more like paliperidone. Paliperidone, once a month injection is available. Once in 3 months injections are available. Once in 6 months injections are available. These are for acute episodes of schizophrenia. Even acinapine transdermal patches are available. Risperidone is there, roluperidone. So there are many things which are there in the market. Now, depression, the second concept, that is depression. Depression, we all know it is a leading cause of disability worldwide. S-ketamine and ketamine, these are actually anesthetics. These are all dissociative anesthetics. They are NMDA receptor antagonists, but at sub-anesthetic dose, ketamine was shown to cause some promising results in depression. So how it facilitates, again, neuroplasticity, improvement in the new memory formation, fear extinction, restructuring of the traumatic memories. So this is done by increasing the BDNF signaling, and that is how it helps in the depressive episodes. Single dose of IV ketamine was found in some of the clinical trial that it has reduced suicidal ideation with major depressive disorder. Cochrane review were seen. So this was again a comparison with midazolam and the placebo, where ketamine and S-ketamine have shown much more beneficial results. Intranasal ketamine is also available, which is showing a very good effect on this depressive episodes. After all this, the therapy, that is the drugs, we have the next intervention which has been tried in some of the centers, that is neuromodulation or transcranial magnetic stimulation, where magnetic impulses are being given to control the depressive episodes, that is to control the emotional and behavioral pattern. What it does exactly is magnetic current has been given, and this is known to cause again or result into modulation of the synaptic plasticity or to modulate the release of fresh neurotransmitters. That is what happens when you give a transcranial magnetic stimulation. It results in action potential generation and therefore neurotransmitter release. So yes, at the end, a plan that you all have been working on, that is the Medical Pharmacology Society, has been taking up into a good shape. All of us are planning up something and it is going up into a good shape. So this has resulted into the successful completion of this conference. So to end with, as I want to justify the first slide of mine, that is Sprouts of Hope, right? I spoke about it. So I don't know how many of you people have watched this movie, Luca's World. It had released in Netflix just 2 months back. And also there is a very interesting novel on this. So why I want to relate this with that thing is because this child, the name of the child is Luca, And he's been diagnosed with cerebral palsy since his childbirth. That was because of the oxygen deprivation during the childbirth. There are two ladies you can see there. One is the on-screen player actually, and the other one is the real mother of this baby. Now, this family stays in Mexico, and the pediatricians and the neurophysicians had all given up hopes on this baby, and also told the parents that there is nothing left to give treatment, and there is no failure to thrive. I mean, it's almost going to end up. So that is how they had told the parents. But yes, this mother didn't stop hoping. Like every other alternate mother, every parent, she didn't stop hoping for it. And then she found out that in India, a therapy has been given. This machine which you people are seeing is a cytotron. It is based in Bangalore, and Dr. Kumar is the scientist who has discovered it. He has more than 170 patents and has worked for nearly 30 years for this discovery. This machine is called a cytotron. Actually, the primary indication of this machine is for the treatment of cancer. But because of continuous pressurizing from the family side, that let us try on the cerebral palsy. Let's see if it works, does some wonders. So this family comes to India and they tries to take up one sitting from this. So cytotron was given, every sitting was given. So after every sitting, the child started improving. It developed all its developmental milestones, and now the baby is doing really well. Yes, definitely it goes to a special label school, but yes, it is definitely doing well. So what cytotron did is it produced neural progenitor cells. That is what it was seen. So this is Dr. Kumar, and this machine has also received a breakthrough device designation from the US FDA and European Medical Council. Though it is going on a lot of clinical trials, still it has not been available for commercial purpose. It is there in Bangalore, and it is doing excellent results for most of the breast cancers and something related to the neurological cancer. So it is doing a wonderful job. So to end with, this slide says it all. So one thing is, I express my heartfelt gratitude for selecting me as the recipient for this prestigious award. I'm truly humbled and grateful to Dr. Shivamurthy sir, the President of Medical Pharmacology Society, and the entire committee for having recognized my work. This award will definitely motivate me further to do a lot of work and for the betterment of our society. So I would also like to thank Dr. Shivamurthy sir and the entire MPS Society for giving us an opportunity to publish chapters. I mean, this dream would have been just a dream for me, writing a chapter and getting it published in a textbook. So this would have been just a dream. So that is one. And I would also like to thank our Chancellor, Sri Dharmasala Manjunatheshwara University. He is also the Member of Parliament of Rajya Sabha. Also the Vice Chancellor of my university, Dr. Ranjan Kumar, my family. My husband is here, and my daughter, Stuti. Thank you so much. Thank you for giving me this opportunity. Thank you so much. Thank you for patient hearing.
Thank you, ma'am, for such an interesting elaboration of this topic.
Thank you.
We all are always fascinated by the history, story behind the psychiatric diseases. I think we will agree on that. Okay. So nice clippings, and the movie you told us to see.
Yes, please do watch it. Like some of the movies are really very, I mean, they have some take-home messages. So because I really didn't know about this machine, that is cytotron. I really didn't know about this. And it is based in my own state, Karnataka. I didn't know that they're doing such wonderful job. And this person who is working for more than 30 years on this, and he's come up with this single-handed. So the initial treatments, initial therapies, what he did was totally free of cost. So their charge is like really negligible charge what they're doing. I don't know. I just went through this and then I started researching on this.
Okay. Any questions from audience?
Yeah, ma'am. Priya, ma'am. Priya, ma'am. Priya, ma'am. Yeah, nice topic.
Thank you, ma'am.
Yeah,
Thank you so much.
yeah. Actually, it's very important that we should have some newer drugs now, because the older drugs, even though it is good, it has efficacy, but then it has a lot of side effects.
Yes, ma'am.
And once the patient don't feel like taking the drug again and again, so that is the main problem in the treatment failure. So it's very interesting to know the new pipeline drugs and all.
And in case of psychiatric diseases, the major problem is patient adherence.
Yeah.
They lack their own insight into their illness.
Yes.
So sticking onto the therapy is really difficult. Once you
Yes.
stick onto the therapy, the EPS, weight gain, there are so many problems. with
Yes,
the
yes,
existing drugs.
yes. Mood
So-
swings, everything,
yes,
everything
yes.
comes
So-
out. So that's very difficult for make them to stay in the treatment.
Exactly, ma'am. So these newer drugs, hopefully they are potential targets, and let us see if we can use them. All these hypotheses,
Yes.
at least we can work on it,
Yeah.
and we can find some newer agents for the
Yeah.
treatment of depression as well as.
Yes, yes. And moreover, I have seen the movie The Lucas World. It's very interesting.
It's very
actually.
nice, right?
Initially, I thought it's not real. It's just fictional. But then when I googled it, then I found out
Yes,
it's
it is
real.
It is real.
Yeah.
Yes. ma'am.
Thank you.
Can I
ma'am.
can
Thank
I
you so
add
much.
on to this with permission of chairpersons? Very interesting presentation, Priya.
Thank you, ma'am.
And
Thank
when you
so
mentioned
much.
Dr. Kumar, I couldn't control myself. 15 years back, I visited his lab in Bangalore. And that time he was working on using this cytotron. He had already come up. He wanted to collaborate. I was in Vaidehi and I was not in that position to take it forward. And we had spent almost half a day with him. 15 years later, after Lucas World, I saw Lucas World and I was approached by one of my, I will just take 1 minute for this because it's very interesting. And then when he told, one of my relative asked me to approach, I went, I took 1 month, I had to wait for 1 month to get an appointment with him. But I should tell you, he has got game-changing equipment. 15 years back, he said, his patient, one of the patient had injured his thumb. Why I'm emphasizing is there is a great way forward for all of us to interact with such people and then get inspired and come out with some breakthrough research. He has regrown a finger, a toe, I think, which was injured. We use so many wound healing drugs that there's so many therapies. This was extraordinary work, and he's an amazing person, down to earth, and very happy to share that very soon we are going to collaborate with him for tuberculosis. So that was one thing which I wanted to share, and I hope he's going to, I'm requesting him to come on a podcast with our webinars. If that comes, I'll be very happy to share with all of you, but I felt so privileged to, interact with him. I will share my photograph with the group. Thank you very much for bringing him into this, and it is a miracle, I should tell you, for cerebral palsy patients. Thank you so much. Thank you for giving me.
Okay, ma'am, this way. Yeah. Okay. Thank you, ma'am, for your presentation. Now I request the chairpersons and the members of MPS to felicitate the most promising MD Pharmacologist Award to Dr. Priya Sodhan.
Good morning, all. Now I call the next speaker, Dr. Shantanu Kumar Tripathi. Sir is the principal and professor of pharmacology of the Jagannath Gupta Institute of Medical Science and Hospital, Kolkata. He's also a vice chairman, Innovocare Health Soft Solutions Private Limited, Kolkata. Sir is an accomplished clinical pharmacologist of national eminence and a medical teacher with 40 years of teaching experience. Sir is awarded National Teacher of Excellence in 2023 by Indian Pharmacological Society and was awarded the Doctor of the Year in 2005 by Students' Health Home, West Bengal on the National Doctors' Day. Sir is also the President and Association of Clinical Pharmacologists India and Chairman, Data and Safety Monitoring Board for Phase 1 Clinical Trial, Heterodrugs India. He's also an expert member standing National Committee on Medicines, Government of India, also co-authored the ISDBEU, that is the Berlin Declaration on Pharmacovigilance 2004. Sir is also the recipient of WHO Visiting Fellowship on Rational Use of Medicines, Geneva in 1998. He's a member of the Committee for Framing New Drugs and Clinical Trial Rules 2019, Government of India. And also a member Committee for Framing of National Ethics Guidelines for Biomedical Research 2017 ICMR. He's an advisor to Government of West Bengal for COVID-19 vaccines and vaccination. He's a member Expert Committee for Clinical Trials, SAE and Compensation Assessment. Then served as a Director, Drugs Control, Government of West Bengal. Also Medical Superintendent cum Vice Principal, IPGMER, SSKM Hospital, Kolkata. And member of Co-Training Panel under Pharmacovigilance Program of India. And also a Vice President Forum for Ethics Review Committee in India. Chairperson for Clinical Research Ethics Committee of many hospitals and research institutes in Kolkata. Expert member of Research Proposals Evaluation Committee, ICMR, and BIRAC, Government of India. Coordinator in Adverse Drug Reaction Monitoring Center under PVPI. Then conducted many research projects, trials as a principal investigator or as a guide for many MD and PhD scholars. Then published 200 plus research articles, reviews in national, international journals, and 100 books. Welcome, sir.
Good morning. It's a proud privilege to be here. And I must thank Dr. Shivamurthy for giving me this opportunity. Since morning, we have heard very erudite presentations on technical areas. And now my talk will be a little relief. Okay. It's lighter. It's basically a personal account. Somebody who has attained certain age and in tune with the Indian culture, we respect age, aged people. I am here only because of my age, nothing else. And you are giving an opportunity to an aged person to reflect. Okay. So two reasons. One, the age, the other, pharmacology, nothing else. And I consider myself fortunate. There are so many people now in the country with these two attributes, but now again, it's not really great, very based on logic kind of selection. It's a random choice just because of, okay. I have been given this privilege of reflecting or reminiscing 50 years of my association with this wonderful subject, wonderful discipline of pharmacology. I remember, as for many of us, the choice of this discipline of pharmacology is, for many of us, particularly during our days, it was by default, not by choice. Things have changed, as I have seen through generations. Now, more people take up pharmacology by choice. It was not the case during our time. And different people used to have different reasons why they have come to pharmacology. The other thing what I have seen that the scenario has really changed in this country as compared to our times. Okay. When we see today, it has been more democratized. For good work, you need resources. During those times, I'm talking of 70s, mid 70s, 80s, even till mid 90s, resources were only in a few big institutions, and it was not evenly distributed. So there was a lot of challenges, particularly people in the peripheral institutions. They had only one obligation, that is teaching undergraduates. But then, this is no excuse. We have seen through these 50 years, I have seen how pharmacology as a discipline has evolved and also gained importance in the country, and how this has been, resource distribution has been more democratized. It is wonderful to see younger people, younger minds today, who are quite serious, and they really, the love for the discipline, love for the subject is also quite obvious, quite apparent when they take pride to present their work before an audience. And I must say that their work in India, the kind of work our youngsters are doing, they are at par with at any standard, international standards now. So that has been a very positive part of it. I'll try to, a few slides I'll be using. It is more of a, you can say, kind of a nostalgic feeling on my part, how I have seen the subject of pharmacology growing all over the world, as I have seen, and also to a certain extent about emphasizing on how it has happened in India. So let's proceed. My journey started in 1975. I was an undergraduate student then, and as per the curricular requirement, I had to study pharmacology. There was no escape. During our days, people used to say pharmacology used to be described as the most volatile subject, and somebody who has, by choice or by default, has landed up into a department of pharmacology for doing post-graduation, people thought he must be a crazy person. That why pharmacology out of all the subjects? But then I started my journey in pharmacology when I was in undergraduate during my MBBS days at RG Medical College, Kolkata. And I was blessed to have a teacher like Professor Srithishampad Bhattacharya, one of my foremost teacher in pharmacology, wonderful teacher. And when I was asked by Dr. Shivamurthy to give a lecture on this topic, I was trying to figure out if I could have access to a photograph of Professor Srithishampad Bhattacharya. I found there is none. So this is also a pity. During our times, many of our teachers and somebody who had also contributed a lot, many of our leaders in Indian Pharmacological Society, so their photographs are not available. If you Google, you will not find, because those days it was not available in the Google. And this is also a pity. And half an hour back when Dr. Shivamurthy was asking me to write something, it makes sense. We need to write. So to get things documented. Even I could not find the photographs of my teachers at Banaras Hindu University, where I did my MD pharmacology. Some of my teachers, my guide, Professor A.K. Sanyal, his photograph is not available. He was also the editor of Indian Journal of Pharmacology. Professor P.K. Das, I don't find his photograph. I did my MD pharmacology at BHU Varanasi, 1981 to 1981 to 84. To have stalwarts like Professor P.K. Das, Professor A.K. Sanyal, Professor S.K. Bhattacharya, Salil Bhattacharya, Professor S.S. Gambhir. Only S.S. Gambhir is alive still. And they introduced me to the wonderful world of pharmacology. I did my DM then in clinical pharmacology at PGI Chandigarh under the guidance and mentorship of Professor P.L. Sharma. I came in touch with Professor Ranjit Roy Chaudhary, who nominated me for the World Health Organization International Training on Rational Use of Drugs at Kathmandu, Nepal in 1998. These are the books I read in my undergraduate days. Two books, one on the right hand side, Pharmacology and Pharmacotherapeutics by Satuskar and Vandarkar. The first edition was in 1969. It was the same year when Indian Pharmacological Society was born, and the same year when Indian Journal of Pharmacology was also started. Okay, 1969. And when I came to 1975, it was the fourth edition of the book. And prior to that, the kind of books we used to have in pharmacology, they are more like Materia Medica and more on the with the natural sources of drugs. Okay, so pharmacology started becoming more interesting with logic, with understanding, with explaining the mode of actions. Okay, that is with these kind of books. And for the first time, Indian authored book, that was Satuskar and Vandarkar, and that was the fourth edition I read. It actually came 1 year prior to us, that is in 1974. For the first time, we came across this book. This was Dr. Satuskar was in Mumbai. The other book I liked very much, and I'm sure many of you also liked, and that is D.R. Lawrence's book, Lawrence and Bennett. Initially, it was D.R. Lawrence, Clinical Pharmacology. So wonderfully written, so lucidly written. And that was also in 1975, it was also the fourth edition. So these are the two books I used to read in undergraduate. And then as I landed up in the Department of Pharmacology in BHU, Banaras Hindu University, and I was told that I have to read Goodman and Gilman. And then it was quite frightening, heavy volume. And how many of us know that when I say that Goodman and Gilman, but then there is another person called Goodman Gilman. So some of us must be knowing, but some of the youngsters might not be. For at least, I think, 6 or 7 editions of Goodman and Gilman was actually edited by 3 persons, Goodman, Gilman, and Goodman and Gilman. So some of the youngsters might not be knowing. Very interesting story. So right hand side, on your right hand side corner, Alfred Jack Gilman. And this side is Goodman, Louis Stanford Goodman. He is a non-medical pharmacologist, Goodman, and Alfred Jack Gilman is a medical pharmacologist. Now, this person, I'm sorry, this Gilman is non-medical pharmacologist and Goodman is medical pharmacologist. So Goodman joined Gilman in the same department, and then they found there is no good textbook of pharmacology. Then they reached out to their head of the department, chief of the department, and said, we should have a good textbook. The head said, why don't you write down one? And both of them sat together and started writing. And a couple of years, they completed the first manuscript and they published it. It was 1941. It was the same year. Okay, Dr. Goodman had his son born. And he named his son, his son should have been named something Goodman, but he named his son as Goodman Gilman. Okay, paying respect to his colleague, to his co-author. Just imagine, okay, mutual respect between two co-authors, two faculty in the same department. So Alfred Goodman Gilman was born in 1941. And eventually in 1994, he won a Nobel Prize. Goodman Gilman won a Nobel Prize in Medicine and Physiology. Goodman and Gilman jointly credited with the first use of intravenous nitrogen master in lymphosarcoma. And the first edition on the left side corner, top corner, first edition 1941. I read this, the first time I read this Goodman and Gilman, that was the 6th edition, 1980. I entered pharmacology, MD pharmacology in 81. And this was the first time when Goodman Gilman joined as co-editor, okay, to 1980. And he continued to become the co-editor till 2000, although his father, Gilman, died in 1984. And but then up to 2000, at the year 2000, Goodman had died, and then this gentleman Gilman, he edited it only up to 2000. After 2000, he also stepped out. So beyond 2000, Goodman Gilman is not edited by Goodman or Gilman or Goodman Gilman beyond 2000 as we entered into this new millennium. So interesting story of Goodman and Gilman. So let's now proceed further. Now, in a couple of slides, I'll try to see it's by no way it is a comprehensive, okay, account of advances in pharmacology through these 50 years, but I tried to identify some of the key, okay, advances. 1975 to 1979, okay, because I started in 1975, nothing else. WHO introduced in 1975, it is actually 29th May, the 28th World Health Assembly report, the concept of essential drugs. And the first WHO model essential drug list was published in 1977. Interestingly, if I go to the next slide, WHO introduced in 1975 this concept, but the Hathi Committee report in India, 1975, which was in April, that is 1 month prior to that Hathi Committee report, in the report, they used the word essential drugs and importance of essential drugs to be made available for all the people who need them. So in India, in Hathi Committee report, we conceptualized the concept of essential drugs and underscored the importance of the essential drug list to be tabled, to be used for the purpose of procurement, distribution, and use, okay, at all levels. Within a month, it was the WHO document, and that became popular, and in 77, they came out with this model list. 77, India declared smallpox free, thanks to the vaccination. The extended program of immunization was launched also in 77, and it was mandatory. It was mandatory primary immunization against 6 vaccine-preventable diseases. In next, Thornton and Prey in 75 again, combination drug therapy in psychopharmacology. I was listening to the previous presentation with a lot of attention. Combination therapy is essential in psychopharmacology. That was the landmark work by these 2 gentlemen, which was published in the Journal of Clinical Pharmacology. That was in 75. Cimetidine was made available in 1976 in UK and 1977 in USA. It was a blockbuster drug. A blockbuster drug is considered as one which otherwise earns more than $1 billion or more per year. That is a blockbuster drug, the concept of blockbuster drug. Cimetidine was found to be a blockbuster drug during those days. And in 1977, the prostacyclin PGE2 was approved by US FDA. This I have already covered, that Hathi Committee report. This is the Hathi Committee report actually. And the last bullet you see, the committee also emphasized the importance of drug quality control, rational drug use, and providing essential drugs to the public. This was tabled in the parliament. And okay, of course, some spirit of this report has also been, government has tried to implement. 81 to 89, next 10 years, the first ACE inhibitor, captopril, was approved in 1980. 82 saw the human insulin created through recombinant DNA technology, essentially marking the beginning of the biotech era that started in 82. And 1983, the Luc Montagnier's team at the Pasteur Institute, they identified the HIV to be responsible for AIDS. So we never knew anything about AIDS before that 1983. 85, the WHO Conference of Experts on Rational Use of Drugs in Nairobi. In order to control drug trafficking and abuse in India, we developed that NDPS Act. And it was enacted prohibiting production and sale and possession of consumption of various the drugs and substances that produce addiction and dependence. 1987, very important year, Eli Lilly released the first SSRI, fluoxetine, revolutionizing the mental health practice. The first statin, lovastatin, was brought in 87. FDA also approved the first antiretroviral drug, zidovudine, in the same year, 87. And after the Montreal Protocol, HFA inhalers replaced the CFC inhalers for asthma. So that was also a landmark discovery. 1990 to 99, the first human gene therapy started at NIH to cure deficiency, bubble boy syndrome, that is adenosine deaminase deficiency syndrome. Clozapine was introduced, clozapine as the first atypical antipsychotic. It was introduced with the lower risk of EPS in the United States and Europe. 95, the agreement on TRIPS of the WTO on granting of patents on drugs for a period of 20 years, and that changed the whole scenario, including in India. In India, we are initially a little resistant, but then we have to also accept that finally and sign it. Final signing was actually in after 2000. And there was flexibility to guarantee access to affordable medication. 1996, Lispro, the first short-acting insulin analog, was approved, and 98, Herceptin was launched for the treatment of HER2-positive metastatic breast cancer. 3 million women's lives saved since. Of course, later on, the Herceptin use spectrum has expanded. Coming to 2000 to 2009, salmeterol and fluticasone was approved by FDA in 2000, year 2000. 2002, Humira, adalimumab, received the FDA approval for rheumatoid arthritis and then for psoriasis, ankylosing spondylitis, uveitis, Crohn's disease, ulcerative colitis, and hidradenitis suppurativa. It is also one you can think of otherwise treatment-resistant kind of issue, problem. 2003, human genome sequencing completed. And again, it opened up a new era in pharmacogenomics and gene therapy, etc. 2004, FDA approval of the first epigenetic therapy with azacitidine, an inhibitor of DNA methylation for myelodysplastic syndrome, and the first anti-angiogenetic agent, bevacizumab, to treat cancer. That was in 2004. And then the two basal insulin analogs, glargine, and approved in 2000, and Detimer approved in 2005. 2009 to 2019, FDA approved an average of 38 new drugs per year in this decade. So number of drugs coming for public use, much more in time. Maximum number of drugs were approved by the FDA in 2018, 59. 2012, discovery of CRISPR-Cas9 as a gene editing tool to revolutionize the biological research and drug development, offering a precise and efficient way to alter DNA sequences. 2013, WHO and the United States CDC recommended that bedaquiline be reserved for people with MDR-TB when an otherwise recommended regimen cannot be designed. 2018, FDA approved the first siRNA-based drug on Onpatro for hereditary transthyretin-mediated amyloidosis. And 2019, FDA approval of siponimod for multiple sclerosis. This is also a very important breakthrough for the treatment of multiple sclerosis after a long expectations, long wait. 2020 to 24, 20 to 21, the whole world experienced COVID pandemic, and for the first time, we understood and appreciated the importance of emergency use authorization of vaccines as well as other therapies for managing COVID. 2020, the key pharmacological advances focused on improved drug delivery system gained importance. the world witnessed the potential for mRNA technologies, mRNA COVID vaccines we know of. 2022, progress in precision medicine, pharmacogenomics, CRISPR-Cas9 and nanomedicines. And 23, CRISPR-based therapies that reached the market, AI and ML gaining importance and revolutionizing drug discovery, accelerating process of identifying and developing new drug candidates. Coming to India again, over the past 50 years, the kind of changes that has occurred in India, so far as clinical trial regulations are concerned, in 1988, for the first time, we realized that we should do something, and the rules were framed. Schedule Y of Drugs and Cosmetic Act came into being, establishing the regulatory guidelines for clinical trial permissions. 2005, India acceded to the TRIPS agreement and had to do some adjust itself according to the global need. And it led to a product patent regime. Earlier, we were on a process patent regime actually. Then we had to accept this product patent regime. And major amendment in Schedule Y was done in 2005, we all know. 2007, the CTRI, Clinical Trial Registry of India, was launched with help from WHO. ICMR launched it. 2013, there was a lot of chaos in the whole country. We all know about the conduct of clinical trials not in a very desired manner, as a result of which there was parliamentary committee intervention, Supreme Court intervention, and Supreme Court suspended approval of new clinical trials for new drugs. CTRI registration dropped drastically in 2013. If you go and check, you will find very few registration was done in 2013. Review of the existing regulatory framework and a series of amendments followed, including provisions for compensation of trial-related injuries or deaths. We all remember Professor Ranjit Roy Choudhury, who led that committee and who gave the recommendation for all this. And accordingly, a lot of changes have been made in the regulations, as we all know, and all these came in installments, and finally all of them were clubbed together in the 2019 NDCT rules, New Drugs and Clinical Trial Rules. In between 2017, Government of India came up with the Medical Devices Rules 2017 also.
Sir, sorry to intervene. 5 minutes left for you.
Okay. Coming to research ethics, starting from 1980, ICMR's policy statement establishing ethical framework for the first time in 1980. Then 1988, Schedule Y of the Drugs and Cosmetics Act further reinforced the mandate for IEC, that is Institution Ethics Committee, to review and approve research protocols. 2000, ICMR's research ethics guidelines was further revised and published. 2005, Schedule Y defined the constitution and functions of the IECs. 2006, revision of the Human Research Ethics Guidelines. 2016, ICMR Bioethics Unit was established in Bangalore. 2017, the ICMR National Ethics Guidelines for Biomedical and Health Research involving human participants was revised further, which is now still continuing. And we also know at the same time, there were also ethics related to use of animals for research, for the purpose of research. The Committee for Control and Supervision of Experiments on Animals, CCACA, earlier it used to be called as CPCACA. The name changed in 2023. It mandates the separate Animal Research Ethics Committee to regulate the breeding of and experiments on the animals. Coming to drug safety awareness in India, it's very interesting. Again, many of us may not be knowing, and there are a lot of misinformation in the social media as well as in the print media also. 1992, Government of India established 5 centers for ADR monitoring. 1989, ICMR funded a project on adverse drug reactions with Professor K.C. Shinghal. He left us a few months, I think a few days back. The same year, Ministry of Health, Government of India started a 5-center program, multicentric monitoring of new drugs with Professor P.L. Sharma, Professor R.C. Saxena, Professor A.H. Siddiqui, Professor Molly Thomas, and Professor Nilima Sirsagar. 1993, ICMR launched a multi-center task force project on ADR monitoring. 97, the first full conference of pharmacovigilance at Mumbai. Professor Sirsagar was organizing. WHO special centers for ADR monitoring at AIIMS, Delhi, Jawaharlal Nehru Medical College, Aligarh, SGSMC, Mumbai. And they became the collaborator of International Drug Monitoring Program. 1998, Professor K.C. Shinghal organized the first international workshop on adverse drug reaction monitoring. 1999, Society of Pharmacovigilance was born, again with enthusiasm of Professor K.C. Singhal. 2005, National Pharmacovigilance Program supported by World Health Organization and WHO and World Bank together. But all these attempts really were not very successful. Finally, in 2010, Pharmacovigilance Program of India was launched with support from regular support, financial support from Government of India. Later on, hemovigilance and metrovigilance programs were also put in place. Indian pharmaceutical industry also has shown from 75 to 2005 and then 2005 to 2025, as you see, a very steep rise in their growth and development. Coming last couple of slides, clinical pharmacology in India. The DM clinical pharmacology course that started, introduced in India at PGI Chandigarh in 1979. Again, Professor Ranjit Rai Chaudhary was instrumental in doing that. Indian Society of Clinical Pharmacology, ISCP, was started in 1980 with its journal, Indian Journal of Clinical Pharmacology and Therapeutics, Aushadhi. But all these have gone into obsolescence. It couldn't create any impact, and gradually it had a natural death. Later on, only in 2024, Association of Clinical Pharmacologists were constituted. I am proud to say Professor Melvin George, he is the secretary of this. I am chairing this association currently, and let's hope we'll take it further, this association. As I end, I have a dream, which is an ongoing agenda. I'm working on this, and that is establishing an active and direct role of pharmacologists in patient care. So when I say this, I mean actually the pharmacologist to practice pharmacology. Okay, a pharmacologist can also practice as a general practitioner. Okay, but then when I say this, I mean pharmacologist to practice as a pharmacologist. So how that can be done, that can be on a separate occasion. I can try to discuss on that. But I strongly believe that pharmacologists can play a very, very important role in optimizing patient care. And some of our colleagues and students, they are actively working on this. We are working in tandem with Association of Physicians of India and many other clinical societies, and we are quite happy with the progress. Finally, these are some of the areas of my interest in which I have worked, tried to work something: rational use of medicines, pharmacovigilance and patient safety, essential medicines, community pharmacology, ethics in clinical research. But most importantly, I have always loved teaching. Teaching and training pharmacology always remained at the core of my heart, and I am proud of my students. As I leave this platform, I remember this came in the annual review of pharmacology and toxicology. As I entered BHU department, I was told that you have to read annual review of pharmacology. Every year it is published, and in your final exam, there might be a few questions from these 3 years annual review of pharmacology and toxicology, and you have to also give your seminars. So the 1975, that is the first time I was introduced to pharmacology. I was looking at that volume, and I found one article in which I see this. Ivan Pavlov, we all know Pavlov. He was, as a teacher, Pavlov believed that a good lecture should necessarily stimulate questions. It was a rule at his lectures that any student could raise his hand at any time and ask a question. Having completed a part of his lecture, he even made a special pause to encourage students to ask questions. It grieved him to hear none. In such cases, he considered his lecture inadequate and would plead, questions, questions, I don't hear questions. So with this, with my tribute to Ivan Pavlov as one of the early teachers in pharmacology or physiology, that time it was physiology and pharmacology used to be considered at the same breath. I rest my case. Thank you very much.
Thank you, sir. So it was a nice topic. Yeah, it's nice to have a reflection on pharmacology and your journey as a pharmacologist. It was very nice, very inspiring. Thank you.
Thank you, sir. It's always a privilege to hear from you and throwing light on these timelines of different important drugs of that time and covering every aspect like drug, clinical trial, patient safety, also the book review, everything. Thank you, sir. Thank you, sir, for such an elaborative presentation. Any questions from audience?
Just to say that, sir, he has mentioned so many, like references today. I believe that sir is there in most of the committees as a advisory board member at the national level and sometimes international levels also. And sir humbly said that age and pharmacology is the reason. No, it is not that. Sir is the stalwart of pharmacology. I hope all of you agree. And I request everyone to give him a standing ovation and for his contribution to our pharmacology. Thank you so much, sir. So just to proceed with the award ceremony. Sir is conferred with Lifetime Achievement Award through MPS Society. It is the conscious decision by the board members of MPS that sir should be considered for this. So that's the reason. And we heard not from him, from his students. So that is the success of a teacher. Okay, so when we hear that my teacher, when they proudly say that he is my teacher and my pranam to sir before starting anything, I think that is the success of any teacher. I hope all of you agree. Thank you, sir.
I'm humbled.
Now we will take a short break and return shortly with more engaging and enriching speeches. Thank you. आपका है
एक मिनट सर
अभी सिस्टम में नहीं मेरा नहीं है ऐसे कुछ ठीक है
register in ESICMC and PGIMSR. She served as the member secretary of IEC and presently member of IECMEU. Then she has many publications in national and international journals. She is also a reviewer in Indian Journal of Pharmacology. So, ma'am, please. Our next chairperson is Dr. Kavita Rajaratna, Professor of Pharmacology at Bangalore Medical College and Research Institute, Bangalore. With 25 years of service, Dr. Rajaratna has made significant contribution to medical education and pharmacology. She has served as a board member for both UG and PG courses at Rajiv Gandhi University of Health Sciences, Karnataka, and is a member and resource faculty at her institution's medical education unit. Please join me in welcoming Dr. Rajaratna at this stage. To the stage, please.
Good afternoon, everyone. It's my privilege to invite the speaker of the next session, Dr. Divya Arvind Vijaypur. She is the director in patient safety, vaccine and immune therapies, AstraZeneca, UK. And currently she is serving as medical director for patient safety in vaccines and immunotherapies in AstraZeneca, UK. Prior to this, she built an impressive career in both the CRO and pharmaceutical industries as a drug safety physician, managing the safety of both developmental and post-marketing products. Her areas of specialization include mRNA vaccines, safety and monoclonal antibody vaccine safety. Key safety areas of special interest are risk or signal detection. Join me in welcoming the speaker, Dr. Divya. Please take over.
Thank you for the warm welcome, ma'am. I'm trying to share my screen, but can you please confirm, am I audible? Am I audible?
Yes, ma'am, you are audible.
Okay, I'm trying to share my screen. Is it possible from the organizer's side to share my screen, share my slides, which I had sent it across? I have a difficulty in sharing my slides. I have sent it to the conference mail IDs.
Yes, madam. They are working on it.
Perfect. Thank you so much.
Ma'am, you can also share, ma'am. That will be more convenient to you. There is a sharing button is there. Just
Yeah, I'm trying. My Mac is not allowing to share. Are you able to see the screen sharing now?
Yes.
Are you able to see what I'm sharing now, please?
Yeah.
Can you confirm?
Not it, madam.
Not yet.
Slides
Okay.
are not visible.
Sam, this is also different.
Only the chat box is visible. ಇವ್ರು ಮಾಡ್ತಾರೆ.
Can you share it once again, or you can WhatsApp me?
I'm trying to share.
Just 1 minute.
The slides are on the mail IDs, 2 mail IDs for the conference communication. Sorry, it's the settings on my Mac. I guess it's not allowing me to share. Yep, that's the one. Thank you. Will I have a control to move the slides, or would someone do it for me?
Done, ma'am.
Thank you. Will I have the control to move the slides?
She can say next and he can change the slide. Yeah. You can say next and he will change the slide.
Okay, perfect. Am I okay to start?
Yes, you are good to go.
Thank you so much, ma'am. Hello, everyone. It's my pleasure to be here in this platform today, addressing to you all. And I know it's not a very scientific topic, but it is very much important for us to understand what are the opportunities we have outside India. And we have seen what are all the scopes within India and what's happening outside India. Let's move on to the next slide, please. So just a disclaimer, as I work for AstraZeneca, so there are no contents are taken from AstraZeneca. This is purely my analysis and my understanding of the scopes in abroad for MD pharmacologist. Next slide, please. So why do we think MD pharmacologists from India have opportunities outside? Because there is tons of opportunities outside because of the strong academic trainings, what we undergo in India, and the thorough clinical knowledge and the hands-on research experience. We have seen from all the speakers how much research is happening and how we can get our hands-on with all the research and how we can build our knowledge. So this becomes a valuable asset in an international healthcare and pharmaceutical settings. So these what we do in India during a medical school, post graduation, that's MD pharmacologist, everything will add on, and all the conferences, paper presentations, interactions, networking, all of these will become a strong base when we want to work in abroad. Next slide, please. So what is the key for someone to take a next steps to move abroad? Please plan this well in advance, and you should have a vision which country you want to move to, what you want to do, which sector of pharmacology or the pharmacology background or the science you want to put your hands on. So we need to do a proper planning, preparation, and the right guidance is the key. With this, we can build a successful and very rewarding career abroad. So, what are the key steps? We need to improve and keep building on the research or whatever academics, what we do in India. And we continue to obtain a lot of certifications. Again, when I say international certification, this is very specific to a specific countries. I will talk about it in detail further in my slides. Mainly, I would highly recommend the strong communication and the leadership skills, which is the key along with the science and pharmacology and all the research we do. Leadership skills play a very key role in building our career outside India. Next slide, please. So I would mention about the few options and I would talk about the technicalities, advantages, disadvantages as we go along. So as we all know, academic position is again an option outside India in the universities and research institution all over the world. Please, previous slide, please. We all know that there is a scope all over the world for us. It's not just within India if we want to step out of India, and we do have multiple opportunities, but we need to work towards those opportunities, find out which country we have to go. What do you want to do? So, for example, we can do masters or PhD and fellowships. What I mean by that, we can continue doing PhD in epidemiology. So this adds up to the medical knowledge, what we have, and the MD pharmacology, what we have done. Along with the safety epidemiology is one of the options I'm just trying to say, but there are many other branches where you can do your PhDs outside India. We need to find out which universities, which institutions offer this, and what are the prerequisites to fulfill this to be able to start in a right time, as the academic years are very different in outside India. Also, there are options in the academic position, such as teaching roles similar to India, and the non-teaching roles where you can purely be in an academic setup to do studies and do your research and publishing the research. Next slide, please. Another option is a healthcare system. Again, as we have all the opportunities in India, there are opportunities outside India in several countries to work in, medication regimes, polypharmacy to deal with, there are multiple patients. There are a lot of geriatric patients in the hospital setup in the Western countries where polypharmacy is the biggest challenge. So they do have an opportunity for MD pharmacologists to work in the hospitals to help assist other treating physicians, geriatricians, pharmacists to build the prescription. And antimicrobial stewardship, this is also a very important sector back in India and also abroad in several countries. And precision medicine is at its peak now, and I think for all the budding young pharmacologists, this is a direction to think, because oncology and other therapeutic areas are mainly working on precision medicine. And being an MD pharmacologist, having a medical background, we do have a different stand in the precision medicine, unlike other scientists, pharmacologists, pharmacists in this sector. So we do have a special place, and I will talk about it in my future slides. So there are certain roles and opportunities which are only signatory, where a physician can be in those positions. So this is where we or you all can be very unique in this precision medicine sector, where you have a medical knowledge, you have a pharmacology knowledge as well. You can bring a lot of research and other aspects into precision medicine. Another opportunity is to be a faculty of pharmaceutical medicine. This is exclusively in the UK, and I'm sure there are a lot of other certifications similar to this in different countries. So just a few points to mention about the Faculty of Pharmaceutical Medicine. This is assigned by three Royal College societies, and it mainly aims to advance and develop the science and practice of pharmaceutical medicine. It is to both the benefit of the patient and the society, and there are a variety of programs. Please get onto this website, Royal College of Pharmaceutical Medicine, and also get in touch with me if you want to understand more about it. I have not personally done this, but this is in my bucket list to do. As we have heard from several speakers, please continue to learn. There is no end to learning. So we have to be looking out for what we can do and what are the opportunities available. And I am more than happy to talk outside this time to understand if anybody is interested to do this Faculty of Pharmaceutical Medicine. Next slide, please. So continuing on to many other options, as we saw how there are opportunities in academics, let's move on to the opportunities in pharmaceutical and biotech industries. I have been listening to few of the speakers since yesterday, as and when I can, and very aptly and rightly mentioned, there is a stress and there's a long working hours. That doesn't mean that there is no work-life balance with the pharmaceutical industry. It's how we define our working culture and how we define our deliverables and how we want to deliver what is assigned to us and what we want to achieve. So there is definitely a work-life balance. And for me, the personal option, and I didn't do pharmacology by default, but I choose to do MD pharmacology because I knew what I want to have a career path as. And definitely I can vouch there is a 100% work-life balance by how we define our work culture. So pharmaceutical and biotech industries all over the world are looking out for MD pharmacologists. And what are the opportunities in those sectors? For example, we can be part of research and development, drug discoveries, preclinical testing. So preclinical testing, being an MD pharmacologist, we all do learn about toxicology studies, assays, and other pharmacokinetic and pharmacodynamic studies. So, but there are a lot of pharmacy and other background, science background personnel interested in these areas. But as an MD pharmacologist, we do have a leading roles in leading the pharmacokinetic studies, pharmacodynamic studies, and work with the team and the colleagues who are all going to support us. And this is very much happening because we all know how many clinical trials are happening in the Western countries. And for all the clinical trials, preclinical testing is the key. So there is a huge scope in the pharmacokinetic and the pharmacodynamic sector. Also the toxicology study sector, if anyone are interested outside India as well. So drug discovery, I meant quite a lot of work happening outside India, and I'm sure it's even happening in India. But outside India, like for example, modification of the monoclonal antibody to just extend its half-life, or to see, by substituting some subunits, what's happening. So drug discovery starts at a very initial stage where the modification can happen in the lab nowadays, and we do have I have personally worked with the molecules where they are all modified in the labs to have a proper structure and design how we want, considering half-life, efficacy, safety, all of these matters. So there's a great lot of scope in this drug discovery sector outside India as well, if people are interested to move abroad and take this as one of the options. Let's move on. Clinical trials. Again, the drug discovery, preclinical testing are all the very basics for clinical trials. But if we do not like to be in those sectors, there are other opportunities where we can lead the clinical trial programs and be a clinical study physicians. By saying clinical study physicians, could be safety physicians or a clinical physician. So there are different roles and opportunities in this clinical trial sector as well. Next slide, please. So again, moving on from research, drug discovery, preclinical testing, we do have a hot topic, drug safety and pharmacovigilance. So my key interest has been the drug safety and pharmacovigilance. There are certain myths that people say, oh, pharmacovigilance is boring, it can get monotonous. But I developed this interest during my MD pharmacology, working with my mentor, Dr. Pratibha Nadig. She's here in this conference today. I must say that My interest and love for pharmacovigilance started then as an MD pharmacology student. And I was very clear what I want to do post MD pharmacology is be a pharmacovigilance specialist or a pharmacovigilance physician, what it is addressed in different countries. So drug safety and pharmacovigilance is one option. Pharmacovigilance is never monotonous. Every day is a different challenge. Different health authorities come back with different questions with the post-marketing products. And we have 100+ countries. Can we imagine how many health authorities and how many questions we will be answering? Every question is a challenge and every question is unique to address to the health authorities. And when I say drug safety, it is also a pharmacovigilance, but it's called drug safety when it's at the pre-marketing stage. So drug safety itself is another very interesting branch where you can get involved in early clinical trials or late phases, which includes submission to the authorities and the markets. So very interesting journey. I can talk about days because I'm very passionate about drug safety and pharmacovigilance. Anybody who's interested to get more details how we can build this, please get in touch. I have my details at the end of the slide and the beginning of the slides as well. So the opportunities could be various. You could be risk management specialist, signal management specialist. So risk management specialist, why I have mentioned here is this is a very big change which is happening in the Western countries with the risk contextualization activities. So more and more regulators are looking for risk contextualization at the drug development stage. Again, we do all know that risk management plans are in place for the post-marketed products. But the risk contextualization is a sector which we can correlate to very close to what Dr. Melvin just mentioned in his talks, real-world evidence, and how we collect real-world evidence to support a clinical trials, identify the safety issues at an early stage, or predict what could be the safety or what could be the challenges as we go along the clinical stages of the program. Risk contextualization is the key, and this is very much happening across the Western countries in the pharma world. And how we do this is, again, very different. And how you can be a part of this as a risk contextualization specialist or a risk management specialist, safety epidemiologist, variety of options here. Safety epidemiologist is, again, another sub-branch, very much booming in the Western countries. What these people do is they do have, as Dr. Melvin mentioned, they do have access to many of the databases where they get the real world evidence. For example, I'm talking about an mRNA vaccine, just an hypothetical situation here. What are the risks we identify and what is the risks, background, prevalence, incidence in the real world? For example, there are a lot of databases which can look into a lot of registries in the US specific region. If the trials are happening in the US, we can just check the registries or check the incidence rates and predict what are the rates of these safety issues or the risks which could come. It's about rightly having the numbers into our anticipation, what is expected. So it is a very much booming sector in the Western countries, and something to watch out for the budding pharmacologists to think as an option. Safety physician, again, they do, safety physician is the most common pharmacovigilance or a drug safety role. Again, in some countries, they are not called physician because this hierarchy of physician, non-physician it goes down very much outside India. So we do not address someone by, sorry, is someone trying to say something? Okay, I guess no. So it's purely not mentioned as a safety physician role. It could be just called as a drug safety officer or drug safety specialist. So it is not purely a physician title attached to the medical degree we have outside India. You being called as a doctor, sir, ma'am, everything decreases or do not exist. So this is again a big cultural shift. I will talk about it in my next slides. But drug safety physician or a drug safety officer, what they basically do, they do analyze and monitor the safety data. So this could be from the medical review of the case processing to an aggregate analysis of data, reports like PSUR, DSUR, PADERS, and moving on to signal detection, risk management plans, all of these. So again, where we come different to other pharmacy and other science specialists is a lot of science specialists in this field in the pharmacovigilance, but how we stand unique or how we play a role because we will have the signatory roles. There are many regulators who definitely want physicians staying in the Western countries, that's outside India, to be the signatory for this documents. And we all know the most of the authoring and all the background work happens in India, but what you see here is more of the review, final signatory positions of this pharmacovigilance and drug safety. There is a lot of scope outside India, saying that it's a very niche role. They do not need hundreds of us. They need one or two, because the rest of the burdens are taken over by non-physicians or a non-pharmacologist. That's from the scientific background people. So they do bring a lot of a value into the drug safety and pharmacovigilance. But when it comes to signatory roles, it's mostly the MD pharmacologist or the physicians who have this roles in outside India. So moving on, medical affairs is another opportunity. How they are placed in the pharma industry outside India is slightly different to how they are based in India. So there are a lot of certifications again expected for these roles based on which country you choose to do this. Again, it could be anywhere from the medical registrations to a specific country to other certifications. There are a variety of different ways the certifications are done for medical affair roles in different countries. So what do these people do? They assist in planning the product launches and there are other activities related to the products, trials, devices, and they also convey the message to the outer world in a way it is acceptable. Next slide, please. So continuing on to the options for MD pharmacologists outside India, regulatory agencies. So we as an MD pharmacologist can work with regulatory bodies. I've just mentioned few here. There are several other bodies, BPharm, ANSM, Swiss authorities, many other authorities, but I've just mentioned the few: FDA, EMA, and MHRA. So MHRA is for the UK, EMA is for the European zone, and FDA is for US. We all know. I have mentioned these just because these are very common regulatory bodies. We know, as I'm addressing to the crowd of post-graduation to and experts here in this crowd. So FDA, a lot of factors to be considered. There are certain norms and conditions for entering into an FDA jobs, but it's very interesting and fascinating to be working in FDA. I have not personally worked in FDA, but I've interacted with the people who have worked in FDA. It's a great experience. Again, what do we do here? Many different roles, where we can bring a pharmacology experience and the medical knowledge and all our other expertise we have learned from the research and other interests we have can be put into place by having several roles in FDA. EMA, again, is another interesting regulatory agencies for the European zone. We all know about it. Very interesting to work for EMA. And highlight I would like to mention, so I applied for a role in EMA. So I was rejected for a reason. I didn't know about this. I was very new to the country in UK, and I applied for this role purely because to have a role in EMA, you need to have one of the European language fluency, reading and writing skills other than English. So it's another European language. So this is a basic requirement for EMA. So what I'm trying to reiterate here is we have to figure out what we want to do, and we have to work towards it from the very early stages. Like for the postgraduates in the first year, second year, third year, do not wait until you finish your MD pharmacology exams, have your certificate, and then think, what am I doing? So please start planning very well in advance. I know one of my friends in my medical college, she started learning an European language. At that time, I was very fascinated and was not very sure what is she trying to do. But she had a clear vision. She's settled.
Dr. Divya, sorry to interrupt. You have 1 more minute.
Sure. Sure. Perfect. Thank you so much. So the language, one of the European language is a requirement to work in EMA. So see, such are the differences to work in India and outside India. So everything has got a requirement, certification, and a lot of tick boxes to be fulfilled to have a position or even to apply for certain roles. MHRA is an easy one, no specific requirement as such, but an experience on medical background and the pharmacology background is the key. Again, variety of roles there. Let's move on to the next slide, please. Next slide, please. Right. So requirements here, I have mentioned few of the requirements such as licensing and certification, which I spoke about I went through different options. Again, these licensing and certification could be anything from building leadership skills, self-lead, strategic thinking, time management, prioritizing the work, could be general medical council registrations of different bodies in different countries is one of the requirement, and all this work has to start way before we think which country we want to go, as what we want to work, where I want to be working, or what's my vision in the next 5 and 10 years. So recognition of qualification, this is something the Indian degrees are not purely identified as it is, but there is something called matching the jobs or the qualifications. So for example, there is a NARIC association in UK who does convert your Indian degrees into an UK equivalent degrees. Similarly, we have a recognition of qualifications across different countries. This is something you need to get on and to start working on, to even just see where your medical degree stands. What is a grade? What are the different levels where it stands in the country where you want to move? Visas and immigrations is a different challenge. Also, a very key highlight, please do not have a big ambitions, oh, I want to move to UK, and UK is closed the visas for next 3 years or 5 years. This is just an example I'm saying. This could happen with any countries you aspire. So, but it's positive with the UK now that a trade deal has been signed off between the UK and US very early in this week, Monday, Tuesday, where it's very positive and a lot of visas will be issued to Indians. So when the visas are closed, we try a lot. We can get into the positions, but without visas, you cannot work outside India. And also language exams and other requirements is the key for getting through an immigration. This is purely separate to what qualifications, recognitions, and degrees we hold. So this could be another challenge we have to beat or work towards it. And different countries have different requirements. Again, starting from bank balance maintaining to language exams and other certifications to hold. Also, to add job market competitions, this is something purely to read and understand, speak to the people who are there in those countries you aspire to move, get in touch with LinkedIn. And I've recently understood LinkedIn profile, or having a LinkedIn profile, is not something very common in India now. I would highly recommend every postgraduate student, medical students, to have a LinkedIn account. It's a very, I'm not marketing for LinkedIn, but I'm just mentioning that's one of the platform I felt it's very useful to network. So a lot of people have contacted me when they want to move to UK, or when they think of moving to UK, to discuss the opportunities and the scope and all the pros and cons of it. Language barriers, we will definitely face the language barriers. Different countries have different languages, but English is a dominated language in most of the countries. But there could be countries where English is not much used. So getting onto this language learning is also the key if you want to move to a specific country. Cultural differences, last but not the least, is the biggest thing. Again, as I said, sir, ma'am, doctor is not followed outside India. Starting from there, how you dress up, how you present yourself, and your body language and the wordings you use, everything is very different. Just to mention, when you talk about a weather for a minute or so, people in UK think you are a very friendly person. So there are different cultural differences in different countries, which we need to understand and give it a time. And weather, not to mention, the weather can be extremely hot or extremely cold, and a short summer, long winters, long days, short days, etc. All of these things have to be considered when we want to think about an opportunity abroad. Definitely there are pros and cons. I wouldn't say it's all pros. Wherever we work, as whatever we work, we always have to face the pros and cons. So I will conclude my talk with this, and can you move to the next slide, please? Thank you so much. So here are my details, my email ID and my personal phone number. If anybody wants to get in touch, or with my name, you can find me on LinkedIn. I'm more than happy to talk to people. I do have connections with different countries where people are working in different sectors. I'm more than happy to help and also learn as we go along with the discussion. I will conclude my talk with this, and I would want to mention special, I want to give a big shout out to my professor who's in this crowd, Dr. Pratibha Nadik, under whom I was mentored She was my guide for my MD pharmacology, and I'm forever grateful for her guidance and advice. From then, I met her in my MD pharmacology
Divya?
till today. Touch with her. Yes,
Dr. Divya, Nadik ma'am is here.
yes.
You can take the mic then. She's very proud of you. She was telling
Thank you.
us
Thank
about
you.
you just before this talk.
Hello, Divya.
Namaste, ma'am.
Namaste. So proud of my student. Actually, I don't know whether it was read out in her intro. She was the winner of, she won the first prize in RSSDI oral presentation, which was held in Bangalore in the year 2015, Divya? I think so. When she was doing MD, and she was one of my, I am also equally proud of her. She followed whatever instructions were given to her, and she had her own ideas also. So I think that team matters. It's not only the teacher or the student. We keep giving instructions. How best the student takes it, it's up to her. And I'm really proud of her. And I should say she will go to great heights because she has not forgotten the contribution made by everyone, and I'm one of them. Thank you so much, Divya. God bless you. It was
Thank
a
you
great
so much, ma'am.
presentation.
I can never forget all the words you have said, because I used to go to ma'am with my personal issues and challenges and advices. She's always said, Divya, this time will pass by. You will have your time. So these words will always echo in my life, in my career, even in my day-to-day work. Whenever there are challenges, I just remember what ma'am has said. This will pass by. I'm not there yet, but this time will pass by and my time will come. So it's great words, ma'am, and great support from you then to where and what I am today. And I would also especially thank my husband and my daughter, my parents, and special thanks to the organizers, Shivamurthy sir and Dr. Fulun Sharma, who has been in touch with me and discussing through this journey. And really thankful. And I must say, this conference is very different to the other conferences I've attended. The topics are very widespread and very apt to the younger generations as well to continue their interests and passion. Thank you so much. Thanks again.
Thank you, Divya. It was an inspiring and motivating talk, particularly for budding pharmacologists. It was very much needed for the young pharmacologists. Like they'll be having so many doubts and queries in their mind, like what to do in future after doing getting an MD degree. So as you said, focus is very important, and I congratulate you for having achieved your goal to, I mean, until now, you are focused and you wanted to work in pharmacovigilance. I congratulate for that and all the best for you. Any questions from the audience, or you can put in the chat box later, she can answer, right? Okay,
Yeah, I
thank you,
can do that.
Divya.
Thank you so much. Thank you so much.
Thank you, Dr. Dibba, for your kind actually acceptance for our invitation. Actually, whenever we wanted to, actually me and Shiv Murthy sir discussed that let's make it international. So then I thought, whom to actually invite? Then I thought, why not our friends and students? So Dr. Sonia and Dr. Abhinav are my students, and this is my friend, and they are doing excellent jobs. So let's make it. So after that, actually, thank you for accepting our invitation. Thanks.
Just to
Thank
again
you.
add
Thank you.
words. You have been a very good supporter of Medical Pharmacology Society. So thank you for joining us and addressing the gathering here. So looking forward for more working together. Thank you. Bye.
Same here. Thank you so much, sir. Thank you so much.
Hello. We would like to invite the next speaker, Dr. Madhav Rao C. He is Additional Professor and Associate Dean, Research, AIIMS Mangalgiri. He is a founding member of EC and IAEC Research Cell, IRC and IAHF. He has received extra mural and intramural grants for some research projects. He is chairperson at EC, life member of ILAE. He has many outstanding research publications and is nominee and assessor of CCSEA and CDSEO and NMC. Welcome, sir.
Thank you, madam, for the introduction. Very good afternoon. First of all, I thank the organizing committee, Dr. Sivamurthy sir and Dr. Fulan for giving me this opportunity. And at the same time, I also apologize that I am standing before the lunch. And at the same time, because of the paucity of the time, and being this subject so wide and so broad, I may be able to give only the few insights about the monoclonal antibodies pioneering the precision medicine. I'll try to give a few important what's the recent things happening in this area. With this background, I'll go through some historical foundations, development journey from how this monoclonal antibodies have started and what is the current status and how the production evolution has also started and the clinical applications in various medical disorders and what are the challenges and the solutions for that and the future directions. Monoclonal antibodies are the major therapeutic class of drugs which are indispensable tools in research, therapy, and diagnosis. And they are form one of the most precision medicine, and they are widely applicable for not only for the treatment, including the prophylaxis and detection. And they are used for the spectrum of medical conditions, including communicable and non-communicable disorders. We see the market. It has been reported that in 2023 alone, $238 billion was been invested on this area, and it has been projected in 2033 by $679 billion, and the expected compound annual growth rate between 2024 to 2033 expected to be around 11%. Sorry, I'm not able to move the slides. Oh, sorry, right, it's fine. Until now, almost 145 monoclonal antibodies have been approved by the regulatory agencies from the 1986 onwards, and among that, 91 for the cancer treatment. And these monoclonal antibodies form the precision therapies and revolutionize the treatment for the various spectrum of clinical disorders, and they also address the various unmet needs for the untreatable conditions, and they become one of the novel molecules for the various conditions which are rare in condition. What are the monoclonal antibodies? Monoclonal antibodies are the specific immunoglobulins produced by the single clone of B cells, and they are highly specific to the particular antigens, and they are homogeneous in nature and with a large molecule, and most frequently the class, it is the IgG class. And among the IgG class, again there are 1, 3, and 4, and 1 is the most commercially used for the production of the monoclonal antibodies. And by size, as I already told, they are very highly big protein molecules with 2 identical light chains and 2 heavy chains. And they have got 2 important regions, Fab and FC. They also have got very important things like have dual purpose both in diagnosis and treatment, and they have important role in like modulating the immune system. And clinical benefits includes they cover the spectrum of disorders, including the rare diseases with rapid therapeutic effect, and at the same time they have significantly improved the quality of life in many clinical conditions, especially the cancer. And if you compare with the other like conventional treatment, they are associated with these fewer side effects, and they have got more precise in nature, giving the most important like personalized approach. And that cover the both including the rare disorders, and with minimal drug-drug interaction, with higher specificity and increased efficacy. And this is a nomenclature. I'm not going in detail. There are different sources of monoclonal antibodies. It started from the murine, then went to the chimeric, humanized, and human. They are the most important and the critical step in the development of monoclonal antibodies is the identifying the antigen, and that is expressed will be expressed during the cells on the cell surface, and then will be guided by the selection process by the various databases, and production of monoclonal antibodies is a very complex multi-step process. It involves the various like including the cell line development, cell culture expression, purification by the various techniques including HPLC, and characterization and identification by the mass spectrophotometry and ELISA techniques. The production techniques for the monoclonal antibodies includes hybridoma technology, CHO line cell expression, recombinant DNA technology, and electroporation. And other various also include like genetic engineering and the protein engineering. And of course, the current manufacturer standard has to be as per in line with the regulatory compliance, including the GMP standards. And this was started with the beginning of the technique. Hybridoma technology was invented by the two scientists, that is Milstein and Kohler. And for that, they have been awarded the Nobel Prize in 1984. With that, the hybridoma technology, it is nothing but the isolation of the B cells, and it is fused with the myeloma cells, and the cells producing the desired antibodies will be isolated, and which will be manufactured in the larger quantities. And coming to the historical evolution, the first thing that with the hybridoma technology was the monoclonal antibodies, which came that is 100% mouse protein, but however they were associated with higher immunogenicity. And later, because of this immunogenicity, they tried, and further evolution happened with the reduction in immunogenicity that includes the chimeric, which contains little bit part of the mouse and human hybrid. And later, humanize only little like mouse binding regions, and finally, the 100% human protein, and that is a fully.
I'll let you know about that.
They were
What will be the
came into the
compensation?
clinical practice with very minimal immunogenicity or hypersensitivity reactions. The first monoclonal antibody which approved in the and marketed in the clinical practice was the Muromonab in 1986, and that was approved for the against the CD3 cell in acute transplant rejection. But however, the earlier challenges for the murine antibodies was the HAMA
I would
reaction,
like to know
that
the
is human
compensation
anti-mouse
for on-time
antibody reaction,
option,
and that
whatever is
limited the
available with you.
further
After that, I'll let you know about my
development of monoclonal antibodies with this murine, and that led to the involvement of the other chimeric, humanized, and the fully human monoclonal antibodies. And purification characterization done by the various spectrophotometric methods, and quantification by the ELISA techniques. Coming to the mechanisms, there are various mechanisms have been involved in by the monoclonal antibodies. One is the neutralization, that is binding of the monoclonal antibody to the pathogens or the toxin, and neutralizing
Fine, I'll
the
just let you know about
toxin which is involved in the pathogens of a particular clinical disorder.
If I'm
Second thing
interested,
is
I'll
antibody dependent cellular cytotoxicity, and complement dependent cytotoxicity, and the receptor blocking various signaling pathways involved in the pathogenesis of a medical condition. Neutralization is the binding of the monoclonal antibodies to a particular pathogen or the toxin, and blocking that interaction with the host cells, and that facilitated the removal from the body. And antibody-dependent cellular cytotoxicity, it is nothing but antibody, as it's self-explanatory. Antibody binds to the particular target, and the Fc region binds to the receptors, which in turn activate the various immunological cells, including natural killer cells, macrophages, neutrophils, and that in turn will destroy the cell by releasing the various cytotoxic substances. And complement dependent cytotoxicity, again, as itself suggests, it will activate the complement system, which in turn form the MAC, that is membrane attack complex, that in turn lyses the target cell. And in between, there are also various signaling molecules which are directly or indirectly connected to the particular medical conditions, like CTLA or like PD1, which stands for the program death. These are all cytokines. And those cytokines which are involved either directly or indirectly in signaling pathway through the receptor interaction also can become the one of the candidate for the target for the by the monoclonal antibodies. And there are also other things like antibody drug conjugates. I will be dealing a little bit in detail about this in the next slides. Then radio-labeled monoclonal antibodies, they are also involved. They are also used in the therapeutic as well as the diagnostic purposes. And there are something called as BiTEs, that are bispecific T-cell engagers. These antibody drug conjugates, they are the biological missiles, targeted delivery of the toxic payloads, which consist of antibody, linker, and cytotoxic drug. And they become the one of the advanced approach in the chemotherapy of various cancer cells. And coming to the various few approved drugs that which are like antibody drug conjugate includes enfortumab, belantamab, sacituzumab, mirvetuximab, and many more. And the next thing, that bispecific T-cell engagers, which are nothing, as the name it suggests, it means these are the monoclonal antibodies binds to two different antigens, and which are connected to the that connect the T cell to the cancer sites. And it has been established that these bispecific monoclonal antibodies have what more powerful with the lesser toxicities. And there are some few examples of like bispecific monoclonal antibodies which have been licensed in the last years. And coming to the common adverse drug reactions associated with monoclonal antibodies, most commonest is the hypersensitivity reaction. And the second thing, like it can also lead to myelosuppression, immunosuppression, and it can also have this like cytokine release syndrome or some neurological toxicity. Coming to the application, it has got wide applications, including the various kinds of cancers, solid tumors, various hematological malignancies, targeting the various cytokines, CD20, 38, and many more. And not only they have been the part of therapeutics in various cancers, they are also the further evolved to treat various infectious disorders, including the bacterial infections, fungal, viral, and including the parasitic infections. Along with that, they are also involved in the various non-communicable disorders, like in cardiovascular disorder, like PCSK9 inhibitors, in the neurology, like in migraine treatment, Alzheimer's disease, and antiplatelet therapy, and various other infectious disorders, including the parasitic infections. There are few mechanisms which are most commonly involved in the resistance to the monoclonal antibodies. Even monoclonal antibodies also go for the resistance, and that could be because of many things. One thing, it could be because of the antigen loss, because of the downregulation or the mutation of the target or the receptors involved, or it could be because of the trogocytosis, that is the transfer of membrane fragments between the cells, or the exhaustion of the complement system itself, or because of the immunosuppression, that is secretion of immunosuppressive cytokines. However, to overcome the resistance, there are various things that can be employed. Understanding the mechanism behind it, going through the various other approach like combination therapy, multi-antigen target, like in the previous thing I highlighted about the bispecific monoclonal antibodies, and also we have something like trispecific monoclonal antibodies, and adapting to the various tumor biology, and there's something called a new antigen. So I'll be talking about that one in the next slides. And coming to the challenges and limitations, immunogenicity is the one important challenges, and cost and accessibility and resistance, and of course tumor penetration is the issue with the some solid tumors. Other challenges include the complex pharmacology, requires further understanding about the molecular biology behind the interaction with the disease, and regulatory hurdles, of course, and the logistic consideration for the complex antibody formats. To address these issues, for especially the immunogenicity, monoclonal antibodies have been evolved from the murine to the chimeric, and now completely the human, 100% from the human monoclonal antibodies. And for the cost-related, affordable cost, biosimilars have been come into the market, and they reduce the cost for the affordable treatment. And something, biobetters are the one thing, they have improved their further efficacy pattern, and that made the treatment to overcome the resistance that happens by the monoclonal antibodies. And the combination of therapy is also one of the methods to overcome the resistance through multiple mechanisms. Biosimilars, as we know, they are the highly similar versions of the reference compounds comparable to the approved monoclonal antibodies. They have reduced the cost in an affordable manner and increase accessibility outreach to the global, and it have a similar efficacy when compared to the approved monoclonal antibodies. Biobetters, by definition, these are engineered improvements over the original compounds with the advantage of having enhanced efficacy with decreased adverse drug reactions and improved delivery by the various routes of administration. Coming to the, what are the future research directions in this broad area? It is a very huge and broad area. I'll just be highlighting some of the important things, what's happening. One is that the production innovations, novel engineering approaches have been employed and going on. There are expanded applications, including pioneering new therapeutic frontiers and combination. That is synergistic therapeutic approach also have been in the pipeline. And other thing like novel formats in the sense multi-specific antibodies and fragments, AI is integrated and making it the affordable cost at the global level, cell-free expression, and these are the next generation platforms. And the novel antibody, other formats includes not only the IgG, it is also they are exploring at the other immunoglobulins like immunoglobulin E and M, and single chain antibodies which are having simple structures with a maintaining the same efficacy but with a lesser associated with a lesser adverse drug reaction. And catalytic antibodies, these are antibodies which catalyze the enzymes with a greater enhancement with their efficacy. And transgenic mice platforms are the another new upcoming, which is a reliable human antibody development without humanization. So that is the other things that are in the newer frontiers. Other alternative, this I have already covered it, and other like novel conjugate also has been introduced. Antibody conjugates for targeted antimicrobial delivery for the various not only the cancer treatment, including the microbial infections, nanoparticle conjugates, imaging and the drug delivery, catalytic toxin conjugates, and antibody drug conjugates. And other advanced antibody format also include tri-specific antibodies and the nanobodies. What are these tri-specific antibodies? As I have highlighted the bispecific antibodies, tri-specific antibodies are the one which binds to 3 different targets simultaneously, and they become the.
Sorry.
Yeah,
I know
I'll be just
it's
completing
very less time.
3 to 5 minutes with a greater specificity and efficacy. Nanobodies are single domain antibodies, smaller than the conventional antibodies, and derived mainly from the heavy chains. Advantage with having the better tissue penetration, stability, and production efficiency. They are applied both in the diagnosis and the treatment of various conditions, including the African trypanosomiasis. Neoantigens are the one which are specific to the cancers, which have evolved over a period of time because of the mutation in the various receptors or the cancer cells. The various like mutation include BRAF, RAS, and here it is targeting that particular neoantigen which has been resulting in the resistance, and the advantage is very highly specific to the tumor cells which have undergone the mutation. There is one more thing, something called glycoengineering. This is the crystallizable fragment region of the monoclonal antibody is modified by glycolization, and that increase the binding of the natural killer receptors and improves the ADCC mechanisms. Other novel this already has been discussed. Radioisotope conjugates also are there. They are used in the both diagnostic as well as the therapeutic purposes. Artificial intelligence is the one which is taking a very important role in the development of monoclonal antibodies in identifying the targets, the AI algorithms which can predict the optimal targets, and machine learning will improve the antibody design, and it can predict the through the various virtual models, and finally it can become the reality in coming to in treating the and coming to them in clinical practice. And other advanced development method include single B cell isolation, plant-derived antibodies also one thing that okay, and artificial intelligence of course. Transgenic mice express the human IgG isotypes, and single B isolation. And transgenic mice that has resulted in completely instead of using the completely humanized, we can use the mice for the larger production of the monoclonal antibodies. And single B cell isolation can also be in the recent approaches, and advanced engineering approaches and various like next generation sequencing is also one of the important things that to understand the various aspects and to in a better specific targeted delivery to the specifically in the cancer biology. Another gene edited approach also has been in the pipeline. The recent thing they involved in the CRISPR technology and personalized therapy, which includes like in the genetic approach like mRNA therapeutics, nucleic acid encoded, and gene transfer technology. In the interest of time, I'll be skipping some of the slides. Antibody plus therapies, that is the one thing. That is also monoclonal antibodies also now taking into part into the CAR cell therapy, and generating CAR T cells using various variable regions, and the monoclonal antibody will be introduced with that one, and it will become the one of the frontiers in the treatment of various malignancies. And plant-derived, this approach, antibodies are expressed in the plant system too, and it is applied again for the various medical conditions. It is a low-cost production, and with the efficient large-scale production can be possible. And various fragment approach also has been in the pipeline, and various fragments can also be used, like using nanobodies, diabodies, that which have many advantages like better tissue penetration, especially in the solid tumors, and enhanced tumor penetration and access to the various epitopes of the antigens, and with the multiple other mechanisms. Artificial intelligence is the one other thing that definitely it is taking its crucial role in designing and optimization with the acceleration in the discovery process. And combination methods other like along with the monoclonal antibodies, various checkpoint inhibitors, small molecules, and they will improve the resistance. And the role of a clinical pharmacologist, that is understanding the mechanisms behind the complex biological process interactions among with the receptors and secondary messengers involved especially in the cancer biology, and designing the regimens based on the PKPD, and predicting the efficacy with the modeling the dose response relationship, and incorporating this monoclonal antibody into the real clinical practice. Finally, to conclude, the future of precision therapy is the monoclonal antibodies. They are very precise, revolutionize the therapy across the spectrum of clinical disorders, including both communicable and non-communicable disorders, address the challenges through the research and multidisciplinary approach, including the bioengineers, chemists, and clinicians, and many things. It is a multi-disciplinary task. And to finally to conclude and take ways, the significant evolution that advanced tremendously since the 1975, after the hybridoma technology has came into the in the reality, that evolved over from the mouse monoclonal antibody to the till currently in the completely fully human monoclonal antibodies, and they have got various clinical benefits with the precise specificity, with the reduction in number of ADRs, with the diverse formats like bispecific, trispecific, antibody drug conjugates. They have improved further their efficacy, and definitely there are many breakthroughs in this area. Definitely will improve the treatment for many malignancies in the coming years. With this, I thank the organizing team for giving this golden opportunity for me.
Thank you, Dr. Madhav, for the overview of monoclonal antibodies. I know that the time was very short, but that was like Star Wars movies. Anyway, but that was new to me, the plant-derived monoclonal antibodies. Can you just...
Yes, madam. This is another one area they are exploring to make it from using the plant derived monoclonal antibody to make it to decrease the production cost. So they are exploring that is the another area.
And what about the safety concern? The plant-based are much safer.
That
and
needs to be. I mean, this is the another
Still in
area that has
research.
now in the recent pipeline.
That is very interesting. And even that was shocking, that resistance to monoclonal antibodies. Because
Yeah, that is because of the various mutations and targets. Again, similar like the other like conventional our antimicrobial drugs. It is the same thing. But still, these are less likely to cause. But anyway, because the cancer biology is the one area where definitely a lot of mutations, newer biology that's coming up, and that is making to more resistant. The only way is we need to combine the combination approach that can overcome this resistance pattern.
So monoclonal antibodies are future of medicine.
That's definitely the future will be the antibodies, very precise, and that is
Only cost and its adverse effects.
only issue the cost.
Thank you, Dr. Madhav. Any questions from the audience?
Thank you, madam.
Thank you.
Can I ask a question online, or is it too late? I can drop a message to sir later.
We'll take the question, sir. Somebody wants to ask a question?
I think we can move for any questions are there. Is online. Let's see.
Dr. Divya has a question for you. She's online.
Go ahead. Please go ahead.
Sir, such a great presentation, and monoclonal antibodies is going way forward with all the development and research. So my question for you today is what is the biggest challenge you have come across with ADCs?
I feel challenge could be one is the cost related factor, and the resistance. So that definitely that is the one issue that needs to be taken care.
Yeah, I think resistance is a big thing
Yeah.
we have to work on, isn't it?
Yes.
Thank you, sir. Thank you so much.
Thank you, madam.
Now I request the chairperson and the MPS members to present the MPS Most Promising MD Pharmacologist Award to Dr. Madhav Rao.
We'll go for lunch now and followed by panel discussion. So please come back at 1:45. When we come back 1:45, we'll be having the panel discussion. So lunch is ready. Please proceed for lunch.
IT is okay? Can we start, Dr. Pule? Okay, so good afternoon, everyone. It is my pleasure to welcome all of you to this groundbreaking panel discussion on transforming MD pharmacologists from employee to employer. I am Dr. Shivamurthy, your moderator for the day. This conclave brings together a diverse panel of visionary pharmacologists who have not only excelled in academia and clinical domains, but have also journeyed into entrepreneurship, innovation, and leadership. Their experience and insights will serve as a beacon for budding MD pharmacologists aspiring to create their own niche in the healthcare ecosystem. Let's begin by hearing their perspective on how we can shift our mindset and seize emerging opportunities. Before starting, I would like to introduce myself. I'm Dr. Shivamurthy. All of you know me already. I'm currently working as Professor and Head, Department of Pharmacology at Dr. Chandrama Dayanand Sagar Medical College, Institute of Medical Science and Research at Ramnagara, Karnataka. And just to say a few words about myself, I have almost totally 23 years of experience. In that, 13 years I worked in the industry and 10 years I worked in the academia. So it's a perfect blend of both experience of working in the industry and academia. And also, all of you know that I'm involved in doing the social activity related to Medical Pharmacology Society. So my objective of building this society is that, I want to see a very confident MD pharmacologist living respectfully, comfortably, confidently, and tell the world that they can do so much. They can contribute to the world so much. So with this vision, and also to bring all the MD pharmacologists in one form, one platform, and create more opportunities, appreciate the efforts. Appreciation is the, seeding, time that will help them to gain more confidence and then go and perform confidently and with proud feeling they can contribute to the world. So with this, I started working towards developing the Medical Pharmacology Society, and I have so many now, more than 160 MD pharmacologists who have joined our society and supporting our developments. So this is in brief about myself. Now I would like to request each panelist to introduce themselves. I have very interesting panelists, Dr. Anand Patil, Dr. Deepak Langade, Dr. Saurav Deka, Dr. Vikash Medi, Dr. Poolan Sharma, Dr. Vikas Sharma, and Dr. Pratibha Nadik, madam. Okay, so I would like to request each one of them to introduce themselves briefly so that we can proceed with the discussion. So let me start with Dr. Pratibha, madam.
Thank you, Dr. Shivamurthy, for organizing this much needed panel discussion on how an MD pharmacologist can become an employer in due course. So I'm Dr. Pratibha Nadik, former professor and head of the Department of Pharmacology at Dayananda Sagar Medical, Dr. Chandrama Dayananda Sagar Institute of Medical Education and Research, Dayananda Sagar University, Karnataka. Before that, I have worked with Vaidehi Institute of Medical Sciences and Research Center and Karnataka Institute of Medical Sciences, Hubli. So after 25 years of academic experience, and of course, I was in industry for a total of 5.5 years with a blend of industry and academic experience. And always I had this to do something different. I ventured into starting my own company in the year December 2022. I have launched it, but now I am entirely into that. I am founder director of Akshay Sarthi Healthcare Solutions Private Limited, which is into consultation for clinical research, for innovations, research and development, and also the required skills to become an entrepreneur. So this is my background, and let me give more as I run through this panel discussion. Thank you, Shivamurthy, for giving me this opportunity.
Good afternoon, all. First of all, let me thank Dr. Shivamurthy and the organizers for having me here for this panel discussion, which I think will be crucial and important for postgraduates pursuing MD Pharmacology. Myself, I am Dr. Deepak Langade. I did my MD Pharmacology from BJ Medical College, Pune. Post that, I joined Government of Maharashtra as a faculty at Grand Medical College. And after 8 years of service, I left that. And in 2010, I founded a contract research organization called as Clean Search. And last 15 years is going well, which provides a full spectrum clinical services organization, all clinical trial related activities, data management, and other activities. So good to be here. I hope I would be able to contribute and help the future pharmacologists. Thank you.
Thank you, sir. Now, can I request Dr. Vikas, the most youngest and most energetic panelist among us? So can you please introduce yourself?
Hello, I'm Dr. Vikas Sharma. Thank you for organizing this wonderful conference. Though the attendance is low, but morale is still high. And thank you for the opportunity and inviting me for this to be part of this esteemed panel. So as far as my background is concerned, I completed my post-graduation MD in Pharmacology from GMC Nagpur in 2018. And even before the results were announced, I started my freelancing career. And consequently, in 2020, during COVID, in the November 27, I incorporated my company, Maverick Medicorum, a medical communication company. And as a part of medical communication company, we provide all the services related to medical affairs, regulatory affairs, pharmacovigilance, data analytics, and consulting. So during my freelance career, I have done all the types of, I have written for all the types of therapy areas, as well as all the domains. So I have a rich background. As far as professional certification courses are concerned, I haven't done anything apart from my MD Pharmacology, but still I can contribute professionally as well as I can mentor my team in all the domains. So there's no restriction as far as education or formal degree is concerned. And thank you.
Thank you so much. Now I would like to request Dr. Saurav Deka to introduce himself.
Hello, everyone. I am Dr. Saurabh Deka from nearby Bhattacharya. That was 9 kilometers away from this AIIMS. But I graduated from AMC and postgraduated in MD Pharmacology in 2008. After that, I was outside my state for last, I can say, 2008 to 2021, 13 years. So I work in industry in India and abroad, and also work in a university, Qasim University, Saudi Arabia for 4 years. In COVID time, I back and I means knowingly, unknowingly, I started my own venture called Health Vidya. So it working on both health. So I have set up my own diagnostic center clinics, and in Vidya, I started my own college, that HS Einstein Academy. And recently we are working for a nursing institute in my locality. So that was my journey. And thank you.
Thank you. Very inspiring. Now I request Dr. Poolan to introduce himself.
I am Dr. Poolan Sharma. I am assistant professor currently in Department of Pharmacology, AIIMS Guwahati. So I have done my MBBS and MD from Assam Medical College, Dibrugarh. Then I moved to AIIMS New Delhi for PhD. But during my PhD tenure, I got selected in DM Clinical Pharmacology course. So I completed that, and subsequently I was there as a scientist for 4 years. Subsequently, I joined AIIMS Mangalagiri, and currently I'm here. So my core area of expertise is your clinical research. Number 2 is systematic review meta-analysis. Then molecular biology, sequencing or genomic, using different genomic platforms, and recombinant DNA technology, and as well as genetic engineering. So those are my few of the areas where I have worked hands-on. So I am basically purely academic. So thank you.
Yeah, so actually pure academics, how it can add value to industry. That's something which we are going to, discuss. Okay, now I would like to, request if Dr. Anand Patil is there online, he can unmute and introduce himself. Dr. Anand Patil, sir, are you there online?
Yes, yes, sir, I'm here. So
We can
my
hear
name
you.
is Dr. Anand. Yeah, my name is Dr. Anand Patil, and I'm basically a pharmacologist who have completed MD in 2004. So since last 2 decades, I have been working in the field. I worked in the pharmaceutical industry. I worked as a consultant, and now back to academics. This is my uh, uh, journey from the industry to consultant to academician, and I have been working as a professor in the Department of Pharmacology. So that's my background.
Thank you so much, sir. Now, can I request Dr. Bikash Medi, sir, if online available, to introduce himself?
So, good afternoon, everyone. It's a good concept that we should motivate the Professor Vikas Medi from PGI Chandigarh. And I was there physically, but definitely I'll address that because it is why I'm saying a good concept is that everybody, when they pass out, we ask, are you going for an academy? Are you going for an industry? And you might have one session that even if you go for an academy, what is your ability? Like you want to do the best skill. Even industry also, you say, are you going for an R&D, marketing, regulatory? There are so many disciplines. And if you wanted to start a career of your own and create a facility, because I have a lot of experience with the people who actually created, so we can discuss over a period of time. And thank you very much for that opportunity. Over to you.
Thank you so much, sir. It's very nice to have both of you online. We understand that due to so many emergency requirements, so you could not be there on site. But we will be very happy to have you online and then again contribute back to our discussion. And it will be very valuable to have both of you throughout the discussion. Looking forward for an active discussion now. Let us get into our actual discussion. So the topic of our first discussion will be related to vision and mind shift. So in this, I would like to ask Dr. Pratibha Madam, what mindset shift is required for an MD pharmacologist? Think like an entrepreneur or employer rather than a traditional employee.
Yeah, so the mindset, while we become an entrepreneur, I'm telling from my experience, it didn't happen overnight. The mindset was when I started my career, it was academics. So basically, the mindset was once we learn the regular procedures of teaching, conducting classes, examinations, everything, as we all do, what next? That comes. what change I can do in this ongoing thing. How do I add value to this? How do I increase the quality? How do I increase the standards in whichever place I am, whether private college, government college, whatever. I have worked in both the places. So that mindset I had right from the beginning. That is how I went into research. And that took me to good research and development centers. So research became my core mindset throughout. Always I was inquiring what best we can do in the best available facilities. So after several years, I zeroed down to one area, therapeutic area, which took very long time for me. Actually, the youngsters can decide right in the beginning. So at that point of time, my mindset was, I should bring some change which is needed, but which is not existing. How do I bring that? So the mindset was that in the limited constraints, and it is not possible for me to continue doing the regular activities and add this passion of mine, which is also the need of the hour for our community, for our country. So from that, I decided that if I have to bring a solution, see, initially we end up doing research, publications, recognitions, everything. In the end, we want solution that can add value to healthcare. So that was my mindset, that I should bring a change. And that was my confidence. That mindset drove me out of academics to entrepreneurship.
Thank you so much, madam. So your interest towards contributing back to the society and develop your own, like whatever the thinking, what you have, you are trying to make them into product. So that is something which drove you to become an entrepreneur. So thank you so much for that, this thing. Dr. Saurav, I would like to ask you, see, you know that, in the industry, we get for so much of money and, very comfort of working 5 days and enjoying the, holidays and trips and ELs. And that you have a job security, you have, like people call you for, as a director of some company and garland you, all those things, you lost the moment you are out of that company. So now you started your own company and so how to overcome this, like egg or chicken condition?
Okay. So, as I told you, when I come back from Saudi Arabia, I joined in a startup company. It's THP, Healthcare Data Analytics, as a vice president for 2 years. So I have seen young boy from IIT IIMS, brilliant boy. They took the pain of establishing a company. It's not easy, he has mentioned. So I read some books, The More You Hungry, More You Foolish. Those books, those are entrepreneurs. They are written by IIM Ahmedabad. So, and Abdul Kalam Azad said ki top of the pyramid people who are highly educated, they have to give the employee to the society. If they become employee, who will employ them? So this employer mindset we have to create. In academic, we can innovative way, we can use more employer mindset. So currently I am giving employment to 50 people. So those are the satisfaction you never get as an employee. As you told, as your own journey, it's okay, very good. You going in a business class, maybe you cannot afford when you use your journey in a rail, means you can have to go to in a rail also. But that mindset will give the satisfaction, okay, I am giving the employment of those much people and you can really create a leadership quality in your mind. And within 5 years, I can say within 5 years, all you get back. Whatever respect, whatever something, all you get back. If you work for 5 years in entrepreneurial journey, that will never, and as you said, that job security. I can tell you that in job, it is not secure. In academic, we are thinking, okay, it's very secure kind of, but once you, in 60 years, once you get retired, every name, fame, everything will gone up, vanished. So you can think of, if I would start something, it would be till I die, it remain. So those are the thing you will miss. So if you start your journey fast and create something, it will be lifelong. Of course, there will be hiccups are there, some industry get bankrupt and everything. That may happen anywhere in your life. But if you create something, that would grow as asset for your lifetime. And that way, my mindset has been created that whatever I left, Uh, yeah. For 1 year, 2 year, I'm missing my salary. Okay, that sweet messages would come and first week of
every month.
every month. But now I have time. I have enough time to think. Now that I doesn't missed. But first initial 1 year, 6 months, you missed that. Thank
Yes,
you.
thank you so much, sir. Actually, Dr. Saurav, you really aptly told that, like if you are on the top of the ladder and still you are thinking of, taking employment from someone, then who will create the jobs and who will give the jobs? I think we should appreciate Dr. Abdul Kalam sir for, putting that into our minds, and you aptly, rolled it out in this forum. I hope it will be taken by all the MD pharmacologists. All of you, if you agree that you are on the top of the ladder, that like highly educated, highest degree you have, and still you are looking for job, then it's something which we need to think about. Okay, now I would like to ask Dr. Langre sir, Deepak Langre sir. Sir, how to start a company, sir? How to start a company and how to move out of the comfort zone? And can we start a big company directly, or should we start as a how to start?
To tell my own story about starting a company, it's a very, I would say, funny or interesting story. I became entrepreneur by circumstances, not because of my choice. So I left a very secured government service after 8 years of completion of service in a government state of Maharashtra. I left and joined an CRO, which was US based. This was the year of 2008. And I started operations in India for that company. I was the director of the company. And we all are aware that the recession came, the big recession came in 2008.
Yes.
And suddenly after my joining, after 3 months of period, the company said, no, we are going to shut down operations in India. So I thought what to do. Then they said, why don't you do one thing? You please run the entire operations by yourselves for India, for the company. I'll not name the company. But then I thought, why to do something for this kind of work for somebody else? Why not do it for our own purpose?
Yeah.
So that sense of creating an asset is what initiated or instigated me to start my own company.
Yes.
Because I had everything, all experience was there, how to start up operations, how to build a team. So with that experience with me, I thought of starting my own company. And believe me, it's not difficult to start a company in India. It's very easy. Of course, ease of doing business is not so great.
Yeah.
But
A lot of taxes,
especially the taxes, yes. But despite that, I think I was able to do it.
Government want 30% of your earnings straight away.
Absolutely, 30% is there, plus this taxation of GST, where we have to pay GST to the government before even we get the money from the clients. So of course that is difficult, but nevertheless, we should all think of having this kind of mindset and never have that shackles in the mind that why not we start our company. We should start our own businesses, and that sense of creating an asset is something which really motivates you.
Yes.
And over the last 15 years, I've realized that the one thing which I always had was motivation. So you need to be super motivated or extra motivated.
You should be, there is a saying that, you should be a lion heart or tiger heart, whatever it is.
Absolutely, because there will be a lot of challenges, lot of problems, lot of disturbances from all sides, right from your team to the government
Family
officials.
members.
So you should be able to face all those and be super motivated and perform well. So I think that is what is the biggest mindset which I think we should have.
Sir,
And regarding the size of the company, you can always start small.
Yeah.
You can always expand rather than having a full spectrum services initially.
Yeah.
Okay?
So there is a, I was going through some literature. It says that you start as a consultant or maybe a freelancer, and then like develop these, develop the confidence of your customers or the like companies who are giving you services, and slowly expand and include more number of team members. Then you can become a manager instead of directly saying that I am the MD of a company and I need this, that I'll spend, cars or, taxis or aeroplane. So wherever you require, you be cost sensitive and manage as much as possible, and then you can move forward. Thank you so much, sir, for adding value in this question. Now I would like to ask Dr. Vikas Sharma. So, like, how did you start your company? Like, what made you to think that you should start a company?
So the main thing is the ability to take risk. First thing is that, but there's a thin line between when we talk about risk, it is equal to courage, and there's a thin line between courage and stupidity. So to distinguish between stupidity and courage, you should have skill set. So you should start with small skill, just like Sir said, start small. Okay, you should develop skill set, develop expertise. Maybe just like for medical communication, start with a small article writing, which you can get skills from the medical college. You have your guide, you have mentors, everyone is there. So you can start small, and then there comes the skills, and then third is the network.
Yeah.
So networking is very essential to sustain.
Yeah.
So apart from that, the network that you have in your medical college is hardly of any use.
Yeah.
So the network that you need in the market is completely different. And apart from that, what I have experienced is that the network that you have at the moment is going to motivate you.
Yeah.
In
So
many
many times,
instances.
our own friends.
It may not be applicable to everyone, but what I have faced is that when I started my entrepreneurship, that nobody was happy. So everyone was joining corporate
Company.
sector, and some were joining medical colleges.
Yeah.
And hardly anyone was doing clinical practice who was already a part of medical college.
Yeah.
So starting on your own and being the odd one is definitely a risk.
Yeah, thank you so much. Now I would like to request, Dr. Anand Patil to, take this question. Sir, what are the services we can start offering as an MD pharmacologist if I start a company? Then, like what are the opportunities available, and what are the problems we can solve that are because every problem is an asset for us. If you are an entrepreneur, so you will always see when a problem is there, it means that is your asset. You will have to work towards solving it. That's how you will have to think as an entrepreneur. So now, what are the problems available which we can solve, or what are the opportunities, business opportunities we can take forward, sir?
So absolutely, I think you rightly said, sir. In every opportunity, in every problem there lies an opportunity. So first of all, we have to identify the problems. Now the question comes is, how do I find out the problems? I think if you carefully keep your eyes and the ears open, finding out problems is not difficult. Just visit hospital and see what are the challenges the clinician is facing. If you find out those challenges what clinician is facing and the patient is facing, because our stakeholders mainly are the clinicians and the patients, and may also be the pharmaceutical companies, because ultimately they are also catering to the patients and the physicians. So if you want to find out the problems, you have to have a inquisitive mindset, work close with them. And just to tell you that we have been always the victim of our own perceptions, and our brains have been washed completely, saying that there are only two opportunities: either join the pharmaceutical company or go into academics, because we only keep hearing this, and whatever we hear we feel is the only right option available. So what things we can do is basically, as some of the panelists say, medical writing is one option. However, we have to find a solution which is a long lasting solution, and we have to go with the market trends. For example, today companies are moving towards something called as a digital therapeutics. Now digital therapeutics is one of the areas which is coming up. So what, as a pharmacologist, I can contribute to their need? Basically, we are good in communication, we are good in research. I think we can leverage very well the idea of research, because pharmacologist is the only specialty who knows research, at least in academia as well as in industry, in detail. So if you want to go with this, just for an example about digital therapeutics. So there's a company which is called as Jogo, and they have developed some softwares. Now what this software does is, basically they provide the biofeedback based on the EMG, and then you can customize your training programs for the patients who have this muscle weakness or the bladder issues. Now this is the concept. Now to maybe validate this concept and to have the evidence in place, we need people who can guide them on doing clinical research. We need people who can do good statistical analysis. We need people who can liaise with the regulatory authorities, giving them the need of this problem. So you can become a consultant in these niche areas. One of the areas that is moving up. Maybe artificial intelligence is another area where we can provide an expertise into, because every small maybe new startups or the new innovations, they also need the rational and the evidence to be built on. And for evidence building, maybe giving them the clinical insight, validating their ideas, we are the best specialty, I think, according to me. We can contribute in terms of the research mindset, and we can develop our skill set into those areas. So those are my thought processes, apart from the traditional medical communication or the writing or the statistical analysis. These are very traditional ways people have been doing, but we have to go beyond it and see what is the next coming up in the market and develop skill set for those. So that's from my side, sir.
Thank you. Thank you so much, sir. Very nicely you highlighted various new age job opportunities that are coming up. Now I would like to request Dr. Pratibha Madam to add value to this question again.
Yeah, Dr. Anand Patil has listed down some services which an MD pharmacologist can provide as an entrepreneur, as a consultancy services. That is what you are asking, right? So rightly he pointed out artificial intelligence. I agree with whatever he says. Apart from that, since I am also working on that area, I think therapeutic drug monitoring services, we can bring out. I mean, it's not there in place right now. Few centers have got. More we undergo training and we set up centers. I have a person who has set up a research lab, a non-pharmacologist. People who have worked preclinical or in vitro labs. It's not at all difficult because I have gone through that in the R&D center. So if we have an expertise, second area could be we set up labs and provide them as a, on hourly basis. That could be a very good source of income if you have had experience in working on preclinical models. So you can have TDM services, you can have that, you can have genomic services. Somebody has come out with Genome Patri. She will screen your genes. She's not a pharmacologist. She will screen and she will tell you the susceptibility. I think even Carconos, another company. So many companies have come up which will screen and tell you the genetic susceptibility. If you know pharmacogenomics, why not set up a center of pharmacogenomics, molecular lab? Expertise is there, employ technicians, employ scientists. Not huge investment, but returns are equally good. It appears to me like that. I'm entering into that. So TDM services, genomic services, consultation, and also, as he said, in design and development of protocols, medical writing, I understand. But since we have a sound clinical knowledge, so it could make a difference as compared to other consultancy services which do not have clinical pharmacologists. We have an in-depth understanding of clinical perspectives, so probably that could make a difference.
Thank you so much, madam. And also maybe, I would like to add one more from my end, what I have something in my mind. Basically, we are MD pharmacologists, and as a pharmacology department, we are doing pharmacovigilance very regularly, and almost every medical college is enrolled into it. But we are just, restricting ourselves to collect and then upload, or maybe collect and even we don't have an opportunity to upload also because manpower is the issue, or we have many excuses to tell. So, but after recognizing that, a person is having sensitivity or hypersensitivity, so we never know exactly what is the causality. Is it really due to the drug or not? Especially like penicillin, we can avoid because we have other alternatives to it. But some drugs like, antipsychotic drugs or some which are like, very limited opportunities are there. Suppose if a person is sensitive to it, then what next? Should we treat or should we stop? Or should we do the re-challenge or de-challenge? So I think there are no de-challenge, re-challenge centers are available. And if all of us, like, as MD pharmacologists can, take some, more training related to this, how to do de-challenge, re-challenge. I attended one program conducted by PVPI. And there, one doctor from Mumbai, like I forgot the name of the hospital, they are doing the re-challenge, de-challenge activity, and they are certifying. So they will do it in a ICU setup. And if all of us master this, maybe we can start our own, re-challenge, de-challenge centers. So which can act as a very important service. And also as an MD pharmacologist, we can also give the, clinical therapeutic advisory services in a private clinics. We don't need to see the patients. Whoever want to, check whether the prescription is written properly or quality is there or any other issues are there or any better drugs are there or any new things that can be added to the therapeutics based on the clinical pharmacology concept, therapeutic concept. So if we can add value into it, maybe we can also start consultations related to that also. So maybe like, we are not getting placed in the hospital, but if we start, independently and start offering, if we are successful, maybe like even the clinicians will start accepting us. So that can be one of the opportunities which I can think of. Now I would like to ask Dr. Vikash Meri sir. Sir, I would like to ask you, since you are a well-known academician, what are the skill sets we have to incorporate to our MD pharmacologists to become more competent, both to become, subject experts, subject matter experts, and also the entrepreneurship skills. So can I have you, sir?
Thank you, because I think one thing is here that how do you get an importance? You get an importance in a society or scientific community if you give a service. So we always tell student that what are services you can give. Like Madam has given therapeutic drug monitoring, you said pharmacovigilance, okay, and drug information center in a hospital. But we have to look at little forward. So if I look at little forward, okay, what are the expertise we have for the devices? Very few. What are the expertise we have for a food or a use product or other division? Very few. So can we develop an expertise there? But otherwise, the theme of this discussion is that can you think like a industrialist or you become an employer? So I'll just give an example. When I was AIIMS, one of the person, he was a PhD. Right now he's sitting in London. So he created a facilities for, pharmacovigilance company. The branches are there in Delhi, city, they open up in Ahmedabad also. There are a couple of people who actually initially decide that they will go for some of that opening the company. So it has to be required determination. Otherwise, she's been my grant from Birexy open up a company. So here the problem it comes is that you have to think like you can think that you can create a company very easy. You can create a company. But when you're going to establish sustainability and expansion, that is what. So you work on one product, so it may not be feasible to run this particular company with, one product. Maybe you can say that if you give a service like pharmacogenomics, you need a huge money. You have all an instrument and how recurrent costs will be there. So that you have to plan it. And if you start doing writing a paper publication where a lot of people like in Chandigarh, maybe a couple of people who are doing it, and this becomes so easy. Then later on you have to have a expansion program. But now the government thing has been changed. Like you have seen that, this I have incorporated CRO and SMO. It was not there. So if I look back in 2008 when we recommended this compensation, I also put the recommendation and CRO registration should be there. So all these people right now they are doing a registration. Okay. So why we are making it like that so that officially it has been recognized that the CRO, SMO, so that you get a business opportunity and obviously it is recognized. But what I'm going to tell you is the government itself is creating a company. Government itself is creating a company. Like I'll just give you an example. NIPER has created a company. So definitely all of us, like I'm looking for 3 decades, we have a lot of potential compound, but we are not into the product development mode. So now the government is totally into product development mode. You might have seen that ICM created a couple of centers for product development. There is a partnership. Even if you look at the NITI Aayog, they say that you open up a company and if you have an academic collaboration, the tax relaxation will be there. So an opportunity to look at BIDAC or other DSR, how you happen. Like first grant I got it is 1.25 crore. When we used to get, salaries around 2,500, 3,000 rupees. It was a simple technology to make a controlled release for pneumocellite. We got a grant of huge grant. Okay, so you work on a technology like Dr. Anand had said, software and all. That is it, but that very, very competitive because market is competitive. You come up with a software, it can be utilized, but that you need to look at that how long it is going to last and competitive one. So so there are a lot of options, but only thing what I say the motivation. Motivation is you might think today I'm going to do it, but that motivation has I can do it and it should be sustainable. So options are a lot of options are there, but you only get a recognition, you give your service, and importance will come. People will recognize, and society will recognize, scientific community will recognize. Over to you.
Thank you. Thank you so much, sir. I would like to ask Dr. Poolan, you are working in a very important area of especially pharmacogenomics and bioinformatics, many things. And what is the since that's a area where you have already generated, I think 10 plus patents, if I'm correct. So I would like to ask you, what is the value of patent? Because that's what you can get from an academics, which can add value to the industry.
Sir, actually, because one of the basic thing is like intellectual property. That is very, very important because that is the first step beyond which actually you can go for like entrepreneurship or any kind of startup. So your securing patent is very, very important, or intellectual property rights is very, very important. It maybe copyright or any other, like for artificial intelligence or some other platforms, copyright may be a better strategy, or if you can integrate with some device.
Yeah.
So the same thing can be actually further validated, and you can come up with startups and all. So many of us actually are doing. We are actually doing in academic setting, because many of us like like many of us are doing actually with artificial intelligence, using that in for clinical research and all. So that same we can, I think, go for the next level, think big, and we can go for startups. And
so thank you so much. I think as you rightly mentioned, intellectual property is the real property, so that nobody can steal. Okay, and once you register, and if it is a highly profitable one, sometimes if you see paracetamol, I don't know who invented it, and I think whenever he maybe I don't know how many years, and he is earning since then. It's very simple. So once you invent something, register in your name, and it is available for your lifelong. Yes, there will be expiry will be there, but people are very clever. What they do, they modify it, they innovate, they keep on coming up with, like initially they might have started with just a powder, then get into some formulation like a tablet. Maybe now we have dispersible tablet, whatnot. You find anything, any different routes that is available. So now I would like to ask you, madam, this will be my last question in the interest of time. Just to conclude, I would like to ask you, how can we combine the industry, academia, and there are a lot of incubation centers and all those things. Since you are I have worked with you very closely, and you have been already working, and now you are you must be working more. So what are the opportunities available for an academicians to transform them? Because one day suddenly I cannot become an entrepreneur. We have to move slowly, and we have to, if we have to, like even aeroplane, if it has to take off, then it will slowly increase its speed and then take off. So now, how do we start, and where do we start, and what are the opportunities, government fundings available? Maybe if you can summarize, it will be very useful for our people.
Yeah, actually, the first one which is available now is from Department of Science and Technology. It is called Nidhi Prayas. And Department of Biotechnology has come up with some schemes such as PACE and SBRI. Like that, there are opportunities for an individual researcher to get funding up to 50 lakhs. But what they look at it, can I take 2 minutes to just describe them? Because first when I applied for Nidhi Prayas, actually my research was just a simple estimations, as I mentioned yesterday. But then I came to know about Nidhi Prayas. I said I will develop artificial intelligence model, predictive analytics based on this data. Then the department, DST, Nidhi Prayas said, you cannot unless you have a hardware. So they want a product. It's not a software product, they want a hardware. So we started building a spectrophotometer. So The Nidhi Prayas is 10 lakhs is available to start with, but you must build a small product. It's not difficult actually. Afterwards, I realized. Initially, I thought you can think of small product. Actually, I was discussing with, if I can say that example, sir, Shantanu Tripathi, sir, his son has brought out a product which measures adherence. Very nice, innovative. And I think that started with Nidhi Prayas, sir. So they also, we just interacted. So measuring the adherence, measuring the pulse, wearables, these are just, we have to change our mindset. Within 5 lakhs, we can produce a product. And my daughter is in NCAM screening. They do such, engineers are doing such wonderful projects. So first thing is, Nidhi Prayas 10 lakhs is available, but tie up with engineers, biotechnologists is important. Idea is yours. Requirement is, idea is ours. Requirement, we know what the patient, usability, everything. And as somebody said, we should not be stupid. It should be viable, sustainable, all those. So that it will push you there. So Nidhi Prayas is first one. And many doctors are getting into that. Then after that, a big grant is there from BIRAC. Once you get the results and all that and you apply and show, there you are literally pushed, Dr. Shivamurthy, there we can get into all those skills which we said, financial, this thing, marketing, to study the market. Is it sustainable? How do you scale up your manufacturing? N number of questions, but the beauty of DST and DBT is that they mentor you. They mentor you so well, as I have given at least 15 to 20 presentations, and we should be prepared for failures. I have failed at least 10 times. In the first Startup India Seed Fund, I have failed because I didn't know. I was involved in academics. I started, as you said, when I was doing I was HOD there. So I missed out. You should apply within 2 years of your registration. I missed out. Somebody said, Madam, 3 months are there, you apply. But there were so many hitches. By then, I mean, few incubators. I'll tell you what are those incubators and all that. So that I missed. So basically, for an academician, Nidhi Prayas will be a very ideal thing to apply, but you must have a product in mind, innovation, very simple also, no problem. Little bit of, some electrical, electronics, tie up with biotechnologists, maybe simple estimation of a biomarker, something like that related to pharmacology or whatever you can do. So that is one thing. Then after that, during that, they will ask you to apply for incubators. There are many incubators in the country which work as independently funded by the central government. You should show an attachment to them and they will fund you. Through them, the government gives you funding to do this development, and they help you also. So that is about incubators. So startup will register with the DPIIT, central government, and then apply for incubator. Incubator will fund through the government funding comes through the incubator. Together you work, submit that report. After that, you apply for patent, get that. Then you apply for other fundings. One is big grant, base grant, SIBRI, all this along with academics you can do. If you don't want, if you want to come out, then you look for agencies which will fund you, investors. They are angel investors. But until you show some proof of concept, and that's called technology readiness levels, if you go up to 5, 6, and it's really performing well, then the investors will come, but they will ask the share. That's called equity. They will ask you to give them some, if you're prepared for that, you can go for that. Or best would be to go through this DST, DBT channel. And then there are multiple other funding agencies, which are called Nidhi, EIR, and all those. So as you run through that, you'll come to know. Be attached to some incubators, which will mentor you, which will give you support. So something like Bangalore Bio Innovation Center, CCAMP, and Venture Center in Pune. And these are the ones which I have interacted with. These are very good. So you should have an idea like that. So as an academician also you can start. You can still keep doing it, but definitely the time involved will be more. So that was one of the reasons which I felt. But if you are able to now, Dr. Shivamurthy is working, he's doing 2 jobs. He's doing conference jobs, MPS jobs, and for you it will not be difficult because you work extraordinarily. After your working hours, you sit and work. So for you, it is not going to be difficult, but for women, I should not make gender bias, but we have housework to manage. After the working hours, I cannot sit and do industry work. I have other work to do. So better to come out and start if you can afford. But there are other issues like financial and other things, if you can manage that. But definitely it is a hard work, but there is a scope. Second thing, before I wind up, institutions are not against setting up startups because it's adding value to their NIRF ranking. They want you to start a startup. It should be in the medical colleges. They encourage, and there is no conflict of interest. If you do a startup which will train medical students, then there is a conflict of interest. Here you are bringing out a product, and their institution also gets a boost. So they are encouraging, and they may even give you some time to do. So for if you have time after doing 3 to 4 hours of work and you delegate other academic works, it's worthwhile venturing into this kind of funding agencies. But that pitching is a very, very tough thing, and Akshay Sarthi can help people to make good pitches also.
So thank you so much, madam. I think you added a lot of value in the final, remarks. I think we have learned a lot. There are so many, grants are available, and I think for the, ladies, government also have special programs also. Women entrepreneurship related. ICMR is also. So like this, so many opportunities are available, and we are very comfortable working in Department of Pharmacology. So I think like you can use you can work in the department. See, I feel that now I'm already 10 years in the academics. So now I understand that almost all the PPTs are there with me, and you call me at any time, I will go and take the class. So because being for MD pharmacology, I've been studying pharmacology since last 20-25 years. And I should be comfortable and go take the class at any time. So what are you going to do next after taking the class? So can we start thinking a little bit beyond, and it is for your own. You can create assets. You can create intellectual property. You can create books. That's what we are doing through the society. You can create many things, like many things, many assets, and which will bring you more respect to. One is immediate benefit is respect to you, and then people will look up to you, and then people will call you for call you as the role model, and management will respect you. And I think you should not go behind money. Money will come back on behind you. So with this, I would like to in the interest of time, hope we, and this is just I want to make a note here that this is only a seeding meeting. This is the first meeting what we are discussing in this. And I promised Dr. Anant Patil that we will be conducting workshops on this. Okay, we will be inviting the experts of finance or any other, incorporators, directors, or like company-related people, experts in this, and we will be creating series of programs. Maybe not immediately. We will go step by step, and as we also need time also. So considering all these opportunities available, and if any of you are ready to join with us for contributing in this line, basically we will welcome you, and we will grow together. Okay, we will grow together. That is one. Second thing is that what I wanted to tell that see, we will MPS. I feel that it will be successful if we can have at least a few successful entrepreneurs in in the few upcoming few years. So that's something which I always envision, and I hope all of you will grow with this vision, and then make it possible, and hope our MPS will conduct more such programs, and I request all of you to contribute more in this direction, and so that we can make independent, confident MD pharmacologists. With this, I would like to end. Thank you so much. Now, can we respect our panelists sitting here with mementos, and we can close the session. Thank you, Dr. Melvin. So you would like to add any? Thank you. Any questions, audience questions before we close? Post lunch, I think, hope we had interesting, encouraging discussion. Online also, if anyone is there, anyone can open. Let me see. Any questions out there? Please type in the WhatsApp group.
Shiva here. I have a few questions.
Can you be loud? Increase the volume.
Okay.
Yeah, your good name,
Is it better, sir, now?
Yeah, it is audible. Can you spell out your name and organization?
Yeah, this is Divya here. I'm from UK. I'm working in AstraZeneca.
Yeah.
in vaccine and immune therapies. Sir, my question is about the genetic testing. One of the speaker, I think Pratima ma'am or someone mentioned about it,
Yeah,
about
Dr.
genetic.
Pulen, and also Dr.
Yeah.
Pratima madam.
Yeah, and how much is it really happening? And I do understand there are a lot of cost factors, which is stopping from people getting through this genetic testing.
Genetic testing actually can be done in a very cheap way also, but that is like standardization problem is there. But if you want to go for NGS techniques, so NGS techniques are actually that require lots of samples. Number one is which machine or which platform you are dealing with. So like if you are taking like next platforms or other high or performing platforms, then the cost will be really high. And whether you are doing some specific genetic sequences, like let it be 10 genes, 20 genes, 30 genes, or clinical exome, or what you are taking actually. So now the cost is comparatively decreased actually. And we are hoping that with further advancement in technology, it will further decrease. But if you are targeting a single gene or single mutation, we can give it at a very low cost actually. But if you are going for a whole genome sequencing, then that is going to take cost. But at the same time, class, with the advent of your Oxford Nanopore and other platforms, and mutation-based screening is also coming up with those platforms. So we are hoping that the cost will decrease. And again, another thing is like that you can design the probes also. That is another point of your intellectual property. So that is another domain where workout can be done, and that can secure your position in the diagnostic technology division also. Thank you.
Thank you, Dr. Fulan Sharma.
Yeah,
But
Dr. Divya,
how?
yeah, Dr. Divya,
Yes,
the
ma'am.
testing in Bangalore, when we test for some mutation, we send single site, is costing around 2,000 to 3,000 per sample, isolated samples, if you want to do some testing. But there is one company, as I mentioned, I'm not very sure, which gives you multiple genes for, I think, 3,000. You will come to know N number. There's a big list of genes which are being tested. So it ranges somewhere from 2,000 to 3,000 is what a commercial company, I think MedGenome, if I'm not wrong, because we had referred from the Anand Sagar. This is what is prevailing in Bangalore and what he said in addition to what Dr. Fulen has added.
Thank you, ma'am. And so
Dr.
is it widely available in India to do the testing, genetic testing to kind of, think in the direction of precision medicine?
Yes, available.
Okay.
Okay, if
Perfect.
no
Thank
further questions, I think we can conclude. And definitely, no, please, our WhatsApp group is active. So at any time, please feel free to, ask the questions. Experts are there in the group, so we can always answer. Thank you so much. With this, we come to the end of the panel discussion. I thank all the panelists, esteemed panelists, traveled all the way for contributing to this conference. Please give them a big hand and encourage us to come up with by participating in future programs also. And I would say I hope we have encouraged you to take up the independent entrepreneurship roles in the future. So we can definitely add value to not only to pharmacology profession but to the country. Thank you.
Hello. I request Dr. Satyajit Mahapatra, sir, additional professor and HOD of Department of Pharmacology, AIIMS Guwahati, to kindly felicitate the speakers from in the panel discussion. Thank you, sir.
Start our session. For this session, please join me in welcoming Dr. Mukundam Bora, Assistant Professor of Pharmacology at Guwahati University, Guwahati Medical College. With 11 years of teaching experience, Dr. Bora has been actively involved in organizing conferences and CMEs and has published more than 15 research articles in renowned journals. He has also contributed to the first multicentric point prevalence survey of antibiotics used in India. Let's give him a warm round of applause. Our second chairperson is Dr. Swapna Fukan, Professor and Head Department of Pharmacology, Guwahati Medical College. Ma'am, please. She has more than 20 publications at national and international conferences. Please give her a warm welcome. Our third chairperson is Dr. Satyajit Mahapatra, Additional Professor and Head, Department of Pharmacology, AIIMS Guwahati.
Good afternoon, everyone. So we are going for the last session of this conference, that is the session 10. And we have 3 speakers. The first one we are going to start with Dr. Sonia Kakkar. She has done her PhD in forensic and now she is a faculty in the Central Arizona College. and she'll be talking about forensic pharmacology. So we'll start with the first session. Dr. Shania can start forensic pharmacology topic. Over to you, Dr. Sonia.
Hi, good morning. Sorry, good afternoon, everybody. It's good morning here, so I was just a bit confused. Are you guys able to hear me? Is everything
Yeah, we can
audible?
hear you, but we cannot see the content. Can you share the
Oh,
slide?
yeah, I would just, yeah. Um, uh, good afternoon, everybody. I understand you had a long day, so I won't be taking a long time to complete this information delivery session. It would just be like 10 to 15 minutes. So it would be about an information dissemination session on forensic pharmacology. So a little something about me here. I worked as a forensic analyst in Central Forensic Science Laboratory, Ministry of Home Affairs, Government of India. I served as a guest lecturer in Central Detective Training School as well, which comes under Bureau of Police Research and Development, Ministry of Home Affairs. I completed my PhD in forensic genetics, and I developed a novel method during my PhD. This method was about AMCOS. We call it allele matching cutoff score method for human identification in mass disasters for forensic purposes. This method was published in Nature Scientific Reports. Special credits to Dr. Poolan Sarma for helping me with this. Without his contribution, this wouldn't have been possible. So, I mean, after becoming a mother, my career took a different trajectory. I pivoted towards a role that would provide me a better work-life balance. So I'm currently serving as a dual enrollment faculty at the Central Arizona College. I'm also serving as a science advisor at a government school district in Arizona. I was facilitated by the Arizona State Teachers Association for my contribution in next generation science standards. I also recently published a genetics chapter aligned with the United States Next Generation Science Standards with Mr. Brett Molding, who happens to be the director of the Utah Partnership for Effective Science Teaching and Learning. So now I would be introducing the forensic pharmacology. I'm not really a pharmacologist, but I would be talking about the forensic aspect of pharmacology, which is usually not much talked about in India. So forensic pharmacology is the application of pharmacological principles and knowledge to legal matters like drug abuse, sports medicine, and doping, off-label drug use, drug regulatory processes, etc. So it involves the study of how drugs and other substances interact with the human body in the context of criminal or civil investigations. So skimming through the surface, forensic pharmacology investigates the link between chemicals and an individual's behavior, illness, injury, or death. It involves interpreting drug effects and their duration of action to aid medical legal processes. It plays a crucial role in both criminal and civil litigation by interpreting how substances, legal or illicit, may have influenced a person's behavior, decision making, health, or death. Forensic pharmacology reports can assist attorney and jury or judges in the decision making of cases pertaining to interpreting drug effects, establishing cause of death or impairment, assessing drug interactions or medical malpractices, determining compliance or misuse, evaluating reliability of witness testimony. So, forensic pharmacology versus forensic toxicology, I mean, there happens to be a lot of confusion that prevails between these two fields. Usually one is mistaken for the other. People can't really tell them apart. So as you can see, these two fields are not very much similar. I would just be going over the differences between these two fields, a visual comparison, as you can see. So forensic pharmacology primarily focuses on the study of how drugs interact with the body and their effects on behavior, cognition, and physiology. On the other side, forensic toxicology focuses on the detection and quantification of toxic substances in biological samples to determine their role in death, impairment, or poisoning. So now the key roles of forensic pharmacologist versus the key roles of forensic toxicologist. So forensic pharmacologist assesses how prescribed or illicit drugs influence actions or decision making, whereas forensic toxicologist identifies and measures drugs, alcohol, poisons, and other chemicals in blood, urine, hair, and tissues. Forensic pharmacologist evaluates drug interactions, adverse reactions, and therapeutic outcomes, whereas forensic toxicologist determines the presence of concentration of substance to infer their effects. So forensic pharmacologist provides expert testimony on the pharmacokinetics and pharmacodynamics of substance, whereas forensic toxicologist collaborates with medical examiners to establish cause and manner of death. Now, applications in the legal system. Forensic pharmacologist determines whether drug use impaired an individual's ability to commit a crime, whereas forensic toxicologist confirms the presence of substances in cases of suspected overdose or poisoning. Forensic pharmacologist assesses the impact of medications in cases of overdose or poisoning, whereas forensic toxicologist provides evidence in driving under influence DUI cases to establish impairment levels. So forensic pharmacologist analyzes drug interactions in cases of medical malpractice or adverse drug reactions, whereas forensic toxicologist assists in postmortem investigations to determine the role of substance in death. So as you can see, this is the summary of both fields about their primary focus, key activities, legal applications, expert testimony. We just talked about it. So now coming to the workplace differences for forensic pharmacologist versus forensic toxicologist. So forensic pharmacologists primarily focus on drug actions, side effects, interactions, and behavior impact, whereas forensic toxicologist focuses on the detection and analysis of toxins or drugs in biological samples. So forensic pharmacologists, for them, the common employers in India are medical colleges, research labs, pharma companies, and National Forensic Science University. While for the forensic toxicologist, the common employers are forensic labs like Central Forensic Science Laboratories or State Forensic Science Laboratories, police departments, like I just talked about, Bureau of Police Research and Development, and medical examiners. So the lab type that they work in, forensic pharmacologists work in the pharmacology labs, academic research settings, whereas the forensic toxicologists work in the toxicology or forensic science laboratories. So working with patients, working with patients, forensic pharmacologists, they get to work with the patients sometimes in clinical or hospital-based settings, whereas forensic toxicologist rarely works with the patients, mostly work with biological samples post-mortem. So forensic court involvement for both the forensic pharmacologist and the toxicologist. Pharmacologists basically provide expert witness on drug effects, prescribing practices, whereas toxicologist provides expert witness on poison, toxin detection, and cause of death. So the sample type that are handled by forensic pharmacologists are drug formulations, medical records, clinical trial data, whereas for forensic toxicologists, the sample types handled are blood, urine, hair, tissue, stomach contents. So forensic pharmacologists, the associated field for forensic pharmacologists are clinical pharmacology, pharmacovigilance, and forensic medicine, whereas for forensic toxicologists, the associated fields are toxicology, analytical chemistry, forensic pathology. So talking about the research orientation, forensic pharmacologists are strong in drug development and behavioral impact studies, whereas forensic toxicologists are strong in poison detection and metabolism. So in summary, forensic pharmacologists are more theory, behavior, and drug effect oriented, often found in academic and clinical environments, whereas forensic toxicologists focus on chemical analysis in forensic or medical labs dealing directly with physical evidence, like physical crime evidence. So talking about the career opportunities for forensic pharmacologists in the United States and other countries, like the workplaces that forensic pharmacologists usually work at in United States are medical examiners' offices. They assist in determining cause of death related to the drugs. Like I know somebody in New York, Office of Chief Medical Examiner. They are a forensic pharmacologist working over there in New York. I barely knew about forensic pharmacology before I met that person, before I happened to talk to him about this field. And I just knew about forensic toxicology. So yeah. So law enforcement agencies like Federal Bureau of Investigation, Drug Enforcement Administration, they advise on drug-related crimes, overdose, deaths. So they also work at forensic laboratories, collaborating on drug analysis and interpretation of results. Academic and research institution, they teach, research, and publish in forensic pharmacology. For expert witness they can also work for expert witness consulting firms testifying in court on drug effects and medical legal issues. They can also work at poison control centers, supporting toxicological assessment and public health. One thing I haven't mentioned here is like they can also work with border security and customs. But if you have to be working with them, you actually need citizenship or the permanent residencies because they do not really provide visas for work-related visas. So common employers for forensic pharmacologists in United States and other countries, especially about United States, it's National Institute of Health, NIH, Centers for Disease Control and Prevention, which is CDC, Federal Bureau of Investigation, and private forensic consultancy firms. So the education background that you need to have to be a forensic pharmacologist in the United States is Pharma D, MD, or PhD in pharmacology or related field, or you need to have specialized training or certification in forensic sciences. Example from AAFS, which is American Academy of Forensic Science, or ASCLD, which is called the American Society of Crime Laboratory Directors. So yeah, talking about the salary range for the forensic pharmacologist in the United States is really impressive. Actually, I was thinking to switch to a forensic pharmacology course after I looked at the salaries, but I think it's too late for me. So the entry-level career stage is $70,000 to $90,000 annually. Mid-career is $90,000 to $120,000 a year. Senior expert, like who has an experience of 8+ years, can earn anywhere from $120,000 to $160,000 annually. An expert witness, like per case per day, they can earn up to $300 to $600 per hour. So it can exceed like $2,000 a day too, depending upon the expertise. So, and the salary ranges actually vary by the employer type too. If you're working for the government agencies like Drug Enforcement Administration, CDC, Medical Examiner's Office, you can earn anywhere from $85K to $130K. Academic institutions have this range. And private sector consulting has $100,000 to $160,000. So legal expert witness services, it highly varies, often $2,000K plus annually if working independently or on a complex litigation. So that's about it. Thank you, guys.
Okay, thank you, Dr. Sonia. We'll take up the questions at the end of the session. So,
thank you.
Yeah, thank you. Next, we'll invite the speaker 2 for this session.
So I think the next speaker is Dr. Dibyajyoti Saikia. He is an associate professor, Department of Pharmacology, AIIMS Guwahati. His area of interest are clinical trials, biostatistics, bioethics, machine learning. And he has 20 number of publications to his credit with 4 patents. And he's a guest faculty on clinical pharmacology at AIIMS Guwahati and member of ethics committee, Apollo Hospitals, Guwahati.
Good afternoon, everyone. This is I am as my thank you, Chairperson, for introducing me. And I know it's getting to the end of the conference. Very few people still remaining. I hope that some more students would have been here. But still, we'll go on. For my aim of this presentation was to emphasize that currently we are in the cusp of great revolution in IT sector as well as in information technology and artificial intelligence, and a lot of things are being claimed as well as a lot of opportunities are there. And I've seen that many people think that it requires a lot of investment or expertise to use these new technologies. My aim is to, as the title says, that demystifying is to make it more relatable, more user-friendly for clinicians, pharmacologists, and more from medical fraternity to be able to use machine learning algorithms. So, lot of jargons are there. I'll try to minimize the jargon into more relatable terms so that we can all use these things. Many people claim, or you can see in the social media, that many things are possible, but many things are not possible. That, like, for example, these are a lot of terms AI generated, but it has committed many mistakes also, like big data. It's not a word. It was supposed to be big data, but it has made those mistakes. But again, it has a lot of powers also to solve many problems. So first, I will start with, as it's evening, so get into a lighter mood. I will ask question also. So you must have seen this, so you all remember from the movie 3 Idiots. So what was this scene about? Was the definition of a machine. So as we all know what is machine, so what Amir Khan told simply was that anything that solves your physical work problem, so that is a machine. So what is machine learning? Our context now is what to do for our mental or cognitive work. So that is where your artificial intelligence. So artificial intelligence is also something that is aimed to decrease your or our mental or cognitive workload. Basically, 3 types are there. We all have heard these terms: machine learning, deep learning, and artificial general intelligence. AGI, as of currently, is still some maybe years, or it's still debatable whether it can be achieved or not. But other 2 things are working, as well as people many solutions to the many problems have been achieved through these works. But before using these tools to solve our or decrease our load on our brains, we should know how does a human brain works. So many of you have come from far off places. So I'll try to guess what was your question. Then when you first thought that you are visiting AIIMS Guwahati, so what was the first question? So I guessed that the most commonly it could be that, what is the distance of AIIMS from airport? So somebody, you must have asked somebody or Googled it. So answer might have come as 30 kilometers. Next question could have been, are you going to visit the national park, Kaziranga National Park? Its answer could be yes or no. So these type of data we already know. So we call those as continuous variable, 30 kilometers, 31 kilometers, or categorical or nominal. Same thing in machine learning. The terms more commonly used are, for same thing is the either for continuous variable, we call it as a regression, or for sorry, or we call it it's a categorical label or a classifying task. So in basic, any machine learning model has the same, this principle that you put some data, train some model and make it do these two work. Either you quantify something or you classify something. And this is what is, in short, machine learning. But then again, the machine has to learn something. So artificially, we have to teach the machine. But again, to understand how we teach it, we can also go back to how we, or what is the natural way of learning. So you all must be aware of that. We have 3 types of learning. Operant, classical conditioning, like the Pavlov experiment, that you associate something without telling anything. You give them the data, people will associate, even dog will associate that, okay, bell is associated with food. Even though there might be no connection, there might be connection without telling anything. Second is operant conditioning, that rewards or punishment or with reinforcement, we try to change some behavior. And third is observation-based learning, that is the most commonly as how we humans learn. For example, in this picture, a kid learning to do exercise just by observing. No reward is involved, no punishment is involved, but just by seeing or looking, you tend to do the same thing. So these are 3 types of learning that we go on. Same thing for machine learning or artificial learning, we use these 3 principles. So their counterpart is supervised learning. So when you tell something, so machine, we tell the machine that, okay, this is what it is, this is what it is not. For example, in the picture, this is an apple, we show some pictures, and then you tell the machine that develop a model so that you can identify this thing as an apple. So this is called as supervised learning, that you are supervising and you are telling what it is, so that any new data comes, you can correctly identify. Reinforced learning, most of the time we are the person that are reinforcing. So most people, we use ChatGPT nowadays. Every time you give a task to ChatGPT, an answer comes, and in the below it will be one thumbs up and one thumbs down. So this is an example of reinforced learning. If thumbs up is there, the model will know that whatever it has produced is correct. So their algorithm will continue to do so. If you do a thumbs down, then the machine will learn that, okay, whatever the output was produced was not correct. So it might need to change the algorithm which was used to achieve that output. And finally, we have unsupervised learning. So here we just give them the data and ask them to classify it as seemed fit by the algorithm. So we'll see what is the unsupervised learning actually looks like. So I will give an example of, let's say we have some values X and Y, and let's say this was an Excel sheet. We just collected this data and we made a scatter plot, again in Microsoft Excel. So this type of graph will come. So we have not told anything about what is this type of data, what it is, but just we plotted a scatter. Now if I ask, try to divide this into 2 groups. So how do you do it? So what I did is, for me, I did it in 2 groups. So for me, I thought that, okay, these types could be 2 groups of data. Same thing, any machine learning algorithm for clustering will do that, okay, divide it to. If I tell it to divide, let's say, for 3 groups, it could be 3 groups. So if you give the data and do an unsupervised learning to any machine learning model, it will give you the clustering. But what meaning can you get out of this? You cannot get any meaning out of this unless you know what the data is. So you can divide it. But if you know what the data is, so what this data is? This data is till yesterday, the purple cap and orange cap of IPL. So x-axis is the average, betting average, versus y-axis is the number of wickets taken. So this was the data, and if you see, so this grouping, now you can make out the meaning of it. What is the meaning? So left sides are bowlers, right sides are batsmen. Or if you see the 3 groups, you can call that one group could be all-rounder. So this was a small data set that I can do. If you have a very large data set and you do not know anything, Then you can use this algorithm to find patterns or groupings that was earlier not obvious to a human being. So this is one type of machine learning that can be used. We'll come later where how to do those things. Other algorithms like linear regression. These are many commonly we have all used in our research studies. Best fit line that you try to do. In machine learning, what is it? It is a type of supervised learning. However, earlier one was unsupervised. This is supervised. By this, I mean that we give the answer beforehand, that this is a data set. Your regression, the machine must put up the formula that will give that answers. So after you train it long enough, any new data point you add, it will put into the formula and then give up the results. Sometimes the association is not linear. It could be some type of curves. For curves, we will use the polynomial regression. So these are some types of regression that are used. So here, formula will change. There will be some coefficients will change as well as the power. So what is our importance in here? So we put any data to any software, some results will come out. It is not only that whatever the result is coming is valid. So for that, unless you yourself know the topic well or the subject is you are well versed and you have some idea, you will not be able to say whether this is a valid result or a invalid result or what meaning or interpretation you can get out. So most common problem is you can have a large data set, but many of those data set features or parameters or variables could be expressions of the same outcome. For example, if you have in one column, you have hemoglobin levels, another column, you have presence or absence of anemia. You can say that I have used 100 parameters, so my model must be good. But many of those parameters will be the same parameters, or they are multicollinear, meaning they are all expressions of the same thing. Many biochemical as well as CBC parameters are derived parameters. So when you select the input for any model, you must know your subject well enough to select that, okay, these are the models. And to correct those, some of those methods are used like ridge, elastic, less and elastic. I will not go to them. Ridge is just like, if you know something are correlated, we decrease the power of the coefficient. Lasso means that we just remove that parameter which is very much associated with the other one.
Dr. Deposit, you have 5 more minutes in your hand.
Sure, sir. For logistic regression, with the same thing we do, what we do is that it is a regression model. It should have been used for quantification, but logistic regression, what you do is you put a threshold in here and divide to yes or no or positive or negative, so that it is used for a classification. Another algorithm is decision tree. So decision tree, what it means that you start with your own parameter. So if let's say you have a 10 parameters and one output. So it will be type of a supervised learning. You give the output and you give the parameters. Decision tree, the algorithm will go on correcting that which decision yes no question will result in the output. So when you make a decision tree, for example, I'll just skip some of the slides. You have parameters you put and you check whether those results are accurate or not accurate, and then you take finally the output. So when you train and then you give a new set of parameters, it will decide which will be the output. So that is one decision tree. Another term you will hear is the random forest. It means that you are using the same data set to train, but multiple trees. So a collection of tree makes a forest. So this model is known as random forest. Again, if you want to do regression, that is quantification, you will take the average of all those things or median, depending on the scenario. Or if you want the classifying, so if you have 3 models, you will take the majority vote, 5 models again, majority vote. So this is what is a random forest. Support vector machine, what does it do? Let's say you have 10 parameters. 2 parameters is very easy to visualize. Our human brains can do. 3 parameters also, some intuition we have. However, if we have multiple parameters, then you cannot imagine the vectors or how they are spread. But for a statistical software or for a machine learning algorithm, it is easy. So what they will do is supervised learning. You will tell this data set belongs to this group, this is to this group. This algorithm will divide the plane in such a way that whenever you put any new data and you ask which group it will be, so it will divide it. Okay, I'll skip it. I'll directly come to the uses. For us, what is important is which algorithm or which type of learning, machine learning, can you use for which thing. So supervised learning, what we can use is what we have to give. We have to give input-output pairs. We have to give data first of what are the parameters and what is the output that is there. In what clinical scenarios we can use? For example, we use for biopsy images. So if you feed biopsy images and you say that this is cancerous or malignant, non-malignant, and this is the image of non-malignant, 1,000 images of malignant, non-malignant, that is a type of supervised learning. Similarly with, if you say that this is responsive, this is not responsive, and you give the parameters, then you can, when a new patient or new image comes, then the machine learning can use it to identify. Unsupervised learning, where you can use, we can use it in rare diseases. For example, you have diabetes type 1, type 2, sometimes type 3 also. So if you do not have that many data points of diseases, you have parameters of patients, and you can give it to a machine learning algorithm to try to find out patterns that are not obvious. So it can group rare diseases into subclasses of the rare disease, which for a human it might not have been possible. Similarly, for anomaly detection, you have a EEG or you have a EEG of 1,000 patients, and you want to find where there is anomaly. So it is unsupervised. It will find that, okay, these things are different from this thing. It is an anomaly. Is it significant? That you as a clinician or a researcher have to find out. Reinforcement learning, we can use it for clinical decision support. So reinforcement learning, you train the model, then you're using it. If your use is good, you'll again say that, okay, it's fine, continue working. If your results are not appropriate or not up to expectation, you can again feed the model and it will go back. So sometimes it's called as back propagation. That is, model is already ready, results you do not agree, let's say you thumbs up, thumb down, that information will go back to neural networks, and then it will modify the algorithms based on the previous trees like decision tree or support vector machine. So bottom line is that it's not very costly to do machine learning. You need a computer and a software. Data is available everywhere. Collection of data is important, good quality data. You can do machine learning even in a primary healthcare center, depending on whether you are good enough in collecting the data and you are cautious enough in using appropriate algorithm and making appropriate interpretation from the data. Thank you.
Thank you, Dr. Debojiti, for your deliberations on machine learning. So any questions from the audiences or the speakers will be dealt with after the next session. Thank you. Can take the questions, Dada,
So,
the end of the session.
so there is a real world evidence, you know, a lot of data is coming. So the point is that now from the data, we have to collect some data, right? We have 1,000 data, but we cannot collect all the data, right? So there should have some, like in clinical trial, we have a criteria, no? These are the inclusion, these are the exclusion. So how to make the exclusion criteria in when I feed the data?
So when you in a real world scenario, when you have a lot of data, so how it is done is that you have a lot of data, you cannot use everything, will not fall in inclusion. So when you have data, but you need it in electronic form, you have the patient characteristic, you have the output, but let's say you have 50,000 data or electronic health record of, let's say, whole state. You want to find something specifically, but then you have to use something called as propensity score matching. So you give the criteria, okay, a patient with this age, this gender, this severity, or this body weight, BMI, this, you feed that into the system so that it can extract those patients only that are of those variables. So what are your variable interests? Like, for example, we do in RCT, that we do beforehand, that we say that this is the inclusion criteria. So that data mining is possible if it's an electronic record. And I think in the morning also, it was so that FDA approved. So those are from, let's say, Mayo Clinic or something, they have electronic records, they text mine it from those words, and then they make the groups like RCT. So it is said that propensity score matching does to observational study. It's an observational study, but analyzed as a RCT.
No. My question is that this is our human mind is doing, right? Propensity or whatever.
The propensity is
whatever, whatever inclusion, exclusion we are doing by our mind. But can we make that this is the data, whole data? So I want to do a study. So can you draw a hypothesis from this? After that, I will relate the causality and all. So means what I can say, can machine like a generative AI kind of looking at the data, they can fix something is coming out of them. Because whatever human mind we have prediction or hypothesis, then we can solve it by all those regression and all. That was this. But what our mind is thinking might be data is not doing. So we are running the data by from our mindset. Whatever hypothesis we want to draw, we want to prove. But can the machine looking at the data, they can bring out some hypothesis or something?
So that hypothesis generation, that is a function of artificial general intelligence that is being tried, that is still not achievable. They can give you insights into the data, but hypothesis still by artificial intelligence is still not possible. So that is the next frontier. So that is maybe possible after quantum computing, not now yet.
Thank you.
Thank you, Dr. Dibajyoti. Our next speaker is Dr. Sam T. Matthew. He is founder and CEO, SciWrite Global, independent consultant, medical communications and clinical regulatory writing. Dr. Matthew is a distinguished medical communication consultant with more than 20 years of industry experience, noted for his passion for AI-driven innovations in scholarly publishing. He has held leadership roles in top pharmaceutical companies and CROs such as Novartis, GSK, Biocon, Cactus Life Sciences, and Accenture, successfully building and guiding high-performing medical writing teams. Some of the honorary assignments of Dr. Sam T. Matthews are Director, Asian Council of Science Editors, Director, Asia Pacific Association of Medical Journal Editors, Associate Editor, Journal of Current Drug Delivery, Bentham Science Publishers USA, Editor, Science Editing, Korean Council of Science Editors, Seoul, Editor, Trends in Scholarly Publishing, Asian Council of Sciences Editor, Dubai. Committee membership, publication ethics and standard committees, and the Asian publication standards. So we proceed with Dr. Sam T. Matthews.
Thank you, madam, for that wonderful introduction. Good afternoon, everyone, and good morning, everyone, based on wherever you are in the world attending this conference. First of all, thank you very much for the organizers for having me here, especially Dr. Shivamurthy and the organizations for having me here and talk on the topic, the impact of artificial intelligence on academic writing and publishing, and exploration of challenges, tools, and technologies. So, I have three challenges while standing here. The first is, I'm not a medic. I'm not a medical pharmacologist. I'm a pharmacist. But at the same time, I understand that the topic is going to be, I'm going to talk on scholarly publishing. And I have seen most of your CVs and biodatas, and I understand that you have enormous number of publications. So the topic is quite familiar with you. And the second is, I've been listening to you all from morning, and it's more of science and research. And mine is going to be quite light. I'm going to talk about a little bit of tools and some ethics that are used in scholarly publishing. And the third is, I understand that this is the last session and you all are in a hurry to leave. So I will not take much of your time, maybe 15, 20 minutes, and I'll try to wind up within that. So a quick agenda, we will see why to publish. I know that most of, I have lots of published authors here. And why artificial intelligence intervention is needed in scholarly publishing. What are the major challenges? We will also see a little bit about what are the tools used in scholarly publishing, but we will not go into deep of those tools, considering the time constraint. So why to publish? Can I have some volunteers from the audience to see why do you publish? Yeah. Any thoughts from your side? Why do you publish? Basically for career advancements, right? And most of us do. And also we also wanted to have dissemination of our knowledge. Okay, and these are major things for personal satisfaction, secure funding sources, dissemination of knowledge, impact and recognition, and establish yourself as an expert. So there was a survey conducted almost 20 years back, somewhere in 1993, which actually found, it's around 20 years back, even that finds the same kind of findings that most of us publish for dissemination and career prospects. Having said that, our topic is about how artificial intelligence can help in scholarly publishing, in assisting scholarly publishing. But the question is, have we really understood artificial intelligence clearly? I'm so thankful to Dr. Dibyajyoti, who just spoke about artificial intelligence and machine learning. So most of the time, we combine or we misunderstood artificial intelligence and automation. But these are two different things. But the tools that we see in scholarly publishing are basically a combination of artificial intelligence as well as automation. So I don't want to go into the in depth of about artificial intelligence automation, which has already been covered now. But what is the future of artificial intelligence? So we hear about AI everywhere. And we also know that the future is artificial intelligence. So artificial intelligence has impact in every facets of our life. Look at some of the market data. We have already been spent around, or we are been spending more than $300 billion by 2026 in artificial intelligence. And most of this investment is also going into the AI-driven scientific research. So definitely medical research is also a part of it. Also, please look at the kind of growth that is going to have. So it is going to have a growth rate of 37% by 2030. We have seen in the morning session, the CAGR of monoclonal antibodies, that is going to be 12.5%. So imagine how fast the growth of artificial intelligence is going to happen in the world. So the future is nothing but AI. And definitely in medical writing, nothing but where the scholarly publishing is also a part of it, also have a significant importance in the market. So currently we have spent around close to $868 million in the market, and that is going to grow further at a rate of around 12.52% in the upcoming years. Also see that currently the US and the West are dominating in artificial intelligence, especially in the medical writing. At the same time, the Asia Pacific, Asia, and other Asian countries are also contributing significantly into that, and they have significant investment in the development of artificial intelligence tools. So why do you think we need to have AI intervention or artificial intelligence tools in scholarly publishing? Any thoughts? Why do we need to have AI tools in the scholarly publishing? Look at the data here. So this actually shows the number of publications that we had for the last 8 years. That is from 2014 to 2022, last 8 years. So you see that there is a significant increase in the published papers in the biomedical area, around 60% increase in the number of papers. And imagine this is just the number of papers that are published. But imagine the number of publication activities that would have happened. At least 50% of that would have been rejected, or double of this would have been rejected. And for writing a paper or a paper to get published or to submit, there are multiple other steps that would have happened. Someone would have written it, someone would have peer reviewed, you have submitted to a journal, it has undergone multiple revisions, all of that. And that means a lot of activities are happening in the scholarly publishing area. And do you think that all these activities need to be done by a qualified researcher, or some of these activities can also be done by a machine if it is programmed very well? It can save a lot of time and it can save a lot of money, of course. And most of you are the peer reviewers and editors of journals. You would have reviewed a lot of papers. But have you ever quantified, and we know that peer review is a service to the, service to what? Service to the profession. And have you ever tried to quantify the time that you have spent on peer reviewing? If you do that, it's an enormous amount. If you do that, there is a study which was published sometime in 2020 about the time spent by different peer reviewers in biomedical research. If you look at it in 2019, from the US, it was around 1.5 billion, more than 1.5 billion. And from the UK researchers or the European research, it is more than 400 million. So that means the time that you spend for a peer review is quite huge in terms of money. So when the money is so precious, the time is so precious, if you can leverage certain tools to support your scholarly publishing, that would be a great advantage. So artificial intelligence is helpful to almost all the stakeholders in scholarly publishing. So who are those stakeholders in scholarly publishing? The first and foremost is the author, or none other than the writer. Then comes the journal editor, the reviewer, and then publisher. And there are different steps in a scholarly publishing, right? And all these steps, there are tools that supports you to publish your papers. The initiation, I'm not going into deep of what are the different steps in initiation, then development, submission, and review and production. So all these steps, all the sub-steps under that have different tools that will help you to ethically write a paper. So let's come to manuscript initiation, nothing but literature review and data synthesis. So as an author, as a researcher, or as a writer, when I write a paper, for me, the biggest burdensome process in my writing a paper is doing the initial literature review, identifying the lacuna, writing the hypothesis, and the second part is writing the discussion part. So do you think that anytime any tools can support you in identifying the appropriate literature, extract the data required, and put it in the format and cite them appropriately? So there are tools which can be used for literature review, like COVID Scholar, Iris AI, Robot Reviewer, and Scholar C. Some of these are not, currently used widely, but Robot Reviewer is also used very widely at the moment, and Scholar C. There are many other tools. I will not go into deep of the tools, as I mentioned in the beginning, because of the time constraint. But out of the other tools like Semantic Scholar, Elicit, Cite AI, Consensus, Ryan, and SciSpace. SciSpace is a very common tool which most of the researchers use nowadays. That not only does the literature review, it also extract the data, cite the data, and even paraphrase the data for you. It will also identify plagiarism. So development of manuscript, writing and editing. I'm sure most of you have used multiple editing tools, writing tools like Grammarly or Pro Writing Aid, etc. So there are some other tools which are much more higher in nature, which can actually develop a significant amount of content for your papers, like Pro Writing Aid, Trinka, Perfectit, and Synode Manuscript Writer. So these all have different roles. Some of them are only the grammar checkers, some of them only paraphrase, some of them even write the content, full content for you based on your prompt. And also for manuscript writing and editing, we have another tools, ChatGPT. I know that all of us are very much fascinated about ChatGPT. And then Curie. Curie is from Nature Science and a very good tool for writing your papers. Then Quillbot. Quillbot is another wonderful tool which can help you to write papers, paraphrase it, add appropriate references, citations, and all of that. And then Paperpal. Paperpal is also a very academically focused research tool that can be used to write your papers. So reference management, definitely we all have used many tools in reference management like Zotero, Mendeley, and EndNote. All of them have their own positive things. Zotero is an open source, Mendeley is a free one, but EndNote is a subscription based. And all of three are equally good, but EndNote is much more professional, and further professional organization usually use EndNote. Another important challenge for any medical writer, any writer or a researcher is to identify the appropriate journal. And nowadays we know that there are lots of fraudulent publications. So unknowingly or knowingly we get published in predatory journals. That is something which will spoil your career. And also identifying an appropriate paper is also, appropriate journal is also very, very important. I used to tell that there is no research, if it is purposefully conducted, cannot be published. That means if a properly conducted research can be published, only thing is you need to identify an appropriate journal for the paper. Okay, so there are tools that will help you to identify the appropriate journal for your papers. The tools such as Manuscript Matcher, Find Journal, Journal Finder and Open Access Journal Finder. These are different tools and web-based applications which will help you to identify different journals suitable for your for your paper. But at the same time, one pitfall for this is most of these are
Sir, sorry for the interruption. 5 minutes left.
Okay.
Fine.
So most of these are actually the proprietary of most of the publishing houses. So generally they prefer their own journals. So plagiarism detection, I am sure that you all are aware of plagiarism detection and have used the tools like Paper Rater, even Grammarly, Authenticate, or CopyLeaks. Peer review and quality checks within the journal, they have different tools that will actually help you to do peer review, like Start Reviewer, PineLop, and AIRA AI. This basically analyze the articles for the preliminary inclusion into the peer review stages, and it will also help and understand what are the different aspects of the paper, whether this is suitable for the specific journal which is being submitted. And image check, any image falsification which is done in a paper can also be identified by different tools such as FigCheck, ImaCheck, and Proofing. Also within the journal publishing, the publishing houses use different tools like Author1, SmartEdit, PubStart, and Unsilo to do their workflow management, including until the paper is published and even after publishing, the dissemination of the paper. So having said that, we still think whenever we usually talk to the academician, they basically think that, okay, IA is coming in, we will wait and see how things are evolving. But that's not true. Already a machine-generated book is already ready by 2019. That means around 6 years back, which was published by Springer Nature. And you can see the authors that are mentioned there. See the artifacts of this paper. The author is mentioned as beta writer. That is again very debatable one, whether an AE can be an author or not, but still this is something happened in 2019. We are living in the era of generative AIs, okay? I'm sure that you all are aware of these generative AIs, at least ChatGPT, DeepSeek, Deep Research, Gemini, Grok, Cloud, etc. So, and most of us also have used ChatGPT. So do you think that ChatGPT can be used for writing your paper? Have you ever asked ChatGPT about it? Because ChatGPT is one source of information for us, right? So here you see Todd Carpenter, who is an editor of a Scholarly Kitchen, have asked ChatGPT that, can AI write a scientific paper? So what he said is, yes, I can write a paper, ChatGPT said, but you should be, it is important that once you write a paper, you should transparently disclose that I have used AI to write the paper. Similarly, he also asked, is it writing a paper with ChatGPT is ethical? ChatGPT said ethics depends on you. Ethics is very individual. Okay, if you transparently disclose in the paper, in the acknowledgement, or in the methodology section that I have used ChatGPT for getting this data, then it is fair enough. And also, it is important that if whatever the tool you use, as a researcher, it's your responsibility to ensure that the data that you presented is correct. So as long as you are doing that, there is no ethical concerns. Look at these 3 papers. So the first one is published. First one is a preprint server. There you can see the authors as ChatGPT. Even the second one in the nurses education and practice, the author is ChatGPT. And the third one, again, one of the authors is ChatGPT. And most of this paper you would have seen, it is happened somewhere in 2022 and 21 and towards the end of 2022-21. But see the last paper, which is nothing but a paper which is published from the preprint server. When that is published in a journal, the author name is removed, as ChatGPT as an author is removed. So that is because of the recent guidelines that are set up by different editorial organizations. So what are the major challenges of using artificial intelligence in scholarly publishing? The first and foremost is unethical AI use. AI can fabricate things. You all must have heard about AI hallucination. Okay, and ChatGPT itself says ChatGPT's accuracy is only 87 to 88%. So in medical research or any other research, we cannot have a even single 0.001% of error. And hallucination issue, which I have already explained, and bias and discrimination. Today, Dr. Subhajit has mentioned about the data. So the output of any AI tool depends on the data that you have fed into. So as long as your data is not correct, so it may not provide you the appropriate output. Plagiarism and authenticity, still this is a debatable topic among the journal editors. And we also have data privacy, because most of the data that you give to a ChatGPT or any other AI generative tools are going to their servers. So the data privacy is also under question. And of course, research integrity. A researcher is supposed to do the research and do their own research findings. But if you ask ChatGPT to do the research and get the findings, then definitely the research integrity will be impacted. So to overcome this, there are lots of guidelines from different editorial organizations, as well as from different top publishers, like World Association of Journal Editors, American Medical Association, International Committee on Medical Journal Editors, and all of that. I'm a member of some of them, and I do have editorial position in some of those editorial boards. If you look at the guidelines differ from each of them, but overall what they say is AI cannot be an author. And if at all you use any AI tool for writing your paper, you should be disclosing it upfront, either in the method section or in the acknowledgement. And always the author is responsible for the accuracy of the data which they have published or which they have submitted. So whatever the data the AI tool gives you, you should always review as an author and ensure that the data is accurate and correct. So what is happening is we are too late in getting into artificial intelligence and embracing these technologies into scholarly publishing. Human-machine collaboration is something which is real, and we have to embrace that. So with that, thank you, and I'm happy to take any questions if you have.
Thank you, sir. It was a very nice, elaborative, and informative presentations. But due to the times was very time constraint, where we could have due to the time constraint, where we could not continue much here today. So, any questions from the audience?
Otherwise, I'll ask a question. Any of you have tried using any of the tools for writing your papers? It's not unethical at all. Don't be ashamed. Any of you have tried it? If not, please go ahead and do it. Experiment it. Because if you don't do it now, you will lag behind with the technology. Technology is just growing like anything. So whatever I presented something related in the 4 weeks back at some other place, I have to revise my topic today because the tools have changed. Some tools are obsolete now. So it's very, very, very dynamic. If we don't follow them, if we don't use them, we will lag behind in publishing our research. Thank you. Thank you.
Thank you, sir, for the wonderful presentation. Now I request the chairpersons to kindly felicitate the speaker of this session with a memento and certificates. I also invite the speakers to come up on the stage. Sir. I invite all the speakers to come up of the stage. Now I request Dr. Dibajyoti Saikya sir to felicitate our chairpersons.
Good afternoon, everyone. So now we are starting valedictory session. It is my great honor to stand before you as we celebrate the academic excellence and innovation that defined MPSCON 2025. So now I invite on stage Dr. Satyajit Mahapatra, Dr. Shivamurthy, and Dr. Melvin, Treasurer MPS, and Dr. Fullan up on the stage. Now let us begin to recognize and applaud the exceptional contributions among you. So let us begin with postgraduate poster presentation category. The third prize winner is Dr. Jamira Liza Thakakchan. May I welcome you up on the stage? The second prize winner is Dr. Shankar Jyoti Bora. Yes, the postgraduate.
So we would like to, mention here that, the first prize winner will get 2,500 rupees as the prize money through the MPS Society. So congratulate the first by your up.
I welcome the second prize winner, Shankar Jyoti Bora, up on the stage. First prize winner has already taken the certificate and the memento, Dr. Sanjaya Dhal. Okay, so upcoming next for faculty poster presentation category, I would like to invite for second prize winner, Dr. Sonali P. Suryavanshi, up on the stage. First prize goes to Dr. Isabella Topno. I would like to invite on stage. For appreciation, I would like to invite Dr. Surender Guram and Dr. Rajkumari Mazumdar up on the stage. Moving on to postgraduate oral presentation category, the third prize winner is Dr. Bishnupriya. I would like to invite on stage Dr. Bishnupriya N. She has already taken her certificate and memento. Moving on, second prize winner, Dr. Vandana Thapa. I would like to invite her on stage. The first prize winner goes to Dr. Abhinav Tiwari. I would like to call him on stage. Finally, in the faculty oral presentation category, the third prize winner is P. Anshuman Abhishek. I would like to invite on stage the second prize winner goes to Dr. Sukanya Borgohain. I would like to call her up on stage. The first prize goes to Dr. Govind Mishra. They have already taken their memento. Okay, moving on. For undergraduate poster presentation category, the third prize winner is Shushunt Yogesh. I would like to call him on stage. Okay, second prize.
I would like to tell that, like, he is my student, and it was a completely blinded evaluation. And only when I got the results, only I know. So on behalf of him, I will kindly accept this and I hand over to him.
The second prize goes to Ramadrish Mab Banerjee. I would like to call him on stage. The first prize goes to Sheikh Ryan. I would like to invite him on stage. Let us give a big round of applause for all the participants. Thank you and congratulations once again. Now, I would like to invite the organizing committee. We would like to appreciate the organizing committee. So first, I would like to give the certificate of appreciation to Dr. Satyajit Mahapatra.
Dr. Karanchen Singh, organizing committee member. At a moment to madam Dr. Debopam Das. Akanksha, তুমি ইয়াতে থাকিবা বা সবে না তুমি ইয়াতে থাকা জাস্ট এনেকে দিম ধরে ভালকে ধরা না কি হাত খুঁজছে সব ডক্টর সুরিন্দর জি Debopam, there is moment of Dr. Rajkumari Majumdar. Dr. Mukundam Bora. I think he's not here. Dr. Saran Mulla. Dr. Krishna Vishwas. Dr. Junmoni Lahon. Pase. Organizing committee member. Are we going to Dr. Vilas Goel. Dr. Jayanto Das. Dr. Kasif Ahmed. Dr. Dibojyoti Soikia. Dr. Indrajit Nath. Miss Akanksha Chakraborty. Miss Akanksha Chakraborty. Photo loba ha. Take photo. Mrs. Pranami Bordoloi. Especially quoting. Okay. So we are very thankful to Madam, because like that is the highest order of actually service she has provided for us and help actually. So thank you, Madam. Mr. Sandipan Das. Mr. Dhiren Roy. Dhiren from Audio Visual. Mr. Mohendra Sorori. Mohendra from Audio Visual. Mr. Dr. Amit Sonkar. Ms. Jayashree Kalita. Dr. Bipasa Kalita. Dr. Parthapati Medhi. Dr. Amit Kumar Malik. Sir. Thank you, sir. Dr. Praveen K. Associate Professor, Department of Anatomy. Thank you, sir. Dr. Rituparna Setia. Dr. Anusha Bhattacharjee. Dr. Samarendra Burman. Dr. Sukanya Buragohai. Professor Bhupen Burman. Mr. Shankar Jyoti Bora. Moment. So next, Dr. Junmoni Lahan, Co-Organizing Secretary, MPSCON2025. Dr. Junmoni Lahan, Co-Organizing Secretary. No, वो दे दिया है already. That is already given. That is all. Momento is already given in this case. Momento दे दिया था. Co-organizing secretary. Dr. Dibujyoti Soikia, co-organizing secretary. Dr. Shivmurthy and joint organizing secretary. Dr. Satyajit Mahapatra, sir, organizing chairman. Already organ.
So, the most important person now, Dr. Phulan Sharma, who is behind the, whole, like at the backbone of this conference, Dr. Phulan Sharma. I want everyone to, stand up and give him a standing ovation and, long one. So let us celebrate his, success, and his success is our success too. And the entire team, please, all of you come inside, na? So we will give along with you. Entire team, please come inside. So all of you, all the team members, please come inside on the stage. All of you arrange yourself. We'll take a one organizing committee good photograph. Please, come front also. Yeah, I would like to appreciate the contribution to Society of Medical Pharmacology Society by the AIIMS Guwahati Department of Pharmacology. So big congratulations and clap for the organizing committee who worked day and night. And I feel that this is one of the great conference, even in the war-like situation. If we can make this, I think according to me, only one person who was supposed to be here, she did not come. She is a postgraduate student from Delhi. Only that person did not come, and including even Sushant Tripathi sir, Santanu Tripathi sir, who even reached in the end. That is also. I would like to thank all the sponsors also, two people. Dr. Saurav Deka from his organization, and Dr. Pinkways Ashok Kalita, so who agreed to partner with us and support our participants for the local trips and all. So in that way, I think we conclude we would like to appreciate the efforts of everyone who took so much of effort since last, I think, 3 months. 3 months, day and night, everyone has worked, and they all made us proud. Okay, MPS society will be, is being appreciated by every participant. Like whoever come in contact with me, they all said that the MPS society has done a great job, and everyone appreciated the efforts of the local committee members and the entire technical team, IT team, administrative team, and like senior management. I would like to thank from the bottom of my heart, who really stood behind us and gave this wonderful facility for organizing our second international conference, MPSCON 2025. I declare this as a grand success. And I would like to, clap for all of you from my, whatever the best way appreciation, I don't know, I can't tell, I don't have words to express. So I thank everyone who are behind this success, and we are really indebted with your support, and even such a difficult situation, everyone came and contributed, and no lectures were cancelled. No lectures were cancelled, no paper presentation cancelled, no, everything. I think we are ending on time also. It is not too far from the time what we designed, and it entirely went well. Thank you so much, and my heartfelt appreciation to everyone. My So I would like to take an opportunity to appreciate my, office bearers, Dr. Pulan Sharma, of course, and Dr. Melvin George, who is always, both of them are there with me. And in addition, we have Dr. Madhav Rao on stage, who is also our office bearer. Dr. Akash Gargade is our office bearer. And then we have Dr. Baswarna, Dr. Sapna Patil, and anyone else? So I think totally Dr. Prakash BM, yes, Dr. Prakash BM, he is the professor of pharmacology at Andaman Nicobar. So I would like to, thank everyone for their wholehearted support, continued support. I think we are on the 10th year of our, operations, MPS. We have grown from zero balance to 15 plus lakhs of rupees in the account just for serving all of you. Okay, the MPS Society is growing. We have 3 books in our account, and we have conducted more than 20 workshops, and we have conducted almost 5 big conferences. This is our national conferences, third national conference, in fact. So with this, we are, we are going with great energy, and we are writing, crucial pharmacology trends, 3 and 4. I believe that it will become 2, 3, and 5 also. No, 3, 4, 5. Most probably because we have 118 chapters that are coming in. And it's a great energy. Every pharmacologist throughout throughout India, they are united through this, mission, and they are contributing for the chapters in various manner. And this will be these books will be the Bible for all of you. So we are sure that these books will become Bible for all your future studies. Okay, so we may come up with we have the aim that we would like to cover all the chapters, all the topics that are asked in your examination. So, and these questions are derived from your question papers throughout the country. Okay, so in that way we are on a big mission, and we will ensure that you don't need to worry from where do I study, from where do I get an opportunity to learn. So you will get an opportunity to learn from Medical Pharmacology Society, and we are working towards ensuring that every MD pharmacologist go proudly and say that they are MD pharmacologists. I can see such messages coming in LinkedIn every day, and once in a while, once in 2 weeks, once in a while, I see that proudly MD pharmacologists saying that I took MD pharmacology, and they proudly confirm, say that I'm an MD pharmacology, and they explain also. So I have so many messages in my, I have taken the screenshots also. So in this way, we would like to, before concluding, I would like to say that next conference will be in our institution, Dr. Chandrama Dayanand Sagar Institute of Medical Sciences and Research at Devara Kaggalahalli, Harohalli, Kanakpura Road, Ramanagara District, Karnataka, India. Okay, this will be your next destination, and I request all of your, continued support and entire, support for conducting the next conference successfully. And I invite you all and welcome. I will looking forward to welcome you all at my destination, that is CD Saimer, Ramnagara district. Thank you.
Punjab, Sindh, Gujarat, Maratha, Dravida, Uttara, Banga, Vindhya, Himachal, Yamuna, Ganga, Ujjala, Jagati, Taranga, Ashubha Name.